timely, fast, and precise diagnosis. Our goal is to make our analyzers even faster and more reliable for doctors, laboratories, and patients like Diana. And we constantly innovate and develop new tests to obtain results for conditions which could not be tested for previously, or more information about diseases that can help doctors make better decisions. Benefit of patients around the world, and to help alleviate healthcare cost burden. Doing now what patients need next.
Yeah, so once again, this in vitro diagnostic, as we all know, in vitro means in the lab or outside of the body, whatever test or diagnostics are done that are in vitro diagnostic devices. And these are the tests which are performed on the sample taken from the human body. As we all know, in COVID also, probably the most test we have seen was the rapid antigen test, and which used the swab of the mucus inside the nose. And apart from that, many of the tests available in market right now, they use the blood sample or various samples like urine sample. So these tests, they usually require the human sample to be taken. And in current research stage, the tears are also used as a sample currently. So I'll use the pointer here. Yeah, and these IVDs, they are used to detect the disease or in other condition that can be used as to monitor the person's overall health, which can help to treat or cure the patient at the earliest possible. So these are like for early detection, these can be used. In the in vitro diagnostic, basically we have seen the two major categories, the lab developed test and the in vitro diagnostics. Most of the time, what we focus on when we go to pathology lab or any physician who suggests us to do the blood test or urine test done, then those are submitted to pathology lab, and these are laboratory developed tests, because the sample is taken by the pathology expert. He processes the sample, carries out the test, and then he predicts the result or interprets the result. In case of in vitro diagnostic, not necessarily that pathology expert may be doing the test, but the surgeon, physician in his clinic, or even patients at home can do this test, and these are called as the in vitro diagnostic test. So LDTs are developed and validated by the individual testing laboratory, and which is subjected to the regulation of CLIA. The CLIA is Clinical Laboratory Improvement Act, which is global act which regulates the LDTs across the globe about their performance, their standard operating procedures of doing the test and analysis. In case of IVDs, these are developed by the device manufacturer. So a lot of R&D is done, and in that research and development process, the test or the assay is developed, and then the product is developed by the manufacturer, and which is subjected to review by the regulatory authority. So there are various regulatory authorities globally, or now also in India, which regulates the manufacturing, import, sales of these medical devices, or in the subcategory that is in vitro diagnostics. So a small review about lab developed test is, these are in the single laboratory. You just can imagine the blood test that is done in any pathological laboratory. So all the procedure is developed by the lab personnel there itself, and then he carries out the test as per their standard operating procedure in the lab indigenously. And in the past, most of the LDTs were relatively for simple screening, all right, and then also for advanced or multiple, like in one blood sample they could get analysis of multiple analytes in that sample. And in the recent years, these have been developed for a wider range of conditions, right? That, as I said, multiple analytes can be detected in the blood sample, and recently the developments are also there, whereas in case of cancer also the detection could be possible using the LDTs. Then coming to the IVDs. So what is the IVD? By definition, the IVDs are any medical device which is reagent product, a calibrator, control material, any kit, instrument, apparatus, piece of equipment, software or system, whether to be used alone or in combination. Okay, so all these sub points or subcategories are considered, like sir said in early introduction, that softwares are also now being considered as medical devices. So yes, this is already implemented globally, that various softwares that are used for medical devices are also considered as medical devices itself. And as the definition says, it is in combination. So combination can also be there in these medical devices and in vitro diagnostics. So these IVDs are used to identify the presence of specific markers or analytes in blood, urine, TA sample, or any other physiological sample, such as pathogen or circulating protein like various interleukins and cytokines they can detect. Or in case of genomic alterations such as gene mutations or chromosomal copy number detection or RNA transcript, or the simple like in case of COVID, the PCR was also made in IVD category as well. So these are the huge number of applications for these IVDs. Now coming to the comparison of these two techniques, one is IVD we are talking, we'll be discussing more in detail about IVDs only, but as it started with LDT, we have to consider them as a comparative parameter with IVDs. So the first parameter comes is a regulatory agency. For IVDs, it is FDA, US FDA. So probably most of the time I'm talking about the global regulatory. For Indian regulatory, I'll be talking in the second part of the presentation. In case of LDT, it is Centers for Medicare and Medicaid Services, which is CMS. For development and manufacturing, these IVDs are manufactured by the device manufacturer, while in LDTs, it is single laboratory which has all the right to develop the SOP and interpret the results. The marketing authorization required for IVDs is through FDA and 520 FDA&C Act under Section 520. In case of LDT, it is CMS again, or the CLIA standard. For quality system, it is required for IVDs, as there may be quality management system implementation requirement for design control, manufacturing control, and complaint handling for these devices. As you know, the safety and efficiency is important for any devices, medical devices or in vitro diagnostic devices. But in case of LDT, it is not required because it is indigenously or individually developed system. In case of IVDs, the clinical validation is required, but for LDTs, it is optional. But the analytical validation is required in both cases, that is IVDs and LDTs. For global reach, yes, IVDs are currently having a huge application in all the diagnostic related field, and that's why global reach or distribution is possible for IVDs. But LDT, it is not possible because it is simply in-house testing domain. So the talk I'll be presenting mostly today is about the lateral flow assay, right? So in case of the simple example could be given for lateral flow immunoassay is, previously it was pregnancy test kit, wherein the pregnancy in women can be detected by just putting the urine sample on the test strip. If the two lines are forming, the test is positive. If only one line is forming, the test is negative. If both lines are not there, then the test is invalid and the individual need to do another test, right? So how this works is presented through the schematics in this video. So I'll be pausing this video at some point of time because there are a lot of description, and this is the important video for the understanding the concept of lateral flow immunoassay, how the immunocomplex works and how the biomarkers work. So what we see in market or when we get any rapid antigen test kit or pregnancy test kit, it looks like this, but what goes inside this is important. So this is the lateral flow immunoassay strip, all right, and there are various components present on the strip. So these are the different components, starting from one end to another end. The sample pad, wherein your sample will be put. The conjugate pad, which holds the labels or the antibodies conjugated to gold nanoparticles or any dye. Then the nitrocellulose membrane, which is very important for detection purpose because the capture antibodies are immobilized on the nitrocellulose membrane itself, and in the end there is a absorbent pad which pulls all the liquid or the sample specimen through the strip and act as a holder, sample holder later on, so that the capillary movement occurs appropriately. So the one which we are seeing is a cassette format. As we have seen, the plastic cassette is there which opens up and then these strips are visible. But there are dipsticks also wherein only plastic backing membrane dipsticks are available, which need to be dipped in a Eppendorf tube or any test tube or any sample holder tube. Yeah, so this is the cassette, and the sample can be different depending on its application, from blood to milk in veterinary applications, or urine, serum, plant material, or food. So these lateral flow immunoassays have huge application, and they are major contributor in the IVDs, that is in vitro diagnostics. So how this works? Your sample goes in the sample well, then it starts moving because of the capillary force. Yes, and the conjugate membrane, there are conjugates which are antibodies conjugated to the markers. The markers may be gold nanoparticles or quantum dots or any other color based molecules. So you see in this particular schematics, a control line is there and two test lines are there. So that can be developed for the multiple or multiplex assays. Yes, so this is the immunocomplex which is developed, wherein I was talking the capture antibody, it is on the nitrocellulose membrane. The target is present in your sample analyte, and this particular yellow and red part is your bioconjugate, where the red is a color marker, that is colloidal gold nanoparticle in most of the cases, but I have worked on the fluorescent particle, that is quantum dots as well, and the antibody is conjugated to this particle. So when this sandwich immunoassay forms, sandwich means there are two antibodies, that is capture antibody and detection antibody, among which the target is bound. So when this target is present, then only this sandwich will form and the color will be appearing on the test and control line. Yes, so this is how the result is interpreted easily. Within 10 to 15 minutes, 5 to 10 is the mentioned one, but 10 to 15 minutes, the assay is completed and we can consider the results. So the application, you see that these are qualitative, could be developed into semi-quantitative or quantitative, wherein the readers are developed. Like I developed the portable fluorescence reader for detection of the fluorescence, because fluorescence is not visible to the naked eye as it is in case of pregnancy test kit or in case of rapid antigen test for COVID. So those are qualitative one. And if you need to go for quantitative, you need to have the reader for that. And there are applications in healthcare, animal health, food safety, plant health and environment, and so on and so forth. So coming to the lab, now these schematics you have seen, but how the material is actually assembled on the plastic backing membrane for development of this lateral flow immunoassay strip is shown in this video again. So this is how it looks. The membranes that we talk: absorbent pad, nitrocellulose membrane, gold conjugate pad, and then the sample pad. Gold conjugate pad appears purple in color. This is because of the gold conjugates that are immobilized in the paper. So there is no audio in this, only video is there. So I'll be explaining meanwhile. So is this video visible to everyone? Just confirm once, because I don't see any response long. If any question is there, that also can be discussed right away.
Yes.
All right.
It's visible.
Yeah. So as you see that there are different rows wherein the plastic membrane is there, and these are stickers. So that can be removed and the membrane can be put there. So these are different membranes. So this is the major membrane. So see that is how the sticker is removed in the center part. And this is the nitrocellulose membrane, which is assembled in the center. And these are then cut into small parts. So that will be also be shown in some final industry video where it preparation of the strips is shown. So this is assembly of the absorbent pad at the another end. This is conjugate pad and this is sample pad. And then finally the sample pad is assembled at one end. Then we do the antibody immobilization on nitrocellulose membrane using this syringe pump. So this is how it is assembled, and they will be then cut into the 4 mm or 5 mm width as per requirement. Coming back to our presentation about IVDs, so whatever we have seen is a lateral flow immunoassay, which is a part of the IVDs, and IVDs are the part of the medical devices. So these IVDs are used in screening process, diagnosis, prognosis, stratification, or in monitoring. And the techniques that are involved in development of these IVDs, so the principle or mechanism, they involve the clinical chemistry panels that are like colorimetric panels or microbiology cultures, immunoassays, which are either colorimetric or fluorescence based, then molecular application including PCR based assays or in situ hybridization. So these are various type of techniques which are used in development of the IVDs. Now coming to the classification of these IVDs, as per Indian regulation, we have now got these categorized into 4 categories, that is category A, B, C, and D. So depending on the risk level, these categories are developed. So starting from the low risk, low to moderate risk, moderate high risk, and then finally high risk, which are class D devices. The example if we take in low risk and low moderate risk are various instruments used in diagnostics or nucleic acid extraction reagents or the control reagent. In class B, which is low moderate risk, it is clinical chemistry or pregnancy self-testing strips or urine test strips are considered. So class A and B are probably similarly could be called as a low risk in vitro diagnostic devices. But in case of C and D, these are high risk because somehow the infectious agents are involved in that. If like blood or any serum sample which has particular infection to be detected, and if any technique is developed for them, then it is class C and D. I'm particularly talking about the IVDs. Overall, the medical devices are also classified in the same parameter depending on the risk. So there are the different tests like genetic test or prostate antigen screening, then cancer marker or blood glucose meter or strip, which are also very common nowadays because most of the diabetic patients, they do know about the blood glucose meter or the strips that are available. The glucometer is there, which most of the diabetic patients use for easy analysis or glucose monitoring. Then the hepatitis B virus, C virus, HIV, and then blood grouping reagents. So all these are categorized into class D as they pose the highest risk to the human health if it is infected. So I hope everyone is clear about the classification, which is risk-based classification. From A to D, risk increases. Now, in case of IVDs, if error happens, suppose there are patients which are undergoing the testing and there is problem with the IVD devices. So what could happen is, first is it can be false positive. That means the test result is abnormal even though the patient is not ill. Okay, so your test has some problem which is showing you positive result, but the patient is absolutely fine, right? In other case, it is false negative, which in case test result shows it is not abnormal, but the patient is ill. All right. So I hope you understand what is false positive and false negative. So in both of the cases, the interpreted information may be non-specific or inaccurate, which may lead to the consequences for patient. In case of false positive, you may have to undergo the unnecessary blood draw for further testing, or the wrong therapies with medicine or even surgical interventions may be required in that case, but which is based on the false positive result. In case of false negative, what could happen is that could lead to the critical delays and to urgently needed therapies or even impact of the public health because it is not detected earlier as the results were false negative. So these could be problems for in case of in vitro diagnostics. I think there is interruption from someone. Can you please put everyone's mic on mute mode? Yeah. So moving forward to the IVD development process, right? So everything in IVDs work in sync with analysis, design, development, and the delivery of the final product. Coming to the complete development process, as I'll show you one slide wherein it is compared with the therapeutic development process as well. But in case of IVD development process, the major focus is demonstrating that the developed assay's analytical performance reflects its capability of detecting the analyte in accurate and reproducible manner. Whatever test we are developing, they should be accurate and reproducible in terms of results or in terms of similar analyte detected repetitively over the period of time, and that is called as a stability study. So herein there are four phases. Phase 0, which starts with the business proposal. Of course, all the funding and all is involved here in this case. Phase 1 is to check up with the feasibility of the assay. Then phase 2A and B are the two parts wherein the design is proposed and the work is done with verification of the specification required. Then phase 3, wherein validation such as clinical validation or analytical performance is evaluated. And in phase 4, it depends on the commercial and commercialization of the product and the feedback from the market. So this is the overall development process for any IVDs. As I discussed, as compared to the therapeutic development process, which takes approximately 7 to 15 years, that depends on the requirements as well. Whereas in comparatively, this process of diagnostic development is little quicker, that is 4 to 7 years. And in case of COVID, you might have seen the rapid antigen tests were developed within the period of 1 year or less than that. So this is the speed with which the diagnostic or the in vitro diagnostic development process can happen. And certainly there is a factor about safety and efficiency or efficacy of those medical devices. And as these are in vitro, so considering the safety and efficiency, they are little bit relaxing in terms of safety. So that's why the process may take the little lesser time than the therapeutic development process. Yeah, coming to the waterfall model for the device development, any device development, we just consider these factors are important, wherein the user need is kept at the peak, and then we design our devices like input, design process is there, then design output with all the specification is tested, and the specifications are verified with the input of the design. And then the final medical devices which is developed is checked for the user need, which confirms its validation. All right, so this is the waterfall model for checking the device development process accepted globally. So it varies like industry to industry or device to device development, wherein all these processes are optimized as per their own requirement, and several other checks may also be kept in between these processes. Coming to the performance evaluation. So as I was talking, development of any IVD, we have to check or evaluate its performance. So what are the parameters that we need to confirm or check after the development for the evaluation? Is there are basically three parameters. One is its clinical performance, its analytical performance, and its scientific validity. So as this diagram shows, there is a three-dimensional approach wherein one is a disease, another is a method, and third is a analyte. So again, coming to the COVID, So COVID is a disease, analyte is antigen, and method is a lateral flow rapid test we have developed. I'm just giving you examples so that it becomes easy to understand these performance evaluation parameters. So in case of scientific validity, we have to just check the association of the analyte, right? That is antigenic marker with the disease, right? If these two are correct, then the scientific validity is put there. Then in case of analytical performance, the method and analyte. So if antigen is present, your assay should be able to detect it, right? And that proves your analytical performance. And the clinical performance in the end, wherein your lateral flow test or rapid test confirms the presence of the COVID. So if it is positive, that means COVID is there. So this is how, in putting in a simple words, these performances evaluation can be taken into care: scientific validity, analytical performance, and clinical performance. Now the global regulation I'm discussing here, because you can see there are class 1, 2, and 3 risk-based devices are there. In Indian scenario, we have A, B, C, D. Okay. In globally or particularly in US, it is class 1, 2, 3, where class 1 is low risk, 2 is medium risk, and 3 is high risk. All right. And that is also internationally followed classification is risk based. In Australia or in European countries, this varies, like class 1, then European class 2A and 2B are there, and then class 3 is there. In US, it is class 1 to 3 only. So it depends on country to country, but globally it is risk-based classification. So how they are regulated is important here. So I have taken one particular example because US FDA comes first in development of these devices in internationally, right? So there is one process, right? 510K. Okay. So if your device is low risk, then you can apply for the exemption from that 510K process. Okay. So it is a simple application process to US FDA that we are developing some device which is probably not very new device. Someone has already developed, or predicate device, or the sample device is already in market. We are just developing something similar. In that case, you can seek the 510K exemption, but the device will be low risk in that case. For the medium risk, it will be 510K process required. Okay, so you have to fill up all the parameters in the 510K, apply to US FDA, and get it approved. Okay, so the example is pregnancy test kit. In case of high risk devices, you have to go for PMA, that is pre-market approval, which is another process by US FDA to be followed if the device is high risk and to be approved. Right? And the example in this case is genetic test used to select the cancer therapy. So depending on the increase in the risk, these processes need to be followed. That is 510K exemption if it is low risk, 510K follow-up if it is medium risk, and pre-market approval if it is high risk devices. All right. So what is 510K approval? As we have discussed, it is FDA process. So it is required to demonstrate that the product or device or assay or any IVD that is developed is equivalent to pre-existing or predicate device, as I discussed. So if it is similar to something which is existing, that is already cleared by FDA for the same intended use, and if the product or device is not substantially equivalent, right? If it is not substantially equivalent to the predicate device, then the sponsor or manufacturer or developer has to seek the approval of device via de novo 510K pathway. De novo means new process approval for the 510K pathway by submitting a PMA application, which is pre-market approval. PMA is a FDA process of scientific and regulatory review to evaluate the safety and effectiveness of class 3 medical devices. So only in case of high risk medical devices, this PMA is required. All right. I hope everyone is clear about it. If any question can be asked now, I'll take pause for a minute or so. So if any question till now. All right. So continuing to, am I audible? Just to confirm, hello?
Yes, sir. Very clear.
Yes. So this is the schematic which again shows how the device comes to the market. So if the device is low risk, medium risk, or high risk. Of course, there will be general control, so you can consider this as a filters, various filters. Okay. If your device is class 1, that is low risk, of course the general controls will be there, but then you can ask for exemption from the 510K process and it can get the clearance for commercialization. If it is not exempted, you have to seek the 510K approval. Again, go to the 510K process, which will prove if it has substantial equivalence to the predicate or existing device. If it is there, then it is approved. If not, then you have to go for the clinical approval and then the PMA approval, which is default in case of high risk devices, that is class 3 devices, general control, special control, clinical data, pre-market approval. Upon completing that will be approved. So this is the overall process protocol followed for approval or commercialization of any in vitro diagnostic devices globally, or in case of USA FDA approval process. Now coming to the Indian scenario. So we did not have this kind of process protocol prior to 2017. These medical devices have been developed in medical devices rules have been developed since 2017, wherein it is regulated by the CDSCO. This is central body, which is Central Drug Standard and Control Organization, which is the CDSCO, and that was considering all the medical devices as drugs earlier to 2017. So prior to 2017, all the medical devices were also considered as the drug only. But now in 2017, they have brought this medical devices rules, which has 12 chapters, 97 rules, 8 schedules and appendix forms seeking for manufacturing, sales, import, storage. So all those things are also taken care separately in the medical devices rules. So we all know that Drug Cosmetic Act was there since long, like 1940, which has various amendments as well. But for medical devices, such a separate thing was required, and that was done in 2017. And currently, like I think from October 1st, 2018, it is implemented, but still various medical devices are still not notified under those medical devices rules 2017, and currently it is underway. So by the end of 2022 or middle of 2023, all the medical devices that are sold in India need to be regulated under the MDR 2017, right? So only the devices that are notified by government are regulated and falls under the provisions of regulation as per MDR 17. This is Medical Devices Rule 2017. So this information is probably like 2 to 3 months old. So only 16 categories are notified, and in IVD only 4 are notified devices and other are non-notified. But by the middle of next year, everything has to be notified. So there is a huge regulatory revolution happening in the field of medical devices, where everyone is wanting their device to be notified by the CDSCO. So that is the recent update happening in field of IVDs licensing in India. And of course, there are two bodies. So CDSCO is a central body, which is Central Licensing Authority, and there are state licensing authorities with responsibility for licensing to import, manufacture, either for sale or for distribution, sell, stock, exhibit, or offer for sale. So these activities related to medical devices are taken care by two bodies, which is state body and the central body. So state body is responsible for class A and class B medical devices, which are little bit low risk, low or low moderate risk medical devices for their manufacturing, loan or wholesale licensing. The central body is responsible for importing all type of medical devices, although they are low risk or low moderate risk. But if it comes to importing of such devices, then the CLA, that is Central Licensing Authority, has to take care of the processing. Then there are class C and class D medical devices. Of their manufacturing, loan and wholesale licensing need to be approved by the CLA, that is CDSCO only. So these are the bodies which are involved in IVDs licensing in India. Coming to the market, so global IVD market, it is depending on the various categorization. So these are market by techniques, products, applications, end user, and geography. So various techniques are there in vitro diagnostics, such as immunology based, hematology based, chemical chemistry based, so on and so forth. So these are like market by techniques. Various techniques have various markets. Then the products, like some are instruments, some are in reagents only, like chemicals and antibodies and all. Then some are in services of those equipments, data management. So currently upcoming field like data management or the patient data handling. So all these are market by product category. Then market by application, wherein it depends on the diseases, various diseases like cardiology, nephrology, cancer, diabetes. So depending on that, there is a global IVD market categorization. And end user can be hospital, laboratory, patient self-testing, POC testing, or in academics even. So these are the end user based markets. And of course, geography wise, the regions of different parts of the world, they have been divided into like North America, Europe, Asia Pacific, and rest of world. So there is a huge market categorized based on these various parameters. The examples of various medical devices, as we are discussing about the IVDs, are blood glucose monitor, the clinical chemistry assay, immunoassay, devices for tissue typing, blood grouping, the cancer diagnostics, for infectious agent detection, human genetic testing, the PCR assays including next generation sequencing panel, the self-test or near patient testing devices. So there are n number of example under each category, which are considered as in vitro diagnostic devices. They are used for the testing and confirmation of the diseases or any disorders are there. So I'll go through simple few examples of such IVDs like blood glucose monitoring. So you might see the electronic devices there and the assay strips are there. So this is just a lancet which is used to prick the finger. You have to put so this pen itself probably is a assembled device wherein the pricking, loading on the strip is taken care. When the strip is put in the electronic device, then it can detect the glucose level in millimole per liter, right? So these such devices for blood glucose monitoring have been so useful for diabetic cases. Then the microfluidic devices, which help in development of compact devices wherein the sample requirement is very low, and such devices are currently being processed in various industries for exploration, wherein these microfluidic channels hold the sample or the conjugates in one form, and then the detection panel can be developed separately. So this is like a chip is developed separately, which is inserted in the equipment, and then the assay reading can be done, which can be seen here. Like the chip is separate, the device is separate, but herein there is no microfluidics. In this particular example, you can see the microfluidics channel fabrication is there. And of course, there are various applications of those currently under development, or few are there in market as well. So coming to the typical example that we are discussing since long about the COVID-19 rapid test. So this is the basic design of any lateral flow immunoassay, as we discussed: sample pad, conjugate pad, nitrocellulose membrane, absorbent pad is there, and there are two lines are there. So earlier it was a strip in tube format. As we know, it was like pathogenic virus, and then there was a lot of fuss about its spread and all. So we had to do it in a test tube so that there should not be any contamination to anyone. Right? It was closed format. But nowadays it is in the cassette format, which is open format, wherein you need to just put the sample, the additional buffer, if any, and then you can observe the test. But earlier it was in a test tube format only, right? So this is how the assay is performed for the strip in tube format and then strip in cassette format. So both have been explored in case of the COVID-19. Coming to another example of IVD, that is urinalysis test strip. So probably some might have seen such kind of panel is there. So this is plastic backing membrane on which various paper square, probably colorimetric squares are assembled, which each of them show the test for various analytes, right? From like nitrite, urobilinogen, so on and so forth. So these are the tests, and this is the pad provided by the manufacturer. So if you put your sample, the color changes to purple, that means the level is this. All right, so and so forth, like pH of the urine, if it may be tested, so this will be 5, and if the color changes after addition of the urine sample, if it is appearing like bluish in color, then the pH is 9. So this is semi-quantitative analysis, wherein these kind of reference standards are provided. You need to put your sample on these urine test strip, and you can detect the level of the analyte comparing the color of the test strip after addition of your sample. So the global market value, of course, huge scope is there. 121 billion by 2027 global market could be reaching there, and CAGR of 6.6%, probably more than that now, as this COVID situation has turned around, and then many medical devices companies have been establishing, their import has been increased. As in Indian scenario, we are now talking about reducing the import dependence and developing or promoting the medical device sector in India itself. By market, so IVD market by technology, as we have seen, the immunoassay, then the clinical chemistry and molecular diagnostics. So these three are major sectors or major technology drives which are taking front seat in case of IVD's development and global market. And in case of Indian market, as we know, the National Medical Devices Policy is implemented in 2015. Then medical devices rules came in 2017, and they gave some time of 3 to 4 years for all the manufacturers or importers in India to qualify for the categories to be notified in medical devices rules. So a lot of regulatory things are happening in medical devices currently. And so the market is basically, so the future for IVDs, what is the scope for IVDs in India? So these are currently in development stage and can be found on the several front. These include the increased full focus on full automation. So what we want in medical devices is full automation, the point of care testing and development, more efficient and sensitive diagnostic applications. So there should not be any false positive, false negative, or inaccurate methodologies for detection. So that is the focus of current industry sector, to develop something which is automated and reliable, repeatable methodologies. Apart from that, we all know the major attention is towards development of something which is personalized medicine or personalized diagnostic. So we don't prefer to go to hospital for any testing. We all like to have something which is at home for diagnostic. So it started with like thermometer, digital thermometer. So everyone has, almost every household has a digital thermometer, which is a kind of personalized diagnostic medicine, right? So it is personalized diagnosis medical devices. Probably during COVID, every household had this blood, what is that, pulse oximeter. So pulse oximeter also started selling, and the market rocketed suddenly. So that's why this personalized medicine also has huge scope, and there are many test strips or in vitro diagnostic devices that have huge potential to be explored in case of development of personalized medicine. So coming to the end of the talk, I'll also show you a small video wherein you can see the how the process works in industry for development of the lateral flow immunoassay strips. So this is just an overview how it happens in industry for all the development. Yeah. Particularly, this is how the antibodies are immobilized on the nitrocellulose membrane. So here you can see the nitrocellulose membrane, and on which the antibodies are printed. So this will be cut into horizontally. The guillotine cutter is also, you can see here, the strips are cut into parts. Right? And few also are semi-quantitative or quantitative methodologies where you can insert your strip into system for quantitative analysis. All right, so this is how the manufacturing in industry works for all the lateral flow immunoassays. Yes. So these are the references for our presentation, and thank you very much for patient listening. If any questions are there, I'll be happy to answer or we can discuss. Thank you very much.
Yes, may I request Dr. Patil?
Yes. Thank you, Dr. Vivek. It was really a nice presentation. You have touched many aspects about medical devices. Particularly, if suppose the audience has not come across these terms earlier, then it will be slightly necessary to emphasize again what you have told. Okay, so I would like to add a couple of things from my side.
Yeah,
See,
sure.
actually, regarding the classification of IVDs, which Dr. Vivek has just now mentioned, this is very important. Okay, whoever is having interest in regulatory affairs and all, or if suppose in biodata, if someone mentions that he's having a interest in medical devices and regulatory affairs, then sure he will has to go through all the details of these classifications of medical devices. He has to actually understand what is meant by class A, B, C, D. He has to understand what is meant by risk associated with these things. Okay. Actually, I appeared for one online course in which these details were taught, and I could not grasp all the details in a single go. So there was a what we can say a punishing examination that was conducted again, and I was asked to write a essay type of thing on this particular regulatory aspects of
Right,
medical
right.
device.
Yes.
Okay. Because what happens is when we say risk of a medical device, it is related to different levels actually. Means if suppose the disease is very severe, and if our medical device is wrongly predicting something, then the patient will be exposed to unwanted treatment, or patient will be going into still
Right.
worse condition. So that particular aspect is considered as a risk of medical device.
Right, right, right.
There is something called as confusion matrix in this case. Okay, confusion matrix means it tells about how many times we are going to predict it wrongly, how many false positive, how many false negatives are there after using that particular medical device. And then there are many regulatory aspects about this. As Dr. Vivek has rightly mentioned, there are few countries which are having their own regulatory aspects of medical devices. CDSCO is trying to adopt to some of these guidelines according to their understanding and their convenience. Okay, so in country like India, the regulations are slightly different, but even though they are taken from other countries' regulations, still there are some variations in Indian scenario. So it is important that if we are having interest in regulatory aspects of these medical devices or software as medical devices, then we have to very critically look into all these guidelines and try to understand what it really means. Okay, it was really important, and you rightly mentioned this in this introductory talk regarding medical devices and their regulations. The technologies which you have mentioned, all the demonstrations which you have shown through videos, all of them are very important. Okay, and to learners, they are going to be the beginning. I am sure that in audience, there are very few people who will be knowing what you are talking about really, but your talk will be instigating some enthusiasm in them to look at all these details once again. Okay, and if suppose you share your presentation with the uh, organizer, and if there is no some confidentiality issues in this presentation, will be necessary for others to go through once again.
Yeah.
Very right,
Then,
sir.
yeah, yeah. Regarding portable fluorimeter, you mentioned something.
Right.
Can you tell something about that? What was that portable fluorimeter that you talked about?
So during the PhD, when I was working on the similar issue for development of the immunoassay, which is fluorescence based. So when the test line, so in case of colorimetry, what we see in rapid test or pregnancy test kit, you can see the result on the test strip. But in case of fluorescence reader or fluorescence bed technique where the quantum dots are used, you cannot see the fluorescence comparatively. So you need to develop a portable fluorescence reader wherein the strip could be inserted and the strip could be illuminated using the UV source, and you can quantify the fluorescence. So that was the portable fluorescence reader I used to work on.
Okay. So this was really a very nice talk. Okay, I request all the participants to just go through all these details once again, And I'm sure that Dr. Vivek will be sparing time if there are some questions and all. Dr. Vivek, if you can share your email ID in the chat box, people will be able to be in touch with you regarding that.
Yes, sir.
And for students, particularly for regulatory affairs related teachers, again, all these aspects are very, very important. Okay, we have to look at these details very carefully. Many times we feel that we have understood something, but that is not the case. There is another layer of information. There is one more layer of information, and they are correlated. So that kind of confusions are there in this particular field. Okay?
Right, and this is developing, sir. I mean, just to add
Yeah,
to
right.
your description, it is developing,
Yeah,
adapting. Government is following the GSTF, that is Global Harmonization Task Force is there, which is working on this. And there are developments in regulatory for medical devices in India currently underway.
that's why it is more challenging, and this is the
Yes.
right time when we should start participating in these discussions related.
Right, right,
It will
right.
help us. Okay, from my side, I would like to thank Dr. Vivek for giving such a nice talk, introducing us to all the new things which are happening in this particular area, and enlightening about many aspects of medical devices, regulatory aspects, the way they are manufactured, the way they are tested, and all. Okay, this is going to help us in many ways. Okay? Thanks a lot, Dr. Vivek.
Thank you, sir. I mean, I'm taking this opportunity to thank once again for organizer considering me for this event. And yes, if any questions, if any discussions required further, I have shared my email ID in the chat box, so can be approached with that. Thank you.
Yeah. Thank you. Over to you, Dr. Chetpalli.
Okay. Thank you, sir. I wholeheartedly thank Dr. Chandragora Patil for anchoring, for describing what Dr. Vivek Borse has detailed for last 1 hour or so. Dr. Vivek has touched all the aspects of medical devices, the regulatory aspects regarding concerning with medical devices, the sensitive talks, the wrongly read test, and all the terminologies, all the newer devices. This talk of Dr. Vivek will go in long run because it is a new child, newly developed aspect, just incepted around 2017, and the younger pharmacist, younger generation will take it, should take it in stride, should go through the presentation, should go through each slide because they were very much informative from laboratory to device. It takes a long run, and definitely Dr. Vivek Borse's help would be sought in coming days. Thank you, Dr. Borse. Now, may I request our technical team to acknowledge the efforts of Dr. Vivek and Dr. Chandragora Patil for chairing the session as well as informing us.
Thank you, sir.
Borse to gracefully accept this certificate.
Thank you, sir. Thank you very much.
On the behalf of organizing committee, although we are very, very humbled and we are very small in front of the stature of Dr. Chandragora Patil, we request you to accept this as a token of love. As
thank you for everything. Yeah, thanks to the organizers. Thank you to Upashni sir also. Thanks to Chetpalli sir for giving me this chance. It was really nice to interact with both Viveks on the same platform.
Thank you, sir.
Thank you.
You, Dr. Atish. Dr. Atish, you are mute.
Thank you, sir. Dear delegates, now let's move on to the next session. The session is going to be delivered by Dr. Amit Bhople, who is working as Assistant Manager, Regulatory Affairs with SNJB Pharmaceuticals in Nagpur, which is a subsidiary company of groups. To chair the session, we have Dr. Shashikant Bagade with us, who is working as Associate Professor in Pharmaceutical Chemistry with NMIMS Shirpur campus. I welcome Amit Bhople sir as well as Dr. Shashikant Bagade for this session. Now,
Thank you, Dr. Atish.
now I will take an opportunity to introduce the chairperson for this lecture. I am really pleased to introduce my best buddy, my classmate and batchmate of MPharm and PhD, Dr. Shashikant Bagade, who is MPharm PhD in pharmaceutical chemistry from RTM Nagpur University. He has also done postgraduate diploma in patents law and he is very good in Yoga Shastra. He has done masters in Yoga Shastra, MA in Yoga Shastra. He has been awarded with travel grant for attending international conference at Mauritius in 2012. He also received the Certificate of Excellence in Teaching from NMIMS University in 2014. He has guided many MPharm students and PhD scholars as well. One scholar has been awarded PhD under his guidance, and currently one more student is pursuing doctoral research under Dr. Bagade sir. He has published many books and book chapters in national and international publisher. He has around 35 research publications and around the same number of presentations in national and international conferences. So we are really happy to have you with us, Dr. Bagade sir.
Thank you.
Now, I would request you to chair the session and proceed with the session. Over to you, Bagade sir.
Okay, thank you, Dr. Atish. Really a very nice introduction of mine. This is a kind of a nightmare to me. And I'm very thankful to respected principal sir and team SNJB for inviting me. Friends, I got opportunity to chair this session for international virtual conference on these global regulatory challenges to seek approvals for ANDA medical devices, herbals and nutraceutical, which is the topic which is near my heart. And here we have a session of Mr. Amit Bopre. He is working with Regulatory Affairs Department at Siddhayu Ayurvedic Research Foundation, Nagpur. He is BPharm and MPharm with specialization in pharmaceutics with certificate expertise in the course of regulatory affairs. He has total 11 years of experience in international regulatory affairs. He is having expertise in marketing authorization, product registration, in semi-regulated and ROW, that is the rest of the work market for pharma, complementary traditional herbal medicines, veterinary medicines, health supplements, nutraceuticals, cosmetics, and personal care portfolios. Mr. Bopre has good experience in regulatory submissions for various modern pharmaceutical dosage forms like tablet, capsule, granules, syrup, ornaments, etc. He is well versed in dealing with regulatory authorities, pre and post submission queries, legalization, and various documentation part therein. Friends, we know that the classification of drug varies from country to country, and this variation leads to requirement in the regulatory aspects, which is having hold on controlling whatsoever the procedures of pharmaceutical steps which are there in marketing, authorization, as well as manufacturing of this herbal and nutraceutical medicines, nowadays which is known as dietary supplement. Herbal regulations are defined as nothing but the concept, power, or legislation designed to regulate the manufacturers of herbal medicines. There is a critical problem nowadays in the analysis of herbal products. We know that this is the complex ingredient combination. Due to this complex ingredient combination, it's very difficult to clean up the sample, and that's why the analysis, just like what we have with the chemical drugs, which is nothing but the impossible kind of elemental thing due to the complex nature of the herbal medicine. And that's why we have the regulatory agencies and the suggestions related to the regulatory agencies to identify the changes in this area, like newly introduced regulation or regulatory market and the drug products, the detailed literacy searches as well as online searches for herbal medicine, herbal medicinal products, even the regulation, amendments in the regulation, in Asia, Southeast Asia, and various countries is very, very important to understand. Even for the academicians, even for the industry personnel, or even for the marketing personnel. So here we have a gem, another gem for this today's session, Mr. Amit Bhopre. I request Mr. Amit Bhopre to take charge of this session. In this, he will share his knowledge and experience on the area, regulation of herbal medicine in India, Europe, and African region, current scenario. Please, Mr. Amit Bhopre.
Thank you, Dr. Bagade, sir. Can you hear me?
Yes, you are clearly audible.
Yeah, so thank you, Dr. Bagade, sir. And thank you, Atish Mundada, sir, for providing me this opportunity to deliver my experience. I am not a native speaker in the international conference, but I am here only to share my work experience. So all I request you, please receive with your patience. So I am sharing my screen. So I'm going to deliver the session on the regulation of the herbal medicine in India, Europe, and Africa regions. So this is the herbal medicine. So this is the restricted area right now. Restricted area because the regulations are well established regarding to the pharma products. So you can say the modern medicine, but for the herbal products, it is quite neglected. But after the COVID pandemic, the world shifting towards the herbal medicine and looking as an alternative to the modern medicines. And also the modern medicines have some restrictions on the treating the chronic diseases. So world is looking toward the herbal medicine as an alternative for the chronic medicines or the chronic disorders. So that's why the herbal medicines gain importance across the world as alternative to the modern medicine, as you are well aware. So now the session is divided into the herbal medicine introduction, then worldwide growth of the herbal medicine, then regulation of the herbal medicine in India, herbal medicine regulation in European Union, and herbal medicine regulation in Africa. So last one is the challenges in the marketing authorization for getting the herbal medicine. So let us start with the herbal medicine as an introduction. So herbal medicine, as introducing the herbal medicine. So WHO is defined the herbal medicines as the herbs or the medicine material preparation and finished product that contains the active ingredient of plant, other plant materials, and the combinations. As in the modern medicine, the medicine contains the active ingredient, which are the synthetic one, like paracetamol, ibuprofen. But in a herbal medicine, the herb or the herb extract is an active ingredient, as you are well aware. Along with the herbs, the phytoconstituent content into the herbs is the active moiety responsible for your activity of the product. So the recorded evidence of herbal medicine used in India, Egypt, or Greek, Roman, Syrian texts added back to the 5,000 years. As India, China, Greece, or Egypt are the main countries where the herb medicine used as a folk medicine or long dated back history. And the traditional system of medicines are the indigenous in medicine existence in the recognized ethnic as a Chinese medicine, Indian Ayurveda, or the medicine African or Latin American practices. So China, India, Egypt, and other countries, the herbal medicine practice as a indigenous system of medicines. And the Ayurveda has a long dated back histories. And Ayurveda is a rich literature of these traditional medicines. And in Asia and African countries, still the 80% of the population depends on the herbal medicine for the primary health care. We, as an Indian, we are use the home remedies as a primary health care for the day-to-day health problems. So herbal medicine gaining popularity in regulated market like United States, Europe, Australia, Canada, for as a several complement medicine approach. So what is the complementary medicine approach? This complementary medicine terms is used alternatively to the herbal medicine, and many countries it considered as a traditional medicine, and a few countries it terms as a complementary medicine. Many regulatory regions is terms as a alternative system of medicines to the modern medicine. So herbal medicine market growing faster worldwide exponentially. So what is the worldwide growth of herbal medicine? So it is an alternative to the modern medicine, and as the mostly if you find the material come from the natural sources, so the adverse reaction is very low in the case as compared to the modern medicine. I cannot say there is no side effect or adverse reaction. Yeah, there is a side effect or adverse reaction in herbal too. People have myth and receiving the herbal medicine that it is safe, but it is not completely safe. Still, there is a adverse effect is there in a herbal medicine. So in last couple of years, the herbal medicine, so there are the number of regulations and the pharmacopoeia standards in the place. That's why the organization of the herbal companies are liable to comply the same. So that's why the last couple of years, the quality of the medicine is improved. Increase in the safety and efficacy studies. So the clinical trial approach as a mandate, as a regulatory mandate for the organization of the companies to launch the product into the market. So before you have to establish the safety and efficacy of the products. So that's why the organization or the companies engage into the execution of the animal human studies of the product. So that's why the increase in the safety and efficacy data of the herbal products. So limited result of the modern medicines. I told earlier that the modern medicines have certain limitations or don't have solutions to the chronic diseases. So the world is looking towards the complementary medicines. Increase in the research and scientific evidence. So the to establish the safety and efficacy and the quality, so mandate from the regulatory authorities across the world. So companies investing a lot of money or a lot of capital on the product and development of the herbal product. So that's why the increase in the research and scientific evidence comes, and it will deliver the quality of medicines and comes with the clinical trials data, animal studies, and also all the quality parameters has been going to be validated. So many herbal drugs are food ingredients. So like many regulated countries considered herb as a food ingredients. So if you go through the United States regulations, so FDA, US FDA does not consider herb as a medicine, but in the US market, the herbal medicine mostly promoted as a dietary supplement under the various dietary supplement regulations. And in many countries, the herbal medicine considered as a over-the-counter medicine, over-the-counter product. So these are the factors responsible for the growth of the herbal medicine, the use of the exponential growth of use of herbal medicine across the world. So now comes to the regulation of herbal medicine in India. So Ayurveda or the Siddha Unani medicine or the other herbal medicines like Unani. So it's practiced in India widely. And Ayurveda has a long history. The Ayurveda is used in India or practice in India, or the folk medicine. So dated back to 5,000 years ago. And herbs have the medicinal value in Indian traditional medicine, that is in Ayurveda, and other traditional system of medicine in India are Siddha, Unani, and homeopathy is widely practices. As I said, there is a rich literature for the use of herbal medicine in India. So our ancestor described the herbal medicine in various literature like Rig Veda, Atharveda, Charak Samhita, and Shushruta Samhita were the ancient one. So now this classical Indian text literature of Ayurveda, now the modern companies or the modern pharmaceutical companies refer these Indian classical texts and further carried out their research on these herbs by referring this Ayurvedic literature. Then the British brought the modern medicine in India. So before the British era, we rely completely on the Vaidyas and the folk medicines. So the British government systematically relegated the Indian traditional medicine and promote the modern medicine. So initially the British government in India has a reluctant approach toward the Indian traditional medicine, and they discourage the use of Indian traditional medicine and they promote the modern medicine. So the growth of the modern herbal medicine during the British era is quite stagnant. So post-independent era, the Indian government tried to restore the glory of the Indian Ayurvedic medicine or the other traditional medicine, but the execution is a very slow still. Then the government of India comes with the special ministry dedicated to the growth and execution of this traditional herbal medicine. So the ministry called as Ayush. A stands for Ayurveda, U for Unani, S for Siddha, and H for homeopathy. So government of India constitute this Ayush ministry in 2014 for the better growth and promotion of these traditional systems of medicines in India. So the regulation of these herbal medicine or the various alternative system of medicines, specifically talking about the Ayurveda. So the Ayurveda is regulated by the various regulatory bodies. Like the first, I said, Ministry of Ayush, Government of India. Then CDSCO, the National Drug Regulatory Authority, that is Central Drug Standard Control Organization. Then Drug Controller General of India. Then state licensing authorities. Various state licensing food and drug administrations are regulated the use and execution of the herbal medicine. And the Clinical Trial Registry of India also involved in the execution of the clinical trials on the herbal medicines. The Indian Council of Medical Research also been involved to carry out various animals and human studies on the herbs or the herbal medicines. So legislative framework of the herbal drugs in India. So herbal drugs is regulated through the Drug and Cosmetic Act 1940. Then there are amendments to the Drug and Cosmetic Act, Drug and Cosmetic Act Rules 2008. So the Ayurveda is regulated according to Drug and Cosmetic Act, comes under the Schedule T, and whatever the product claims for the Ayurveda, product claims and advertising is being regulated by under the Drug and Magic Remedies Act. So legislative framework of herbal drugs in India. So Ayurvedic system of medicine is regulated in India by legislation of Drug and Cosmetic Act. According to 1964 amendments of the Drug and Cosmetic Act, Ayurveda, Siddha, Unani medicine specified in the Schedule T are proposed for the diagnosis, prevention, and treatment of the human beings and animals, and developed under the procedure in the book of classical Ayurvedic, Siddha, and Unani system of the medicines. The Drug and Cosmetic Act 2008 by first amendment, utilization of the raw material by licensed manufacturing units of Ayurveda, Siddha, and Unani medicine of the product. And by Drug and Cosmetic Act Rule, Second Amendment, manufactured of herbal drugs can be used as excipients mentioned in the Indian Pharmacopoeia. So the herbal medicine is regulated by Drug and Cosmetic Act. If you say that if you develop the finished product, the herbs are the natural in origin. As this is a natural origin, so herbs are liable for the microbial contamination or the growth of the microbial contamination. So the Drug and Cosmetic Acts amend its regulation and come up with the use of the excipient. Those are specified in the Indian Pharmacopoeia. So the use of the excipients along with the herbs, that is very required to be delivered the herbs in a sophisticated pharmaceutical dosage forms. So the use of excipients nullify the growth of the microbial contamination in the herbal drugs. The Schedule T. So the Indian government or the Ayush Ministry come up of the herbal medicine should comes under the good manufacturing practices. Likewise, in a Schedule M for the modern medicines. So specifically, this alternate medicine applies for the good manufacturing practices. So all the manufacturing by the company should be regulated as per the good manufacturing practices for the Ayurveda, Siddha and Unani. See, it comes under the Schedule T. So according to Drug and Cosmetic Act, it is mandatory to register the herbal medicine product in each state drug licensing authority. If the company develop the product and want to market this product in the local market, so the company should have to register this product with the state licensing authority. The state licensing authority deliver or issue the product permission of that product. Along with that product permission, the state licensing authority also provide the free sale certificate. So the free sale certificate is the certificate that the local licensing authority provide you to sell your product in the local market as well as in the export. So there are the various product licenses issued by the state licensing authority, like product permission to manufacture the product, the free sale certificate to sell the product in the domestic as well as in export market. So another certification is provided by the local authority is the COPP, Certificate of the Pharmaceutical Product. So the certificate of pharmaceutical product is issued only to those companies which having the manufacturing plant WHO GMP certified. So the Ministry of Ayush or the CDC also issue the COPP certificates So COPP certificates is a product specific and the country specifics. The country specifics means in which country your product is going to be export. So it's a COPP is a country specifics. So company have to apply for all these licenses to state licensing authority for catering the domestic as well as the export market. So if you found many herbal companies engage in promoting the product in the domestic market with the very falsified claims. So as a herbal medicines, you cannot claim to cure, to treat any disease or any disorder. So many companies practicing these falsified false claims. So to control that falsified claims, the herbal medicines comes under the Drugs and Magic Remedies Acts. So it provides the falsified advertisement and label claims, which are mislead to the patients. The law provides advertising of drugs and remedies inducing following are the main terminologies discouraged by the Drug and Magic Remedy Act. Like if you are claiming that your product is inducing miscarriage or preventing the conception in women, improving and maintaining the capacity of sexual procedure, correction of menstrual disorders, or the cure, diagnosis, and prevent any disease or condition mentioned and included in the schedule. Or the Drug and Cosmetic Acts, Drug and Magic Remedy Acts stated the 45 element disease or conditions you cannot claim in your advertisement or the product labels. You cannot claim these 45 conditions. These are prohibited by Drug and Magic Remedies Act. So initially, the herbal medicines does not have any regulations or the standards to ensure the quality of the products. But in India, the Ministry of Ayush come up with the pharmacopoeias. The pharmacopoeias is legally binding to collection of standards and a specification of medicine used in the country of the region. So the pharmacopoeia standards provide a reliable mechanism and independent assessment for the quality of the herbal medicines. So Ministry of Ayush provide the standards for quality standards for the herbal medicines. So as per the Drug and Cosmetic Act 1940, official compendiums for the herbal medicines. So there is a specific pharmacopoeia of every alternative system of medicines. For Ayurveda, there is a Ayurvedic pharmacopoeia of India. For homeopathic medicines, the homeopathic pharmacopoeia of India. For the Unani pharmacopoeia of India, and Siddha, so Siddha pharmacopoeia of India. So not only these specific individual alternative system of medicine pharmacopoeia, but the Indian pharmacopoeia also includes the monographs on the herbs. So the companies are liable or the mandate to follow the standardization of their product to the Indian pharmacopoeia and the specific alternative system medicine pharmacopoeia. So standardization of these herbal products. And one more thing, that across the world, Indian pharmacopoeia of India is widely recognized as a legislative or the standard for the herbal medicine across the world. If you've gone through the ANVISA, the regulatory authority of the Brazil, if you find the Ayurvedic pharmacopoeia of India is an official compendium for the standardization of herbal medicines, as well as the ZAMRA, that is the regulatory authority of Zambia, then regulatory authority of the South Africa, that is SAFRA. If you've gone through the complementary medicine guideline of these regulatory authorities, so they officially declare the Ayurvedic pharmacopoeia of India is the official compendium, and its monograph on the herbs is widely accepted by these countries. So these are the various editions of the Ayurvedic pharmacopoeia of India, and they publish the number of monographs on the herbs. So the Ayurvedic pharmacopoeia of India is classified in the part 1 and part 2. So in the part 1, the Ayurvedic pharmacopoeia of India published the monographs on the particularly specifically on the herbs. In the part 2, the Ayurvedic pharmacopoeia of India come up with the formulation, but these formulations are the traditional one. The formulations for the traditionally which are included or the published in the traditional literature of Ayurveda. For example, the Ayurveda traditional dosage form like Bhati, Bhasma, Taila. So these are comes the Ayurvedic pharmacopoeia of India recognize these formulations, and the Ayurvedic pharmacopoeia of India come up with the formulary for these Ayurvedic traditional medicines. But still, the Ayurvedic pharmacopoeia of India does not publish any monograph on the finished products. You can say the finished products. So which are the finished products? The finished products the company developed in our in-house R&Ds by the formulation and development team, so and which are not referring to the classical formulation which are prescribed or the published in the classical text of this Ayurveda traditional literature. So the API, Ayurvedic Pharmacopoeia of India, also the Indian Pharmacopoeia does not bear any monograph for the finished herbal products. So this is the all about the regulation of the herbal medicine in India. So now we are come up with the herbal medicine regulation in European Union. So initially the European countries discouraged the use of herbal medicine due to the lack of the safety and efficacy data. But by the time as the studies and research increases in this domain, so many companies come up with the various studies and evidence of the safety and efficacy. So the herbal medicine regulation in European Union is the well harmonized and well structured. So the European Medical Agency is the prime body to develop the regulation, drafting and develop the regulation for the herbal medicine in the European Union. And all the European member states, which are all the countries considered as a member state of the European Union, liable to follow the herbal regulation medicine which are developed by the European Medical Agency. The marketing authorization are granted by the National Regulatory Authority. Although the regulations are same across the European Union, but the product registration and the marketing authorization for the product is granted by the respective national regulatory authority of that member state countries. So marketing authorization are granted under the European Union Directive 2001/83. So many this directive by the EMA, it's a little bit challenging for the companies to comply. So that's why there is a delay in the marketing authorization in European Union for the company. So the European Union or the EMA come up with the new directive, that is 2004/24. So this regulation come up into the force in 2004, and this regulation by the EMA for the herbal medicine is well structured, well harmonized, and a simplified one. The Committee on the Herbal Medicine and Product, that is HPMC, which is a part of the European Medical Agency, is the prime body to drafting the regulation under the guidance of the European Medical Agency for the herbal medicine. So responsibility of that committee is that the evaluation of the medicinal products. So whenever the company files the registration dossier of the herbal product along with the registration samples, so this is the prime responsibility of this committee to evaluate this product for the safety and efficacy. Another responsibility is the simplification of the registration. So the simplification of the regulation is the prime responsibility of this committee. The authorization of the herbal medicinal product. So this HPMC is engaged with the national drug regulatory authorities for the authorization of the herbal medicine products, establishment of list of herbal substances. So the European Medical Agency and HPMC also published the negative list of herbs. These HPMC or European Medical Agency carried out the various humans or the animal studies on a specific individual herbs. If they are found these are not safe for the use in human medicines, so the EMA published the negative list and a positive list of the herbs. So these we are the company, the R&D personnel referring these negative or positive list of the official herbs So while developing the product and development. The another responsibility, the preparation of the community herbal monographs. So the HPMC doing the thorough quality analysis, animals and human study, as well as the toxicity studies of the particular herb, and they publish the monograph officially on the European Medical Agency official website. So the scientific arena or the companies across the world refer to these monographs while developing their products. The safety and efficacy of the particular individual herbs is published into that monograph. Another responsibility is the addressing quality, non-clinical and clinical efficacy and safety issues in the guidance documents. So there is a very limited data available on the safety and efficacy of the herbs. So that's why these HPMC is also involved. Is there any issues on the safety and efficacy of this particular herb of the product? So they carried out their trials in their laboratories or the authorized center of the EMA and publish the data on particular herbs. So herbal medicine regulation in the European Union. So this European Medical Agency comes with the Directive 2022/24. So they stated the herbal medicine as a herbal substance or the herbal preparations. So herbal substance is unprocessed. You can say the herbal substance is in a raw form, which is unprocessed, usually dried from the form and sometimes fresh from whole plants, fungi, algae, lichen, or certain exudates, for example, gums and resins, are considered to be the herbal substances. And second, the herbal medicine, they categorize as a herbal preparations. The herbal substances are subject to various processes. So various processes carried out on the herbs, that is extraction, distillation, fractionation, purification, and concentration and fermentation to convert into the herbal extracts, essential oil or juices. So this directive classifies the herbs as a herb substance or the herbal preparation. But for the medicine, the EMA classify according to this directive the herbal medicine into three categories. First category is a traditional herbal medicine, which is a simplified registration procedure. For the herbs, there is a sufficient safety and efficacy data is published by the EMA or the HPMC, or the sufficient data on the safety and efficacy is published in the community. So these herbs can be classified, or this product can be classified into the traditional herbal medicine, and this process is a very simplified one. So these member states of the registrations, member state of the European Union consider this product comes into the traditional category. And also this category of the product must have the history that this product or herbs should be having the history to be used in the society from more than 5 decades or the 50 years. Second one is entrenched therapeutic use, that is established use. Established use that the company must liable to establish the safety and efficacy of the particular your herbal product or the herbs. Third one is the most complicated and a stringent one. The category is marketing authorization after the assessment. So you have to fulfill the full dossier of the product. So you have to provide all the safety studies, including the all the clinical trials, and you have to provide all the toxicity data with all toxicity endpoints like chronic, subchronic, reproductive, carcinogenic, all the studies. Also the clinical trials, all the phase 1, phase 2, phase 3 is required to file the under this category of the marketing authorization. The full dossier submission is required, and this is the most stringent for the company to comply this to comply and get the marketing authorization in this category. The herbal medicine regulation in European Union. So the third category, which is a stringent one, as I explained. So we have to file, a company regulatory department have to file the registration dossier according to European Medical Agency guideline for the herbal medicine. So the regulation of EMA encouraged to file the dossier in a CTD format. So CTD is simply the common technical documents. So this CTD format of the European Medical Agency for the herbal product mostly inspired from the ICH M4 guideline. ICH M stand for the multidisciplinary. So M4 guideline, ICH stated this M4 guideline to encourage the companies liable to file the registration dossier into the common technical documents format. So this format also mostly inspired from the ICH M4. So they are the 5 modules. The modules 1 is the administrative information. So administrative information, it comes the administrative information of your manufacturing plant and your products. So all the licenses like the manufacturing license, then WG GMP license, GMP certification of your plant, then site master file. Also, along with that, the administrative documents of your product, like forward permission, then free sale certificates, certificate of pharmaceutical products. All these administrative information comes into the module 1. The module 2 is the common technical documents. So common document summaries. So all the quality documents of your product comes under the summarized form. So all the quality documents as well as the clinical and non-clinical data of the product in a summarized form comes into the module 2. And the module 3 is the complete quality document dedicated to your finished product. So the quality modules comes to the, you have to provide all the quality documents of your raw material, active as well as the excipients. These quality documents of the raw materials include the specification and analytical methods for the standardization of your raw material, as well as the certificate of analysis which you are used in the product for the active as well as excipients. Also, you have to file the drug master file. Along with that, there is a method validations for the analytical methods. So initially, in the first part of the module 2, you have to establish the quality evidence of your raw materials. Then comes to the finished product. So all the quality documents of the finished product, right from the manufacturing from formula record, that is MFR, how your batch is prepared, that is batch manufacturing record, then all the process validation, analytical method validations, then specification of the finished product, as well as the method of analysis, analytical method development, validations of the finished product, and the stability studies should be of the finished product. So all the quality data we have to provide. The last one is the packing material. So you have not only have to ensure the quality data of the raw material and finished product, you have to provide the quality data on the packing materials also. So module 4 is come up with the non-clinical studies. So all non-clinical studies you have to provide in the non-clinical module. The clinical 5 is the clinical studies. So in the module 4 and 5, so all the studies you have to provide, like the pharmacodynamics, pharmacokinetics, and adverse drug reactions, side effects. All the clinical and non-clinical data should come in the module 4 and 5. So these module 5 are very extensive and very deep, and due to the time limitations, it's not possible to explain in a half and 1.5 hour session. So now we are come up with the herbal medicine regulation in Africa. So the African countries are the very low economic countries. That's why the regulation, this Africa continent is not well harmonized and regulated. So there are the 58 countries in Africa continent, and in some countries there is no regulation still in the Africa region of these countries for the herbal medicines. And the traditional medicines is practiced as a folk medicine by the African tribals for the primary health care. But now the few African countries are come up with the regulation for the herbal medicine, and herbal medicine is well regulated by these African countries like South Africa, Zambia, Zimbabwe, Kenya, Nigeria, Uganda, Ghana, Mali, Republic of Benin. So these countries have their own independent regulatory authorities to regulate the herbal medicines. So if you're talking about the National Drug Regulatory Authority, the SAFRA is a regulatory authority of South Africa. There's ZAMBRA for the Zambia, MCAZ for the Zimbabwe, TMDA for the Tanzania, NAFDAC for the Nigeria, Food and Drug Administration for the Ghana, and National Drug Regulatory Authority of the Uganda. So this independent regularity of these member state of the Africa regulate the herbal medicine in well manner. So the Zambia is the regulatory authority of the Zambia Medicine Regulatory Authority. Formerly, it is called a Pharmacy Regulatory Authority. So Zambia has its own regulation to regulate the use of the herbal medicine in the society. So before export to the Zambia, you have to comply with the Zambia Medicine Regulatory Authority's regulation for the herbal medicines, and you have to file the marketing authorization file of the herbal product with the ZAMBRA. So the structure of that your registration file is that it's first, as part 1 is the general requirement, part B is the summary of the product characteristics of your product, then part C is the quality requirement of your finished product, part D is the safety and efficacy studies of your herbal product, have to file in this part D. Part E is the efficacy data of your product, and part is the application form. So the registration dossier for the herbal medicine for the Zambia. So this is the structure of your registration dossier, or these are the main section of the registration dossier for filing in the ZAMBRA. So if you found the application form also, now we are come up with the SAFRA. The SAFRA is the South African Health Product Regulatory Authority. So SAFRA is responsible for the regulation and the registration of the herbal medicine in the South Africa region, South Africa country. So South Africa has their own regulation for the complementary medicine, and there is a own ZACTD, Common Technical Document format for filing of the registration dossier in the South Africa. So there are the various modules we can discuss in the ZCTD. So Z1 module is the administrative information. So all the applications, all the licenses of your products and manufacturing plant comes into the administrative information, ZA module 1. Then there is a various summarized studies. The summarized studies you have to file into the module 1. The summarized quality studies are clinical and non-clinical studies of your finished product should be comes into the module 1. The module 2 is a common technical document in a summarized form. So all the technical documentation, right from the manufacturing to quality testing of the raw material and the finished product, the clinical studies and non-clinical studies like pharmacology, the pharmacokinetics, pharmacodynamics, all the toxicity study in a summarized tabular form. You have to file in the module 2 of the common technical documents. The module 3 is mostly quality documentation dedicated to the finished product and raw material. The quality, we have to provide all the quality documentation of your raw material, active excipients, as well as the finished products, right from the validations, analytical testing, stabilities, drug manufacturing, drug master file for the actives, and quality data of the packing materials. The module 4 is a non-clinical studies. You have to provide, if along with the non-clinical studies you have performed your finished product, but you have to provide, is there is any literature published, bibliographic literature published on the finished product. And module 5 is the clinical study reports. The clinical trial studies have to be filed on.
Sir, I think you have muted yourself. Just unmute.
Can you hear me, sir?
Yes, yes. Now
Yeah.
it's
So along with the registration dossier in ZCTD format, you have to apply with the application form for the registration of the complementary medicine. So this is the section 121 application form for the herbal medication or the complementary medicine with which you have to apply in the SAFRA. So by filing this application, you are going to intimate the SAFRA that you are going to submit your ZCTD dossier for the registration of the product. So you will find this application form on the SAFRA official websites. So the next regulatory authority in Zimbabwe for the Zimbabwe is Medicine Control Authority of the Zimbabwe. So MCAZ have its own set of the regulation for the registration of the traditional medicines. So I am come up with the shortly the index of this registration dossier we are filing through our company. So it's very difficult to explain all the registration dossiers or the regulatory guideline due to the limitation of the timeline. So that's why I come up with these index or the registration file index. So we'll get a thorough idea in a short. So this is the index of the registration dossier to be filed into the Zimbabwe for the product registration. The first one is the cover letter. So cover letter, you have to intimate the regulatory authority, and in the cover letter you have to provide the power of attorney. So I will explain in a short what is the cover letter and power of attorney. So in Zimbabwe, according to regulation, you cannot directly export or cannot reach to the Zimbabwe market directly. So you have to tie with any distributor or any pharmaceutical company in the Zimbabwe origin. So you have to tie with the legal agreement with the any Zimbabwe pharmaceutical company, and you have to provide the power of attorney. So you have to grant the rights to the Zimbabwe pharmaceutical company that your product going to be distribute or market by that Zimbabwe pharmaceutical company in the Zimbabwe market. So all these formalities comes into the cover letter. So through the power of attorney, you are appointing and recognizing this Zimbabwe pharmaceutical company as your sole distributor come local regulatory agent. So this Zimbabwe pharmaceutical company act as a distributor as well as a local agent. So what is the responsibility of the local agent? So this local regulatory agent serve as a intermediate between the exporter and MCAZ. So all the communications for the product registration or there is any post submission queries. So all the submissions and the queries are communicated through the local agent, and the local agent is liable for the compliance of all the regulation in MCAZ. Also, the local agents also responsible for the any adverse reaction in the Zimbabwe market after the product registration and issuance of the marketing authorization by MCAZ. So CM1 form is an application form. You have to apply in CM1 form for the product registration application in MCAZ. And there is a declaration by the applicant on the product quality, safety, and efficacy. So product safety, if is there any complaint or any market complaint or the quality complaint from Zimbabwe territory? So the manufacturer, exporter, as well as the local agent liable for the same, and the said declaration on safety and efficacy have to be provided by the company in such declaration. The proof of payment of appropriate fees while filing the application form and the registration dossier of the product, you have to pay the registration fees to the MCAZ for the registration, for the sake of the registration. And registration samples, the product samples, you have to provide the product samples along with the registration dossier. So what is the purpose of these registration samples? That the samples submitted to the MCAZ, to MCAZ analyzing your samples on the quality and safety parameters in their own laboratories. Does it meet your samples or your products meet to the quality criteria of the MCAZ? Then the registration status, I forget to explain the registration status point. Though registration status there, the MCAZ has some mandate that your registration, the finished product must be registered in the country of origin. Country of origin means that if I am the manufacturer and exporter in India, so the India is the country of origin. So firstly, your product must be registered in India. If the product is registered in the country of origin or in India, if you are a manufacturer in India, then you are eligible to export or to apply in MCAZ of your product. Then it also has the mandate for the herbal product that your product must be at least registered in one country other than country of origin. If I am the manufacturer and exporter from the India, so firstly, its product must be registered in the India, but it also the mandate your product must be registered in any other country across the world. Then you are eligible to apply for the marketing authorization in MCAZ. Again, this section A, you see across the globe, if you have gone through the European Medical Agency structure of the registration dossier, then African like MCAZ or SAFRA, you will find the same modules. So nowadays the registration dossier format based on common technical documents getting harmonized. So it is very good and in favor of the company that if the format of the registration dossier or the regulation is harmonized across the world, then it's very easy for the company to develop the documentation, and you don't have to prepare the separate documents for the specific country, and your time and energy is spare for preparing the technical documentation for country specific regulations. So if you find across the various regulation, these dossier sections like section A administrative, section B, sometimes many regulations called as a module 1, 2, 3, 4, 5. So here is the administrative section. Section B is the particular of the product. or sometimes in many countries, the regulation is called as SMPC, Summary of the Product Characteristics. Section C is a composition of product. Simply, it's the manufacturing documents, your manufacturing procedure and the manufacturing process validations comes into the section C. Again, the same section D is a safety and efficacy. You have to provide the non-clinical and clinical studies. The section is comes again, the clinical studies you have to provide in these sections. And the section F, the post-marketing surveillance. Once your product is registered and to be placed into the market, then if there is you have to monitor the pharmacovigilance of your product. So this is a section in details of the MCAZ. The section 1 is administrative, sections B is a particular of the product, section C is composition of product. So all these guidelines and this structure for the registration dossier you will find on the MCAZ official website. And this is the CM1 application form. You have to intimate the MCAZ, and before filing the registration dossier, you have to fill and file the application through the form CM1. So lastly, the challenges in the marketing authorization for the herbal medicine. So many regulatory authorities still look towards a herbal medicine in the concept of the modern medicine. But the principle of the modern medicine and herbal medicine is quite different. Likewise, in the modern medicine, we are used as a synthetic active pharmaceutical ingredient as an active constituent. But in the herbal medicine, we are using the herbs or the extract as an active constituent. But itself, herb is not active. The phytoconstituent present in your herbs is the active moiety responsible for your product therapeutic activity. So this is the big difference in the pharma product, modern pharma product, and herbal medicine. So there are the various challenges to comply the regulations of the registration of the herbal product across the globe of the various regulatory authorities. So first challenge is the pharmacopoeia and monographs on finished products. As I said that Ayurvedic Pharmacopoeia of India, Indian Pharmacopoeia does not provide any finished product monographs. So in such a case, the companies don't have any standards to follow for the to standardize or provide the technical or quality data for the finished product. In case of the modern medicine, the monographs and the methods and various analytical parameters and their methods are well established in the pharmacopoeia. Recently, United States Pharmacopoeia come up with the monographs on the finished product. But here is the challenge is that the USP come up with the monographs on the finished herbal product, but these herbal finished product mainly contain the single ingredient, and these single ingredients mostly based on the standardized extract. So in such a case, they try to come up with the monographs, but you find the herbal finished products are polyherbals. So in such a case, it's more challenging to establish the quality data for the finished product, polyherbal finished product or polyherbal, because it's a complex mixture of the herbs. And if you find, if you say that the herbs are the natural in origin. So as a natural in origin, the phytoconstituent into the herbs, if you consider for example, Vidania Suminipra contains the vinolonide. But if you are developed the ashwagandha tablet or the polyformulation contained in the Vidania somnifera containing the vinolides, but during your manufacturing process, for example, the tablet, there is a granulation, drying, compression. So during that process, the content of your natural viranolides into the finished product get declined due to the process of drying and compression is that. And in your finished product, from that complex mixture of herb and phytoconstituent in your tablet, it's very challenging to estimate quantitatively a specific phytoconstituent in the finished product. So that's why there is no monographs or official standards for the polyherbal formulation. Also, likewise, in a pharma product, fixed dose combination, these monographs are available, but in herbal medicine, there are n number of herbs. So there is no any fixed dose combination of the herbs. Few companies use 5 herbs, then other companies use a different 5 herbs, and there are thousands of herbs are available. So it's very difficult to develop the Compedia or the monographs, because there is no such any fixed drugs combinations are available for the polyherbal formulation. Second one is the reference product. As I said, the herbal products are polyherbal in formulation. So as in case of pharma product, there is a reference product is available for the comparison, but in herbs, there is no any reference product for the comparison. Official analytical methods. In a lack of the as I said, as in the lack of the official monographs, official compendium for the monographs, so there is no official analytical methods to standardize the your finished polyherbal or the herbal product. So in such case, company have to develop the in-house analytical methods and validate the same. The markers. So for the markers, it's there is no availability of markers, although there is a markers available in the market, but these markers, there's an n number of herbs, so the markers are not available for the many of the herbs. So that's why it's very difficult to standardize your active herbs or active extract in case in absence of the markers availability into the market. Also, second is that the markers are the very costly, the very expensive. So the cost and expenses for the standardization of medicine, herbal medicine is getting costly, and it affects the cost of your product in the market. So assay methods. So as I said, there is no official monographs is available. There is no methods for the finished product, and there is a challenge to estimate the particular phytoconstituent in the finished product, because it contains n number of herbs, and a single herbs contain n number of phytoconstituent, and from that mixture we have to quantify that herbal medicine phytoconstituents. So there is two challenges: the lack of official compendium and estimation of phytoconstituent into the finished product. So that's why, so for the analytical development laboratory or the departments has a challenge for the development of the assay method of the particular phytoconstituent in a finished product. Again, the assay indicating stability study. So the regulatory authorities asking for the stability study data in accelerated as well as intermediate and long-term accelerated studies. They asking for the assay indicating stability studies. Means you have to provide the assay values for all the sampling interval into the stability study data. So the challenge is here that as if you find out any phytoconstituent in a finished product in an initial stability study sampling interval analysis, but as it is a natural in origin, the quantity of your phytoconstituent decline on the storage. So on the shelf life period, your assay of the phytoconstituent is declining. Again, the purity of the actives. as the herbs are the natural in origin. So the purity of your phyto herbs is a challenging because it's natural origin. So as in case of the purity, active pharmaceutical ingredient in pharma product is almost 99%. But here the establishment of the purity for the raw material or the active herbs is very difficult up to that, because the actives are adulterated with the heavy metals, then aflatoxins, and other materials. So it's very challenging to establish the purity of the actives. The heavy metals, as in a natural origin. So heavy metals test of the raw material and finished product is mandatory in every regulatory authority regulation across the countries, because the heavy metals are the carcinogenic, and as the natural of origin of the your raw material or actives. So many times your raw materials is fails in the heavy metal testing. So the heavy metal is big challenge for standardization of the raw herbs. The dissolution study, this is again the challenge. It's very difficult to find out the phytoconstituent into the in vitro studies. So that's why it's very difficult, and estimation of the phytoconstituent in a finished product. And you cannot ensure the solubility of that phytoconstituent from the finished product to the particular dissolution medium. So this is again the challenging arena. Bioavailability and bioequivalence. Here we are facing the challenge to estimate the phytoconstituent into the in vitro studies. That's why we are not able to establish the dissolution test for the finished product. Then also it's very challenging for the estimation of the phytoconstituent from the finished product into the blood. So that's why bioavailability and bioequivalence studies are also quite right now the challenge for this. ICH guideline for the compliance. Now the many regulatory authorities come up in a concert with that many regulatory authorities drafting their regulation on the basis of ICH for the herbal medicine. But ICH itself, you've gone through the quality studies or given guidelines. The ICH clearly indicate in the regulations that these guidelines is applicable for the synthetic drug or the pharmaceutical product, but not the herbal medicine. So because of these challenges, the ICH guideline is not we are not practically apply this ICH guideline for the herbal medicines, and ICH also itself declare in their regulation. But many regulatory authority drafting their regulation on the basis of ICH guidelines. So as a regulatory person, it's very difficult for us or the company to comply that regulation and provide the quality data according to that ICH regulation. Again, the polyherbal products, if your products contain n number of herbs, it's a polyherbal formulation. So it's very difficult to analyze the product because of its complex mixture, and in turn, it's very difficult to establish this quality data for the finished product. Another one is the radioactive contamination. Many regulatory authorities has been naturally and herbal in origin. So radioactive. So many authorities ask for if the herb is your radioactive contamination. So we have to provide the herb as well as the finished product. Does your product or active ingredient is radioactive contaminated? And this is also one of the very costly procedure. Last one is good agricultural practices. So initially it's not a stringency, but right now it's a stringency. The regulatory authority come up with the traceability of your herbs. So what is the sourcing of your herbs? either it is cultivated or comes from the or collected from the folk tribals. So if it is cultivated, so the cultivation of herb, if you are sourcing from the cultivated fields, so farmer have to follow the good agricultural practices because this is mandatory, because there is a contamination from the soil, there is a contamination of the fertilizer, pesticides and insecticides used in the field while growing these medicinal plants. So it comes, the traces of that fertilizer, insecticides, pesticides, or the soil contaminations or the water contaminations comes into your raw material, and then likely to be in your finished product. So there is a separate regulation of good agriculture practices. So these good agriculture practices ensure that these medicinal product free from this contamination. So where your raw material active herbs are sourcing from that particular farmer, so you have a legal agreement that with that farmers, and you have to establish the farming with the good agriculture practices certifications. So these are the challenge for the herbal medicine to seek the obtain the marketing authorization. So there's all about the India herbal medicine regulation in India, Europe, and Africa. These are the references. You can find all the regulations for these regions for the herbal medicines from the official authority, official website of these regulatory authorities. So that's all from my end, and thanks a lot for listening me with the patience. Thank you.
Thank you, Mr. Amit, for this great insight. Now, what we understood, what we seen as an audience, that the regulation of herbal medicine in India, Europe, and Africa region with this current scenario, Mr. Amit nicely explained all the aspects and the requirements for the regulatory guidance, even getting approval. Ms. Tejal, please mute your account. Okay. So here he covered the regulatory agencies of India, Europe, Africa, and regional markets. He explained the need of pharmacopoeia of herbal medicine. We are all in that to know that the regulatory authorities like ANVISA recognizes Ayurvedic herbal pharmacopoeia, Ayurvedic pharmacopoeia of India as an official compendia. So this is a rare to rare case, and as a part like our cultural heritage, we are even from centuries believe on the concept that Vasudhaiva Kutumbakam, and the same evidences we created during the COVID-19 scenario, that India supplied vaccines to the entire world. And a similar way, the Ayurveda is based on the same concept, that the medicine which is there, and the medicine which is for everywhere and for anybody, and that too in a what we say, the free form. So considering the similar aspect here, he explained herbal medicine regulation in Europe or European Union with the objectives of HPMC, and as a regulatory agency, he discussed the responsibilities of HPMC like registration, authorization of herbal medicinal products, establishment of herbal substances, preparation of the community herbal monographs, as well as the publication of the same on the European agency websites, because this website could be one of the source of guidance to the manufacturer as well as the scientists. Moreover, he explained the classification which is extended by the European Union, like traditional herbal medicinal product, intended therapeutic or established use, that is based on the traditional use of herbal medicine. So this entrenched therapeutic use is even present in India. So a similar kind of aspect like Indian herbal medicine is approaching. The similar kind of approach is there in entire world, right from the European Union, which is the biggest union. And similar way, the marketing authorization after assessment of the full dossier, that would be under the European Union. That is a common practice with the this chemical or what we say the allopathic drug product also. So further, he shown the importance of ICH M4 guidelines, multidisciplinary guidelines, where there are 1 to 5 modules and their significance. He further discussed the herbal medicine regulation in Africa, how the 58 countries of Africa actually progressing through this drug regulation. He discussed the Zimbabwean regulatory agency, and he mentioned the ZACTD common technical document format. That is a huge format and informative things which is shown through this presentation. Then the South African Health Product Regulatory Agency, SAFRA, for submission of ZCTD dossier for herbal products and herbal medicine. Further, he mentioned the regulatory aspects of Zimbabwe as an index containing various important points required for the registration, filing of registration dossier, as well as the filing of the medicinal products to the regulatory agency for marketing authorization. He mentioned how the MCAZ acts as a distributor as well as a local agent, as a what we say the single intervention or platform for compliance of the laws, and how MCAZ analyzes the samples before getting the proper registration to the regulatory agencies. Also, which is a very the very important part, that is challenges in marketing authorization of herbal medicine. The herbal field, herbal technical field is greatly facing those challenges, right from the inculcation of the herbal medicine in the main market, like challenges in marketing authorization of pharmacopoeial monographs, finished product, reference product, official unavailability of the official analytical methods, then unavailability of the markers, their cost, as well as assay methods, assay indicating stability methods like SIAM, like purity of the active ingredient, which is in most of the cases a bigger challenge to pharmaceutical industries. Even the heavy metal dissolution study, BABE studies, ICH guidelines compliance, and not availability of the well-established ICH guidelines. Polyherbal products which are having no limitations, even the radioactive contamination in the polyherbal products through this GAP, like good agriculture practices, how the pesticide encroachment into the herbal product is there, and what sort of regulation and what sort of precautions are required for the regulatory agencies to cope up all such challenges or to face all such challenges. So he has given a great insight in all aspects of the regulatory agencies worldwide, in the major part including this India, Europe and African region. And a similar way, he has mentioned how the approach of any regulatory, any pharmaceutical industry towards the regulatory agency is there, and how one can get approval of the herbal product or the regulatory authorization of the herbal product in any country. So it's a great idea and a great insight of knowledge, a bundle of knowledge about this regulatory aspects. Now, at the end of this session, I request the audience to ask the question. Now the session is open for the questions. Also, I have 2 small questions to ask, but it's a turn of the audience to please ask any question if you have.
Hello.
Yes.
Hello.
CR
Sir, here. Actually, there is some development in terms of phytopharmaceuticals where it was expected that 4 constituents or markers from a herb, if we can prove the presence of them, then that particular medicinal extract can be marketed as a phytopharmaceutical. So what is further development about this? This discussion was going on since last 3-4 years, but then what are new updates regarding phytopharmaceuticals related regulations in India particularly?
In European countries?
No, in India. In India, how there is a new term that has come up, phytopharmaceutical. So is there any development regarding this?
Yeah. So for the phytopharmaceuticals, if your phytoconstituent is more than 90% of purity. So it considered as the active pharmaceutical ingredients. So in case diasmin, hesperidine are the two phytoconstituents used in various herbal medicines. But now I am giving the specific examples. If they are the purity is more than 90% and there is no other herbs if you are using in your finished product, so they considered as active pharmaceutical ingredients, and it considered as a modern medicine if you are delivering in the modern pharmaceutical dosage forms. So there is no clear approach regarding the phytopharmaceuticals. The terms comes up phytopharmaceuticals, but if your moiety is as a purity more than 90%, then it considered as a modern medicine, because it has a potency as compared to API. And if you comes for the polyherbal formulations, so the extracts are mostly that 5%, 10%. Even though we considered as a herb as an extract form, then its potency is 5%, 10%, or 20%. And if we're taking the finished product with the herbs as a polyherbal formulation, so herbs contain the phytoconstituent in a complex mixture. So that does not have that much of potency compared to the active pharmaceutical ingredient. So it is quite gray area. So the government don't have any regulations, specifically CDCSO, right now on the phytopharmaceuticals. There are a number of companies come up with some extracts in a control or release fashion also. But still, even though if you are come up with the phytopharmaceuticals, you have to fulfill all the animal as well as human studies to establish the safety and efficacy. And then only after that you will get a green signal from the CDCSO, Ministry of Ayush.
Okay. Thank you.
Okay. I hope this addressed the question. There is another question from audience. Mr. Anup Thakre is asking about, is there any specific regulation for use of excipients from natural origin used in allopathic formulation?
So now all the regulatory authorities across the globe, so it's a mandate that the excipient must be included in any official compendium, that is pharmacopoeia. Non-pharmacopoeial excipients are not accepted in the medicine category. So your excipient must have the status of official compendium pharmacopoeia. And specifically, if you are approaching the EU countries, so the specification of your excipient is mandatory. The IP grade excipient is not accepted in European Union. So you have to follow the excipient should be British BP grade and USP grade. And these USP and BP grade ingredients are well accepted across the globe. The IP grade ingredients still don't have a good acceptance across the world. It's accepted in non-regulated countries, but even though the IP grade ingredients in a semi-regulated and regulated countries is not accepted. So that's why for the development of the product, company runs the country-wise project.
Okay. So I think he has given a good insight for these two questions. There is another one question that Ms. Snehal Wani is asking. that overheating of natural polymer using formulation does affect the particle size of any changes. I think that is a little bit a technical question.
Yeah.
So can we handle this? Please, sir.
I don't think this questions goes to the product development or formulation
Yes, very
development
specific
sides.
question is there.
Yeah, it's not related to regulatory. So I am not the right person for this question.
Yes.
Dr. Shashikant, Dr. Vijay Are from JRS Pharma here. There was a question on excipients.
Yes,
So
sir,
I thought
please.
I will add something because I'm from the excipient background.
Yes, sir,
So
please.
as Sir said very clearly, correctly, all the pharmacopoeial acceptance and everything is required. At the same time, there are very high regulatory requirements on the certifications like GMO, non-GMO grade, origin, TSC/BSC certification, animal origin, and other regulatory requirements which needs to be fulfilled for any excipient. So it may be of pharmacopoeial grade, but you should have additional certifications which are required for the for this all any type of, whether it is oral, topical, injectable, dosage form. So all the excipients, they also have very high demand on the regulatory regulatory documentation part and the purity part. Yes, that is from my side.
Yeah. So, sir, I respond with some technical challenges we are facing for the excipients. So additional certification, if you're talking about the additional certification for the excipients, if we go for the capsule shell, so capsule shell mostly the gelatin origin. So the TSC/BSC certification is a mandatory because the gelatin, if the animal in origin, so it carries the genes responsible for the encephalopathies. So you have to ensure your capsule shell is a TSC/BSC free, and so the vendor for the capsule shell is come up with the TSC/BSC studies. So these TSC/BSC studies and certifications also need to be filed along with the registration dossiers. Regarding about the animal origin ingredients, other requirements also is there. So TSC/BSC is the main requirement.
Okay. Okay. I think we have taken sufficient questions for this session. Now, to the end of this session, here is a small request to the audience also. You will be definitely, with the permission of the speaker, you'll be getting the presentation or PDF file for this presentation. It will be circulated to you. I hope it will not be a confidential one. Also, here is a small request. Audience need to provide a feedback of this session to the organizer through the proper way or proper mode, which has been extended by the organizer. So, for this session, I must say thank to Mr. Amit Bhople for his night insight, and the regulatory agencies, their working, their perspectives in India, Europe, and African region in case of herbal medicine. So thank you very much, Mr. Amit, for your great insight and your overwhelming knowledge extension for the audience for this the international conference, which is on the challenges to seek approvals for ANDA medicinal devices and herbal and nutraceutical formulations. So thank you very much. Thank you.
Thank you, sir.
Mr. Atish.
Yeah. Thank you, Amit Bhople, sir, and thank you, Dr. Shashikant Bagade,
Thank you, sir.
your valuable time, accepting our invitation, and being there with us on day 2 of SNJB PharmaCon 2022. As a token of love, would like to appreciate your efforts by giving a certificate to Amit Bhople and Dr. Shashikant Bagade as well. So I accept the certificate as a token of love and appreciation from the organizers and management and faculties of SNJB's SSDJ College of Pharmacy.
Okay, thank you, sir.
Thank you, sir.
Can we have a certificate of Amit Bhopre and Dr. Shashikant Bagade? Thank you, sir.
Thank you.
Thank you very much.
Thank you, sir, for this opportunity.
Thank you, sir.
Thank you.
We move on to the third session of the day 2. The session is going to be delivered by Dr. Pramod Agrawal from Netherlands. And to take over, I call upon Dr. Sanjay Patil.
Thank you, Dr. Atish Mundra. Very good noon to one and all. I, Dr. SNJB Patil, welcomes you all for the next technical session of day 2 of International Virtual Conference SNJB PharmaCon-2022. Respected Dr. Pramod Agrawal, sir, who is technopreneur from Netherlands. So on behalf of team SNJB PharmaCon 2022, I warmly welcomes you to Dr. Pramod Agrawal, sir. He is going to talk on cross-technology innovation, building ecosystem for delivering holistic health and care. To chair the technical session of Dr. Pramod Agrawal, we have Dr. Vijay Aire, sir, from JRS Pharma India. I also welcomes Dr. Vijay Aire, sir. It gives me an immense pleasure to introduce Chairperson Dr. Vijay Aire, sir, who is Head Pharma Business Unit at JRS India. Dr. Aire has completed his PhD from MS University of Baroda, Gujarat in 2005. He has also completed Executive Program in Management, Marketing and Human Resource Management from Shailesh J. Mehta School of Management, IIT Bombay. Sir is having almost more than 20 years of industrial experience. He worked in various reputed organizations. To cite few, he worked as a project manager and research associate at Inventia Healthcare Private Limited for 5 years. He worked as senior research scientist at Ranbaxy, where his role was secondary API source addition to the existing filed formulations for the global markets. He also worked as senior manager at Advanced Drug Delivery Research Department at Panacea Biotech Limited for 2 years. He has worked as regional business manager, pharmaceuticals, Gattefossé, India for 4 years, and senior account manager at Lubrizol Life Sciences Health India and is for 4 years. Experience Hello.
Yes, sir.
Am I audible now?
Yes, sir. You are audible.
Sorry for the technical glitch. Thank you, Dr. Atish Mundra. A very good noon to one and all. Greetings of the day. I, Dr. Sanjay B. Patil, welcomes you all for the next technical session of day 2 of SNJB PharmaCon 2020. The technical session will be deliberated by Dr. Pramod Agrawal, sir, who is technopreneur from Netherlands. He will talk on cross-technology innovation, building ecosystem for delivering holistic health and care. So on behalf of team SNJB PharmaCon, I welcome Dr. Pramod Agrawal, sir. The technical session of Dr. Pramod Agrawal will be chaired by respected Dr. Vijay Aire, sir, from JRS Pharma India. It gives me an immense pleasure to introduce Dr. Vijay Aire, sir, as a chairperson. Dr. Vijay Aire presently head Pharma Business Unit, JRS Pharma India, who is responsible for Pan-India Pharma excipients business. He is having vast industrial experience in different industries. He worked as a project manager and research associate at Inventia Healthcare Private Limited, then senior research scientist at Ranbaxy, senior manager, Advanced Drug Delivery Research Department at Panacea Biotech, then regional business manager of pharmaceuticals at Gattefossé, as well as senior account manager at Lubrizol Life Sciences, Health, India and Middle East. Dr. Aire sir is experienced techno-commercial manager with work experience in the different areas like pharmaceutical dosage form development, business development, and selling of excipients, as well as contract research business development, B2B business, lead generation, and new business opportunity creation. He is having significant experience in drug delivery, US Food and Drug Administration, formulation research and development, pharmaceutics, patent laws, and management and leadership skills. With this brief introduction, I request Dr. Vijay Aire sir to chair the session for Dr. Agrawal sir. Over to you, Dr. Vijay sir, for further proceedings.
Thank you, Dr. Sanjay. Thank you, Dr. Upasne, for inviting me, remembering me. And thank you, Sanjay, for taking me down the memory lane. It was really a good experience, long experience. And I'm really honored to present Dr. Agrawal, who is exceptional. I can say Dr. Pramod Agrawal is a highly skilled technopreneur, not only entrepreneur, technopreneur with 20 plus years of multidisciplinary education, research, sustainable functional product process development, and entrepreneurial background. He has working experience across the geographies, including Europe and India, cross-functional, cross-cultural teams. He developed course curriculum, delivered lectures, guided bachelor, master, PhD students in the area of material science, functional textiles, biotechnology, and sustainable chemistry. He is a principal inventor of 4+ international patents, numerous international publications, and peer reviewer for 10+ international scientific journals. And he is busy along with all these things, besides all these things, delivering lectures at international conferences like one of these today. Not only this, he is also founder of Agrawal Ecolabs, a high technical consultancy company for textiles and related industries, co-founder of Saxel BV, an innovative cellulose fiber produced via sustainable process from cotton waste. This is really interesting because JR is also in the cellulose fibers, and this will be really interesting to follow. And a co-founder of Dev Toppers and digital evangelist working towards digital transformation, Industry 4.0 and Factory of the Future. Along with that, he is a chairperson of Aadhaar, the NGO working towards UN Sustainable Development Goals. He is vice president of FCCI, Foundation for Critical Choices for India. It's one of the oldest think tank of overseas Indians working out of Netherlands. He's a member of steering committee of FIDE, Foundation for Indian Diaspora in Europe. With this as very long career and different, as correctly said, it's vast career and in different multidisciplinary aspect. We are fortunate to have him today, and we are looking forward to listen to his experiences and his presentation on the topic. Thank you very much, sir, for making this happen, and we are looking forward. Over to you, Dr. Agrawal. Thank you.
Thank you, Dr. Vijay. It was overwhelming to listen to your own biodata in certain sense. Just to mention, if you see my voice a little bit dumb, we still have to have sunrise. It's still dawn. and outside temperature is 0 degree. At this moment, I can show you on the screen. So, for us, still early morning. Thanks to Vivekanand, who was my colleague and senior in UDPS Nagpur, who invited me and introduced me to PharmaCon 2022. And I'm delighted to be here. As Dr. Vijay rightly mentioned, the broadness and the variety of my background, what I thought is I would try to bring some of my learnings in my presentation, because most of you are, a postgraduate, PhD students in educational profession, and some of you are in industry related. I have observed and seen this world from a bit different perspective. And I think I have picked up a couple of aspects which match with today's theme. I will not go too detailed into regulatory issue. To be honest, if we keep talking about regulatory, then it might be very boring as well, because there are a lot of experts who spoke probably yesterday, who spoke this morning, and who are going to speak again tomorrow on the topic. But I will touch some of the aspects. So I'm trying to share my screen. I don't know if it works in the right way. Is it visible?
Yeah.
Yes, sir.
Perfect. So as I said, I would talk on cross-technology innovation, building ecosystem for delivering holistic health and care. I put deliberately "and" in between health and care, because there is a difference between two. I would try to bring a little bit, not too much focus on medical device regulations in the EU. You see, often when we talk pharma, we talk about medicine, be it the Ayurveda or be it the allopathic. But there is another angle to a medicinal thing. If your bed is helping you to recover from any illness, then your bed is also a sort of healing product, and that also have regulation behind it. Just to give you an idea about it. I have just put my LinkedIn profile, so anyone who's interested who can visit there, and please feel free to connect with me. Very basic when we talk about any innovation, because I think that is the key ultimately. All the industries, all of us as a researcher, as a student, we have to discover something new. And you can broadly define it into three groups. It could be copycat approach. Someone come up with a very innovative idea and we copy it, and then it's say, okay, me too. You could also have this incremental efficiency, instead of one blade, you have 2 blades and then 10 blades, and then you say, oh, we are improving. Or you could have breakthrough innovation, and then you have a transformational innovation, basically. And these two categories are very much needed when we talk about innovation. And to achieve this sort of innovation, what ingredients are required? Because we need to create certain fusion with different minds and different sectors and different ideas to come to this. And I will also reach there. When you talk about health and care, basically health, you can divide into primary, health and care. Let's say the more care part is about maintenance or improvement of health via, prevention, basically. So the care sector. When it comes to, I could also say health and care more as a cure and care, more prevention and cure in that aspect. And I don't have to go in detail about, their primary care, secondary care, and tertiary care, whether it's happening in your general practitioner level or at physiotherapy or dietitian level, or really in the specialty hospital. Coming back to India, because I think 21st century is the century of India, and there is no doubt about that. At this moment, we are the 5th largest economy in the world. And by 2050, we would be the 3rd largest economy in the world. We are a very unique country with so much of diversity and expertise also in terms of knowledge and ecosystem. But we still have to make best use of our ecosystem and diversity by interconnecting it to achieve, unrealizable, basically. When you look at ambition of India, what India want to achieve. At this moment, we only have 2% of our workforce, which is really skilled, India is a very unique country, as I said, but also one of the youngest country in the world. And India want to create, these numbers are in millions, but in crores, close to 50 crores strong resource pool, the young workforce who could contribute to knowledge economy. What knowledge economy means is not we are talking about if I have sugarcane and then, we cut sugarcane and we just sell it for making some jaggery or good. What we talk about is, can you make something very sophisticated product out of it? Instead of selling something raw, can we convert our raw materials into something valuable and useful? As I said, India is really diverse in terms of material resources, but also as well as mind resources. So I think this transformation is very much needed. Instead of a labor-intensive processes, we have to more go towards knowledge-intensive processes and products, basically. And I think there we need this, for this sector, we need at least 5 crore additional quality workers. And how to be one of them is also a big question, And of course, India has identified 20 sectors in which he want to be in top. I think this is relatively new concept, and it was quite common in Europe. Like Netherlands, say we are such a small country with only 1.7 crore people, but then we have companies like Philips and Unilever and Shell and the big names. So they only said that we cannot do everything, because if we try to do everything, then we would go nowhere. So we have to really focus. So they call it top sectors, and they come up with 9 top sectors. And I'm happy now India has also identified 20 sectors where it want to do excellence, basically. So how India want to make best use of its demographic dividend? That means we want to use our young workforce to achieve this. If we do not use our young workforce in the right way, instead of dividend, it would be a sort of a liability on us. Sometimes people say that population is a big problem, but population could be also a very good, let's say, a solution which world is looking towards India, basically. If you look at the India's population, at this moment, the average age of India is less than 30 years, basically. By 2050, we would crossing easily 150,000, 150 crores in numbers. By 2028, we would be the #1 population country in the world. But the beauty with us is we are still very young country. That means our average age is not going above 35, 36. At this moment, it's 23, 24 in 1990. And now somewhere around 29. That means we are one of the youngest country in the world, and which is a very luxurious position, because the rest of the world is really aging. Just to give you a number, in next 5 years, Europe would need at least 5 crore new workforce to enter in the market. At this moment, as I'm speaking, Netherlands has more than 10 lakh job vacancies open, where no one is, there is no one to fill those gaps, basically. And that's why they really allow that one person can wear multiple caps. And that's why you see from my profile that I have one cap of being a founder of a company, but I'm also connected to university, and I'm also could possibly deliver lectures and so on and so on, because they don't have people to do this sort of things. But what is very important, that not everyone can come and just enter in a workforce. You have to have a right skills that matches with their requirement, and this is very, very important to understand the world from a different perspective, basically. So if you look at India's position, then India would be probably the only country who would be having the workforce in the age group of 15 to 64-year-old by 2050. Rest of the countries like Japan is seriously struggling with it. That's why they have, at this moment in Europe, the The retirement age initially was 60, then they did 63, then 65. I think by the time I retired, the retirement age would be 70. And they even slowly started allowing people to work from age 15. That means they are making use of limited resources to achieve this. But this would be the case not only for Europe, Japan is even worse. And because of one child policy, China is also going in the same direction. That means there is no doubt that we are the only, probably apart from one or more, if you see this global map, all the red dots, all these red dots are negative workforce. And yellow dots, okay, they are not so negative, but not so positive. But there are only one or probably one or two more countries like India, which has a surplus population. But Indian workforce is very flexible. We are not fundamentalist. We are, sort of can communicate and speak in English very well. So we are really, and because of diversity, we are accommodating each other culture in a very neutral way. So these are some of the quality that makes Indian workforce really, really, appreciated worldwide. And you see that slowly the vice president of US is Indian, the prime minister of UK now is Indian at the age of 42 and so on. So really people are looking probably India to, and this is our soft power. And once we could reach to certain position, we could work for a harmonious way. So the challenge still for India is how to create 1 to 1.2 crore job every year. Or is it we should think also about self-employment as a possibility, and our education and research should focus on how this knowledge could be commercialized overall, and a skill development part, but and the productivity. Sometimes what also happen is we say, oh, we have 50 students and I don't have research topics. So sometimes same student get 5 students get kind of a similar research topic. So we are making our workforce less productive in that sense. So we really need to make also one negative thing about Europe is we have a lot of less people. So we are trying to be as efficient as possible. One person is trying to achieve the job of 2 or 3 people being efficient. But in India, we have a lot of people. So sometimes we are becoming becoming less and less effective in our productivity. We have so many people and we try to achieve the job of 5 through 10 people sometimes because they're much easy to put them in a system, basically. So we really need to think about this from a very open-minded approach. So these are the 12 sectors I was talking about, from automobiles to aviation. What could be very relevant for this conference is the sectors such as pharmaceutical, you see it clearly here, but also biotech and chemicals. So out of 20 sectors, 3 sectors are really focusing on these 3 areas. I can also connect it little bit of wellness, in this part. And if you think out of it, then almost 20% of India's focus is on pharma related activities. And of course, India is the world's pharmacy, and COVID has really revealed that and the role India is playing in this regard. So if you look at pharmaceutical industry, basically, of course, it's growing, and I think there are more experts than me on this area. Anti-infective, cardiacs, and gastrointestinals are the biggest market share, basically. And innovation is really leading India in R&D, basically. But allied sectors such as medical tourism, it's also one of the very cost-effective to come to India and get your treatment done and things like that. And slowly, but in last couple of years, very rapidly, India has the highest number of US FDA plants outside the USA. We have a very strong formulation and drug manufacturing industry. We are not yet very strong in bulk drug manufacturing. It is still China, which is there. And we have our own market, 140 crore people plus in that area, basically. So if you think about idea, I talk about incremental innovation, breakthrough innovation, or transformation innovation. Similarly, ideas are very, very powerful, And often you could talk about very ordinary idea, let's sell vada pav, for example, or and they could be realizable. We could put that into a blue domain. But then there are some original ideas which are still realizable. We could call it green. And this is what we need to achieve as a low-hanging fruit when we talk about. Either be it be research. When I'm talking, it could be also achieve this concept in research as well as in industry, basically. And there are some yellow ideas, which are still original, but sounds very unrealizable. So I will come back to this sort of ideas. Think about when we talk about pharma sectors in India, we often talk about drugs and medicine. But why don't we also talk about technical medicine? Technical medicine need not to be really something that you need to consume inside. Think about a patch. It's a textile patch. If I try to load with insulin or something like that, I'm just creating a situation, and you could place it inside. Then it's not just the sustained release knowledge you needed, but also the textile knowledge or material knowledge is required behind it, basically. That means there is two clusters are combining together to achieve this sort of activity. And this cross-sectorial or let's say the how you call it, very innovative concepts need to thought about. And that's possible when you merge different disciplines together. Of course, they require more time to market, but also high preparation. But the strategy could be to achieve green to start with, but keep always something in your back burner, yellow ideas, on which you're working through collaboration, basically. Yeah, of course, these are the 20 odd sectors of Pharma Hub, or R&D units or formulation cluster or bulk cluster in India. Tarapur, Mumbai, Pune, Aurangabad, Maharashtra still leading apart from, let's say, Andhra or Telangana in that regard. Gujarat, I think these two or three states which are really leading in pharma sectors in general. Coming back to me, I think in a way I was fortunate to have a very diverse exposure. And sometimes when you have diverse exposure, you get some sort of mutant. Mutant could be positive way or negative way in that sense. I come from a very tiny village in India. I had my basic education in Marathi medium till 10th standard. I did D-Pharm to start with because then my grandfather thought that I could start a pharmacy shop. But then that transformed me. I really liked that study because you're studying and learning about yourself. So I was fortunate to score good, so I could go to B Pharma. But those days in 1994, 1998, the trend was pharmaceutical industry in India was not that knowledge intensive in a way because we had our own we have our own commitment to contribute to Indian workforce. So the more focus on manufacturing rather than R&D activity. I saw the trend of biotechnology, which was shifting. So I did bioprocess technology from UDCT, now they call it ICT, downstream processing of enzymes, proteins. And then I was directly hired in Twente University here in Netherlands to use enzyme technology for green processing of textile material, because enzymes can also dissolve fat or background starch and polymers. And they say, why don't we bring such knowledge to a larger industry, which is highly polluting? 50% of the global population wears cotton, but they use caustic soda to make it hydrophilic. Why don't we use some enzymes such as, polysterase or cellulase or pectinase and those sort of things. And I had a lot of research in that area. I tried to establish, I was 10 years in fundamental research. I did my PhD, postdoctorate. But every time I was a little frustrated because you come up with new knowledge and then you publish. And then you ask, okay, but this knowledge has the potential to go to the market. Forget about market, to the society, because every innovation has an aim to reach to society. It should serve the society. Otherwise, what's the purpose of that innovation? And then my professor said, no, Pramod, you have chosen a path to be in research. That means you start with a question and you solve it in the lab and you publish. And once you publish it, you either go more detail in that publication, or you pick up a new question and you solve it again. And you stay in this box of one, a fundamental research. You only do this throughout your life. And I say, but I feel a little caged there, because what if we come up with a new idea and we now have a knowledge? Why don't it goes to society? And they say, okay, then you have to liberate yourself and go outside fundamental research, but then don't be part of fundamental research, but go to more a different domain where you use fundamental research for picking up right innovations and bring it to society. And that was the transformation I had back in 2009. And with that intention, I started a consultancy company here in Netherlands, that I will bridge the knowledge gap between fundamental research and I will bring it to society. That's the mission of my company. And I started working for mainly the small and medium-sized companies to help them innovate, because the big entrepreneurs or big companies have very good R&D infrastructure, basically, but smaller companies do not have. But on the contrary, they have more pressure on them to come up with a new innovation. Otherwise, they cannot sustain in industry or in the sector, basically. So with that idea, I started this, and then I found a couple of very innovative ideas, and then a couple of companies, as you see here, I've started or still working and communicating with it. So what often, I think this is a very basic slide. I don't know if you know the concept of T-shaped skills. Often most of us are having an I-shaped skill, some of us are expert in one domain and we go deeper and deeper and deeper. But with that, you only achieve, this, let's say the straight line, how do you call it, the vertical line of depth into certain sector. And then you are really expert in that sector, or within that small part of the sector, you're becoming more and more expert. Some people often are very generalistic, when they become manager to more vice president or that sort of position in a pharma industry and so on, they often started losing their core skill and they become a manager or more expert in one domain, or how to manage things, sort of a jack of all and master of none sort of approach. And then we develop a little bit of horizontal, let's say, a skill set. And what is more and more required in today's context is to have both. That one way you have, a sort of general expertise, and you have a specific expertise. I think for most of you, pharma is a sort of a I-shaped area, but see how much T-shape you have created, how much horizontal. That means, do you have some skills which are a little different from others, like maybe in IT, which is really, really needed at this time, some management, maybe even in marketing or PR, or some soft skills, or you are more aware about the government sector, how things work, or you're more aware about the private sectors, gaming, whatever you could name it. The more you'd have a T-shaped skill, easy to anchor. If I have to hold a boat on a shore, then I need to throw this hook. And if you need to grab this opportunity, you need to have this T-shaped approach. And if you have this T-shaped approach, the chances that you fit in different teams is much higher. But also, this would help you to understand how people think, because, as I said, I have pharma and then biotech and then material and now also ICT. When I look at a problem or challenge, I don't look the solution from one sector, like I'm thinking as a pharma, but I also think as a biotech. Sometimes I look at that same problem as a material scientist, or I see is it a simple automation problem and it can be solved through an ICT background. So my approach there more is how I can solve it. The problem is often when we have a similar mind in a group, then they would think alike. and they are caged themselves in their own way of thinking. And this is very, very important to understand. And I don't care what T-shape you develop, but slowly, if you go to LinkedIn Professional, 1 month you can get it for free. There are a lot of Udemy or those sort of areas where you can nowadays. I think COVID brought one good thing to the world is the digital transformation. This conference is also happening digital, which was otherwise not possible a couple of years before. I could be available to you in the morning, which was otherwise not possible to you. So we really have to see that some of those skills, we have to pick up whatever your interest lies. And I'm sure everyone has certain interest there. Because one thing to implement finally as a product or process or service, this breakthrough innovation, which is very, very unique, is sort of our golden egg. To reach here, you need hundreds of ideas, and only couple of them are studied, and only few are developed, and maybe one or two are tried, and only one possibly would be implemented eventually. So this is very, very important that we have many ideas as possible. That also means that we should create a culture of letting everyone open their mind and speak. And that is also a very important message here. This following 2 or 3 slides are very important because this would help you to visualize where the world is going. That means the future problems or solutions you are probably finding through your sector, you could see what is there for you, So I think the humanity, the biggest challenge we are facing as a humanity is climate change, resource efficiency. What does that mean? We have a very limited resources. Water bodies, some people say that by 2050, the 10 big cities in India probably have a water level 0 completely. So the groundwater would be dried. Soil erosion, it's like a very thin crust on our area. Desertification is a big issue. Food, because of the uneven weather condition, the food need to be more and more drought proof or flood proof. The climate refugees will soon noticing, probably Bangladesh would have a big climate refugees coming out of it, but countries like Maldives would disappear from the map in coming years. And what I'm talking it's scary, but I'm talking climate change effect in not more than 10 to 20 years. That means all of us probably I see in this screen talking to me and listening to me, this will happen in our lifetime. But often we do not want to see this sort of things. And that's what I put this really, really on top. So any activity or any innovation or anything which you use this trends in mind would really help. United Nations come up with 17 sustainable goals. Please have a look. I don't want to talk about it, but these are the 17 goals which now India has received the presidency of G20 nations. We are one of the front runner of it. And probably India is now also bidding for the United Nations permanent seat. And India has this largest workforce. We have to prepare ourselves, Think about a land. The farmer prepare it well before the monsoon so that as soon as rain comes, the soil is ready to produce. That means my message to the young workforce is if you have to prepare yourself for these trends or certain conditions, when you have conditioned with that mindset, as soon as the opportunity would come, you would be the first one to respond. Otherwise, you are waiting, oh, what's happened, what happened? And then the opportunity is gone to others, for example. And as I said, young population, and our young population is not curse to us, but also boom to us. But also the mega cities India was probably witnessing. Employment is a big problem. So instead of running behind employment, think about self-employment still as a women participation is very, very important. It's still relatively low, but in pharma sector, that's not the case in general. This is less relevant, but in past, there was USA and the whole world revolves around it. Now we have 2 poles, US and China. So it's a bipolar. And now slowly Russia and India also having its own stand. That means we are going from one polar world to multipolar world. You probably listened to Mr. Jaishankar, and he said that, look, when we say that we will buy oil from Russia, then we'll buy it, we have to fulfill the needs of our own people. So you see now our politicians also talking the language of multipolarity. In past, you have to be in one camp of USA or not in that camp. I mean, of course, we maintain the neutrality for long, but this will also have really, really big effect on all of us. There are two forces in the world. It's always good and evil. The force for good, we could say the democracy, the rule of law. Here in India, you still can walk to the street and shout against the government and nothing much will happen. You cannot think about that in other countries, for example, including China and so on. But other trends, and this is very interesting for all of you to think from a T-shaped scale, society 5.0, and some of the key issue, it's like nutrition, because young population needs a nutrition, education, more equal wealth distribution, gender equality, rapid urbanization, women empowerment, mass migration from villages to city, and future pandemics, because COVID was not last pandemic, but what the forecast is that in coming time, the period between two pandemics will be shorter and shorter. We would be more used to the pandemics in future. That means we really have to talk about and prepare ourselves, and the sector has to prepare ourselves to deal with this. Of course, India is still one of the leading when it comes to the vaccine production and the quick research and so on. But I think if we empower our R&D with more automation, I think we would be much better off. Society 5.0 is a very unique concept where the society is living in harmonized way by means of both digital and physical way, like robotics and things like that together. But I think India has everything. We still have society 2.0, 3.0, 4.0, 5.0, depends which part of India you live. And technology playing a big role like digitalization, automation, industry 4.0, artificial intelligence, artificial e-governance. To give you an example, because there is a serious lack of teachers in Netherlands, what they're doing is they are creating a lab course with virtual reality. What does that mean? That a student before entering in a lab, he will learn all the good lab practices itself, so when you wear a glass, you feel like you are in a chemistry lab and you smell also a little bit of ammonia, for example, or acids around it. And then, they would ask you to hold a pipette and try to do something like that. And you could do those sort of things and you could gain certain score, and then it will have all the good practices which you need to do. I mean, this is an example for the chemistry lab, but this could be also an example of dealing with very sophisticated equipment and machine, which is available in industry. And you don't have to have all the time an expert to give you, the training on that regard, but there's such trainings like a flight simulator, we could also think about it. So my focus, if you notice it again and again, is multidisciplinary or combining two sectors, basically. But in economy, mass customization is very, very big topic. What does that mean? That you customize your product for a targeted audience. In past, you used to get vitamin tablets, multivitamin tablets for everyone, and now you have 30 plus, and then you say 40 plus and 50 plus. But then you also say, oh, for kids, for women, for man, more segmentation around it. That means you have more specific group to offer, basically. And data is new gold. What does that mean? The data will help you to identify what market is required. And you see, for this sort of things, you need a different sort of skill set, basically. Anything to happen, and these 5 factors are very, very important. Industry has to come up with this idea that they want to achieve this. Economy should support this, okay? India is very luxurious position because we have our own home ground market. Society should be ready to adopt this sort of thing. Technology should be at high level that you could really realize it, and the government policies has to be favorable. And when these 5 things comes together, often you see the innovation comes to society part. This is very, I don't know, have you heard about this concept, but this is relatively very new, but very structured concept. What does that mean? When you're doing a PhD research, you're often in a TRL, technology readiness level of 1 to 3. That means you come up with a very fundamental research question and you only come up with a solution. At the end of the 4 year, you have a couple of publications. And this is sort of one box, which we call it TRL 1 to 3. And this is the box where I stayed for 10 years. And then I said, but why I cannot do and go towards market, money? And then he said, no, that's not possible because you chose to, you have a skill set that required for TRL 1 to 3, basically. And then I said, no, but this interests me a lot. This interests me a lot. What often happens is people spend their time in this sector and they do not know about it. And often what you see in India, we have two distinct sectors. One is fundamental research and other side is industry. So industry talks about production of how many million tablets we can produce, lakhs tablets per day and things like that. How we can have a formulation at a scale and supply chain and those sort of. Here we talk about a gram or microgram or milligram quantities. Do I have one agent available in the lab or not? So though these two people speak same language, be it Marathi, Hindi, English, they do not understand each other because the missing link is the innovative research or the applied research, as we call it, which is the link between it. And for many time, this is the value of depth. That means the research do fundamental research, the universities, industry do a lot of industrial research to commercialize it, but no one bridges this gap of knowledge to a prototype. So what is TRL? TRL 6 is more coming up with a prototype from a knowledge. And I think this is the real missing gap. And I think the simple solution at this moment is universities should go a little bit towards TRL 5 and industry should come a little bit also towards TRL 5 and they should bridge the gap. Or you create a different sector or university which will bridge the only applied gap. They would say that, okay, guys, we will not do the fundamental research anymore. We would only look at what has been done at a top institute like NIPER or the different departments, and we'll pick up that knowledge and use some of the master students to only do the applied knowledge, because the PhDs have 4 years of time or 3 years or 5 years, but master student has only 1 or 2 years. They do not have enough time to start something fundamental and go really to the top level. They could only start what is available in the literature and they would go to this concept. And I think this is very, very important. Also, what we have to see that the disciplines are mixing. Nowadays, you cannot call someone who's physics or chemistry or biology or math as a person or process, but you talk about really, advanced production technologies or polymer chemistry, where you need a knowledge of more than one discipline and so on. And of course, eventually you have product or production or systems or enablers, and then that translated into money. So what you need now, I think we need to merge a little bit of education, like a master's, PhD, and bachelor students, and R&D of a sector together. And we should, some of the research questions should come from R&D, from industry. And what they should do is they should not just, and of course I understand about the confidentiality and IP and those sort of things, but these sort of constructions are very much possible. The R&D can break down their whole innovation into small bits and pieces and just release one to one university and couple of students to give them assignment, try to see what they come up with. They can also organize this sort of brainstorming session with them to see what sort of ideas they have. Probably the young workforce have a very different ideas. By merging these two, we can make really best use of our resources, which could help us to go to the market again. Of course, when you look at the whole pharma fraternity and sectors, there are multiple stakeholders. Often we do not even recognize them as a stakeholder. There are doctors, professional companies, but also government and labs and universities, but also investor and insurance companies and association. I think each has a say and a way to play. I think this is also time to bring all the stakeholders together to talk about how this industry should go ahead. If you're thinking of starting your own company, and I think that's not a bad idea, I would say, you have to take certain risk. You have to work with certain, we call it business canvas. You know, this is one pager basically. And in one page, you could say, what would be your value proposition, what you're going to propose. Could it be an Ayurvedic formulation? Could it be a technical medicine or whatever you have it? What sort of activities you'll do, with whom you'll partner with, how you're going to structure the cost, and what are your resources, and how you're going to look at your supply chain, and how you're going to have a revenue stream. Is it through licensing, or one-time selling, or retail, or B2B, B2C? To whom you are targeting your product, basically. Of course, again, the focus should be on co-creation. That means the education, research, entrepreneurs, and government has to come together, basically, to think about such a ecosystem. It could be also seen as a technology park, so the institute SNJB can take a lead and say, okay, we have our own knowledge innovation park here, where we try to bring this sort of government, but also the industry and student together. So industry would be very happy because they get some resources for doing their research. But once they are used to with that industry working, the industry could absorb them in a much better way, basically. So we have to think about beyond medicine and drug. That was my message. Of course, that doesn't mean that you should stop thinking about medicine and drug, because that's an I-shaped pillar you have. But to get a T-shape, we really need to think about a very new concept. I could also here appeal to university like SBGM to think about a smart solution, where you mix, if you have a different discipline in your university apart from pharma, then the master students from different disciplines can come together to solve the problem of a society which is related to health and care sector. I will show you some example. I'm associated also with Section University here in Netherlands, and they have this sort of concept. So this is first example, like in Netherlands, as I said, the population is really aging, and hospital, this is the hospital mattress. Inside is foam, and it's really water impermeable polyurethane cover, basically. But what happens because of that, a lot of old people develops decubitus, a pressure ulcer, and it's a category 1, 2, 3, 4, 5, basically. So, and because of that, to develop such pressure ulcer, 1 day of bad sleep or 2 days of bad sleep is enough, basically. So I'm involved in this project myself now. We are leading together with the local 8 to 10 companies. We are completely reinventing the mattress for the hospital. And though Netherlands sounds a small country, but there are at least 2 lakhs such mattresses in the hospitals, basically, and the elderly care houses. And another 2 lakh mattresses are required in home where people above 60 are sleeping, basically. So what we are thinking about a concept where it still remain hydrophobic. So if something spill like saliva, blood, urine, whatever, then it still doesn't impermeate, but it still had a micro climate management. That means the air can go through it, the heat can transfer through it, the moisture can percolate through it, instead of a foam which can accommodate bacteria and viruses for contamination and cross-contamination. You can have a spacer fabric, which is much more easy to compress, basically, and breathable as well. And now you'll be surprised that this product fall under the medical device regulation of Europe, because this product in future is claimed for decubitus 2 prevention mattress as a system. So then you say, where is the medicine? But healing need not to be medicine all the time. And that's one of the message I also wanted to give you here. Another example is, this is heart on chip, basically. And this was done by some of our bachelor master students. And when you look at the project team, you have a biology student and chemistry student and medical laboratory research student and chemistry student. And they come up with a very small microchip, basically, because it can mimic the environment of a heart. and it can be used as a heart model. So basically, if you do, want to do some test or see the effect of your drugs or medicine on it, you could really use this sort of things as an example. But such innovative things, and these products are very fast-paced project of 6 months only. And different discipline of student comes, they get a coach from a university and they get a coach from industry, and they together develop something in this regard. This was another idea of real-time bacteria count in food and nutrition. This is another very big thing, again, coming to resource efficiency, because often you see a lot of supermarkets here have expiry date, and you say, oh, this paneer was expired yesterday, but should we throw it or is it eatable? But then if you have a scanner or real-time bacteria count, you could really see if the product is consumable or not. And you could prevent a lot of food wastages, because we have a limited resources. We should not unnecessarily eat a day before, but we could really see if this product could be used. And so you need a skill of IT, information technology, but also with bio and lab together to make this sort of ideas to realize it. Another example is, a metastasis on chip. Can you identify if you have cancer cells or some sort of uneven growth of cells in a body? And so what we need is a very quick detection tools. A lot of time the pathology labs have very standard, old-fashioned processes which takes enormous time. But what if doctor could immediately identify the cause and think about from a resource perspective? A villager living in remote Maharashtra comes to Mumbai or Pune or Nashik for treatment. He stays there, he goes to doctor, and after 5 hours of waiting, doctor sees him, and then he writes couple of treatments. Okay, do this test, and then they go to test, and then the pathology say it has to be empty stomach, so they have to stay overnight. Next day morning they do it, and it takes another 48 hours before it comes. Then you have to go with those tests to the doctor again, and so on. So it's such a traumatic process of going through it. That means we need a lot of automation in this regard to speed up. And here the knowledge of pharma or process technology more at a micro or nano scale could be very useful. But such tools could also help our pharma industry to develop their product in a very quick way, basically. This was a project which was related to cirrhosis care, and then we need a student from nursing, but also mechanical engineering and bio to identify the strength of the skin and things like that. So what we are here talking about, very affordable, alternative and innovative, close to home tools, which could help people to detect themselves. Like sugar testing is now at home, blood pressure testing is now at home. A lot of such tests can be, made it available. But this was another, can use textile for healthcare, for making let's say a lab coat, which material would have less resistance to, more resistance to bacteria. I mean, often we have pockets, and when you put your hands in pocket, which material would prevent bacteria growth. I mean, according to one study, most of the hospitals in India, the aprons are not washed, which is supposed to be washed every second or third day. Often they are not washed for months. That means think about how much cross-contamination we have. Often you hear that the patient was admitted to intensive care unit and then he got new infection because the air filter, which is coming from air condition, which is again a textile material for us, a non-woven material, has so much of bacteria and pores living there in that cold area that it infect and bring a new contamination to the patient. And no one realized that sweeping floor or cleaning certain utensils around it will not help to make the whole room bacteria free. You also need to change the non-woven filter, for example, in that regard. And they all fall under the medical device regulation in Europe. And this is completely, so when you talk about medical devices in general, I mean, of course, this is a boring, long definition, which you can read yourself. But in Europe, there are more than 5 lakhs medical devices, we call it, that falls under the MDR rule, medical device regulation rule in the, and it could be as simple as hand gloves or bed or a mask, but they are broadly defined into non-invasive device, invasive device, or active medical device, whether they are placed inside, outside, are they doing something actively, are they more passive, and things like that. In past, the medical device market was close to 50 billion in 2022, and I think it will have a very good jump to 61 billion. And now there is a new medical device regulation, basically, and the old directives will vanish slowly. That means everyone in the market need to resubmit it for this sort of things. And again, it's a long list of things, like you need to reestablish the regulatory status, placing in the market, claims, there will be obligation to manufacturer, representative, importer, distributors, regulator. You should have a legal entity in Europe, whether it's about the, there should be an implant information card if it's an in vitro device and things like that. And you should also have a UDI, unique device identification. Again, it's an IT connection, RFID tags and things like that. So I'm now coming more close to my presentation. That means there are nearly 1 crore students appear for 12th standard examination. We know that. Only at least 10%, 10 lakh deserve the A grade college, but we have a limited IITs. And now it's also the responsibility of a lot of private institute and college to take that prime position through this innovation as a leap. Often private colleges are labeled with low quality but expensive education, and often the industry is asking that when the student comes out of their college at master or bachelor level, they are not ready for industry. Probably some of the people present here would also agree on that. That means they need to relearn. There is a lot of focus on theoretical education. That means we need to bridge this gap with more product oriented or project oriented thing. And this gap of TRL 4, 5, 6, which is applied research, not fundamental research. I mean, if you have your own PhD, that means a lot of knowledge is generating. Can some of this proven knowledge be used to make certain prototypes which can go to industry? And I think this is the area where industry and universities can work together. We need to work more and more on green and yellow ideas, as I talk about, that is TRL 4 to 7. Universities like has great possibility to come up with such knowledge hub or technology park. Or when I say this word knowledge hub or technology park, it need not to be a very complex hi-fi infrastructure. It could be one room of 50 by 50 feet where you have certain possibilities like a brainstorming room, a sketch board, and those sort of things, where people from different disciplines can come together. And every new innovation and ideas you have or your college have, you could display there. So it could also act as an inspiration for it, basically. Think about equipment. If your college has it, then you could see what has been achieved in past through this equipment. You can make such list and place it there. So not everyone has to reinvent it again and again. They would know what are the possibilities with this equipment and what has been achieved. What's also more important is this equipments are up and running. Is there a digital directory of it? You could even make a simple video of each equipment and see if your equipment, how it works, what are the these things to be used, and so on and so on. So we really need to, connect and go beyond that. So we have to, of course, leverage from the government policy. And that's why I try to connect with the mega trends, but what India is looking into 12 top sectors, where our fit comes into it. And then you're suddenly part of the global story because you're fulfilling and ticking all the boxes there. So to conclude, we need to, we have enough resources, materials, knowledge. Naturally, India is very blessed country. I mean, we are all blessed to be born there. But we now need to create connections, like neurons create connections to achieve something. And if we really need to make this leap of innovation, coming up with something unique, then we have to create it. Regulatory, I see, is very important. Regulatory, I see more as a barrier as well for many time to enter in the market. But when you are aware about these sort of things, I think it's more a process which you need to go through. You cannot avoid it. Laws are law, rules are rules, basically. But when you are more aware about what's going around it, then you are much better off prepared for. So thank you for listening. I hope you didn't fall asleep in last, I don't know how long I took, but I'm open for any questions. Thank you. Couldn't hear Dr. Vijay. I think you're muted.
Yeah, I was on mute. So I was having a fan moment, you can say, Doctor. It was really great insight. It was gold, you can say. And when Sanjay approached me, like you have to chair the session, I asked him what I should be doing. So first introduce it and then recap what he said. It's a difficult job. What you presented, Doctor, it's difficult to recap. It's a distillation of what you went through the 20 years.
Exactly.
And we are really fortunate to witness this presentation. And you talked about so many things. It's amazing. It's amazing. And it was all into the figures and the concepts, which I really liked. Everyone liked it. And probably we also need to achieve certain awareness to understand what you presented. And it was good for everyone, like for the faculty members, for the students, where they may feel that, oh, what is there in the pharmacy, and I have to do B.Pharm, M.Pharm, PhD, and then I don't know whether I will get a job or not. But I remember my own story. When I completed my PhD, I went to Sun Pharma for interview, and then I can say his name because he's a great personality. Dr. Nitin Dharmadhikari told me, like, you did research in NDDS, and I have to teach you how to make billions of and millions of tablets, which you don't know, obviously.
Yeah.
But that is what you emphasized, like industry orientation is very much important, and the cross functional interaction is very much important. And the faculty here is really wondering whether they are talking to each other or not, and they should really introspect here and go back and see where
just to give you one idea, where I'm now, my home is within 1-2 kilometers. I have University of Twente, where I did my PhD. Yes. This is 270 acres campus, in-house, like IIT. And they have disciplines like management, but mathematics, nanotechnology, but also mechanical engineering, civil engineering. What they did is, in 30 years before, they were standalone buildings. And what they noticed is that the people don't talk to each other. So what they did is they built physical bridge between these buildings. They broke a window and they created a bridge, so you can walk to other building without much effort, you know. And they centralized couple of canteens, that not every building will have a canteen. That means you have to walk to other canteen for having lunch or dinner. That means you're forced to sit with a mechanical engineer coming from a chemical engineering background and eat with him. So you're forced to talk. And when you talk different, and they say, oh, we could really think about
it. One small thing, sir. Sorry for interruption. What you said that all buildings are interconnected, even physically connected to the bridge. So if you talk about, or if you think about research and the idea research, which is benefited to the society. So what we consider as a person from scientific field, all real life problems are interdisciplinary.
Yes.
That means if you think of any pen or any article, that requires a design scientist, that requires a polymer scientist, that requires a biological scientist, that requires a pharmacist in some cases, and everybody, and all persons working in a single intervention and producing one kind of a thing which is directly get benefited, having benefit to the society. So that is the thing. That may be the concept behind that, connecting all.
No, but that is the thing. That means when the final product needs a multidisciplinary input, our education should also adopt that. To
Yes.
give you an example, here at bachelor education, nowadays it's a semester pattern for everyone. You know, in my time we had a yearly pattern. We used to
Yeah.
only appear for one exam at the year, but now we have a semester pattern. So what they say, out of the 8 semester, the 8 semester will be a research project where the student will do a research project. Semester 6 and 7, one of them student will do multidisciplinary group project. That means there will be a student will opt, you know, industry will give their problems and they will see to solve this problem. We need this skill set. And then student can see all the skill set. So I can show you a screen. You can even get inspiration from it later on. Do you see screen? Do you see it?
Yes.
This is called Section. You can go to section.edu, and it has a smart solution semester. That means in education, every student has to take one multidisciplinary semester. That means a pharma student will work with mechanical engineer or robotics or you know, the hydrophobicity, like your bandages and whatever you could think about it, basically. And industry will give their challenges here. So there is an open platform where industry can give. We have some open assignments. They need not to be a complete project, a part of a little bit project where they need to check the stability or viability of something, or you know, they need to find a new glue, for example, or adhesive. So there is a video. I would mute it, but it's in Dutch. So the renewable energy, the robotics in healthcare, you see, is really making a lot of progress. The clean room chemistry and things like that. So they say that, and material science. So when you all combine together, you can deliver a lot of news. And these are bachelor students basically, and they are forced to take it. Doesn't matter how shitty product they make it, they are forced to take this semester. What happens is they come up with some beautiful prototype, then they display it. But next time, the next smart solution semester becomes even better, basically. And it's becoming better and better. That means once they go to society or industry, they think with a very wider and open approach. That means the textile person can also talk about bacteria, and bio person can also talk about ICT little bit, not, and that allows them to get this you know, a sort of a T-shaped skill, basically.
That's the basic takeaway or the learning lesson which I definitely learn out of your session, sir. I and the horizontal and the T-shape, and everybody should make yourself pharma education as an I, and then put something on the top, which will give you an
for your information, yesterday I gained the digital marketing certificate, you know.
I'm
because
still,
we need to learn to handle data. There's so much of data, and digital marketers know how to make best use of data to make a right data-driven decision, and not decision through gut feeling, you know. So that's also very important in this case.
So just to add to that, I'm pursuing a course from IIFT in supply chain trade logistic.
Exactly, you see.
And the world changed in 2022, and then everything is now making material available from one part of the world.
Exactly, because supply chain has a big pain areas. Yeah.
As a business head, you should understand. So learning never stops. That is what you exemplified and showed in different ways. And the ideas and the presentation was full of very pictorial presentation, which are very easy to understand. And the idea growth, innovative ideas, the green, yellow, blue, that is the quadrants are really useful. I'm just trying to summarize so that
No,
I
but
remember afterwards. So,
that's why I gave also my LinkedIn profile. Please
yes, yes.
feel free to connect me. You know, just
Even
put in
sir,
reference that.
I have something to say here again. This is Dr.
Please.
Shashikant Bagade. I'm working with NMIMS, School of Pharmacy and Technology Management. I even completed my M.Pharm and PhD from Nagpur department. And
Okay.
Chetwal sir is my senior. And
Nice, nice.
the thing what sir has discussed about the entire presentation, your entire session, as if we are sitting in a session that new education policy is going to implement and how. So you are totally tilted towards the new education policy, and definitely it is a must in India. And your presentation is an entire extract what India is needed in future, what type of skill development, what type of orientation to the graduating student is required. So that is a kind of one kind of a coincide with the new education policy. It's a great session.
Thank
And
you.
the demographic dividend part, which is most relevant to here as the students are here.
It's
And
really important, and many people think that it is not our business, so they stay away from it. But this is our business. I think we would be in business only. Rest would be.
Skill development, which is important, and then that puts a lot of positive pressure on the faculty also, because they are also need to be updated and align with the industry.
But I think you could even think about, you're from Sun Pharma, Dr. Vijay, and here is the university. Think about if you could cater some of the small assignments to master students, you know, which are non-confidential in nature a little bit. And of course, you still could sign a joint IP, you know, agreements and things like that. These sort of ideas could be.
Dr. Sandeep.
Yeah, actually, I was about to talk about it. So in India, as rightly said by Pramod, that the requirement, industry requirement, and the student, the gap there we should breathe through the last semester, which should have some alignment with them. In current scenario, there are some institutes which are guiding students who have completed their masters to be industry ready. So that what it means that our academic last years, we should focus on the part that the student who are doing their masters or B Pharma, they should know what is going on in industry and what is the requirement or basic understanding they should have from the college itself.
Sorry to interrupt, but
Denmark, right?
yes, the new education system in India is going on parallel lines, and yes, Dr. Pramod Agrawal's lecture was also suggesting the same. We won't be missing Dr. Pramod Agrawal because he is with us for the whole day, and he will also be judging the evaluation, the presenters who will be presenting in the afternoon session. May I request Dr. Sanjay Patil to present the vote of thanks to the chairperson, today's chairperson, Dr. Vijay Aire, and also to Dr. Pramod Agrawal for enlightening our crowd, enlightening our delegates regarding the policies which are coming now, regarding the horizons which they have to hit.
Thank you very much from my side, sir. Thank you.
Thank you very much, Dr. Pramod Agrawal, sir, for highlighting
Thank you.
It was a good topic, that is how cross-technology innovations plays a very important role for building the ecosystem for delivering the holistic health and care by highlighting different attributes like interdisciplinary or transdisciplinary technology drivers, then different steps in idea innovation to market, by highlighting the different business model and its development, and finally highlighting how to make use of young human resources. So on behalf of team SNJB PharmaCon, I am very thankful for accepting our invitation and deliberating a nice session. Thank you very much, sir. Thank you. I request our technical team to present a certificate as a token of love and affection to Dr. Pramod Agrawal, sir. Sir, I request you to kindly accept it.
Special mention that Dr. Pramod Agrawal, being a busy person, being an entrepreneur, technopreneur, still on one WhatsApp message, he was ready to be available and help us to enlighten our delegates. to be a part of this conference. I thank you wholeheartedly, Dr. Pramod Agrawal, for being a part of
I think
SNJB
now it's
PharmaCon
becoming a little
2022.
formal. I think what I personally feel that after 20 plus years, whatever you have gained, the more older we grow, it's also our responsibility to give back to society, you know. And I think it's a part of that gesture. I try to always available myself for this sort of activity, you know, because this is really, if one person could make use of even one of my learning, might be very useful. So it's all my pleasure, you know. So no thanks.
Definitely, your encouragement will be encashed by the younger, I believe.
Thank you, sir. Thank you.
Okay.
I am also very much thankful to Dr. Vijay Aire sir for chairing the technical session of Dr. Agrawal sir. So on behalf of we all, I am very thankful to you, sir. I request our technical team to present a certificate to Aire sir as a token of love and gratitude. Kindly accept it, sir.
Thank you, Aire sir.
Thank you, sir.
So with this, we are ending the technical session. Now we will move forward to the next session, that is oral presentation sessions. So for that, Vijay, I hand over to the Dr. Chappaliwar sir. Over to you, sir, for further proceedings.
Yes.
Yes, sir.
Today,
Good
I
afternoon.
welcome you for this afternoon session. Everybody is ready with their presentations. This is the day 2 of SNJB PharmaCon 2022. We have got 2 evaluators today. The resource person today, Dr. Pramod Agrawal, has been requested to continue. He has got a lot of experience, a lot of interest also in looking at the progress of science in our country. The budding scientists will be presenting their research and their findings. There are 10 presentations, and the other other evaluator will be introduced to you by Dr. Sanjay Patil.
Thank you, sir. I welcome both the evaluators, Dr. Pramod Agrawal sir, as well as Dr. Sandeep Chauhan sir. Dr. Sandeep Chauhan is currently working as Principal Scientist at Glenmark Pharmaceuticals Limited, Navi Mumbai. Dr. Sandeep has received his PhD in Pharmacy from MS University of Baroda, Vadodara in 2013. He received his Bachelor's of Pharmacy from Amravati University and Masters from Savitribai Phule Pune University, Pune in 2004 and 2006 respectively. Dr. Sandeep is a gold medalist in B.Pharm and qualified GATE with All India rank 67 in 2004. Dr. Sandeep has been involved in industrial research in the field of pharmaceutical technology and foundation development since 2006. His area of specialization includes formulation development of controlled nanosizing approaches in drug delivery systems. Dr. Sandeep has over 12 original research publications and 2 book chapters to his credit. Dr. Sandeep has presented his PhD research work in CSPS, Montreal, Canada, and CRS, Maryland, USA in the year 2011. Dr. Chauhan has more than 15 years of research experience. He has previously worked in many reputed pharmaceutical companies and companies like GlaxoSmithKline, Dr. Reddy's, Watson Pharmaceuticals Limited, etc. And presently is working as Principal Scientist, Glenmark Pharmaceutical Limited, Mumbai. So I welcome Dr. Sandeep Chauhan for evaluating the oral presentations of today's session. I also welcome Dr. Pramod Agrawal sir for the same. Welcome to both of you, sir.
Hello.
Thank you. Thank you, sir.
Now hand over session to Professor Manoj Mahajan for further proceedings. Over to you, Dr. Mahajan, sir.
Thank you, sir. And a warm welcome to the evaluators, presenters, and delegates for this, the second day of this international conference, SNJB PharmaCon. Today we have 10 presentations. I'll read the names of presenters kindly make sure that all the presenters have joined this meeting. Mr. Asim Kumar Sen, Ms. Dipali Bhandari, Mr. Onoja, Mr. Lokesh Kothari, Ms. Rutuja Natkar, Mr. Sarfaraj Khan, Mr. Siddheshwar Jadhav, Ms. Sneha Duff, Ms. Snehal Mule, and Ms. Priyanka Sasani. Okay. So today's first presentation is by Mr. Ashim Kumar Sen. His title of presentation is Novel HPTLC Approach for Concurrent Estimation of Dapagliflozin, saxagliptin, and metformin hydrochloride in ternary mixture. With the permission of evaluators, we will start with the oral presentation session.
Hello.
Please. Yeah.
Hello. Good afternoon, everyone. Hello.
Yeah.
Hello, sir. Can you hear me?
Yes. You can proceed.
Sir, Ashim here. So I'm going to good afternoon, everyone. I'm going to present particular topic that now I think, sir, video is not coming. I think some technical issues there. I'm not able to switch on my video. So is it possible to proceed with this audio only?
Yeah, please go
some
ahead.
Technical issue. I think one of my friend logged in Rajesh Maheshwari in my computer, so it is same thing. It is coming. It is showing, I think, Rajesh Maheshwari only. I'm not able to see my. Hello.
Please continue, Ashim.
Okay. So my topic of presentation is method. This HPTLC method is related to the development and validation of this method that for
Ashim, there is an echo.
Yes.
Could you use headphone or
Mr. Asim, I think he lost his connection. He's there.
Yes, he is there.
Yes.
probably is trying with headphone on?
Yes. So shall we continue with the next presenter, or we will wait for?
Let's wait for 30 seconds at least. Okay. If he's trying something, then.
Ashim, please unmute yourself.
Hello.
Yeah.
Yes. Sorry for interruption. So I'll start my presentation. That is a method for estimating dapagliflozin, saxagliptin, and metformin in ternary mixture. So I'm not going much into introduction, as all know. It is pharmaceutical analysis, a part of analytical chemistry, where usually dealing with the chemical characterization and mainly used for qualitative and quantitative analysis. And whenever new drugs are coming into the market, there is a need of new methods for estimation of all the drugs. So this is a near combination dapagliflozin, saxagliptin, and metformin. And for that, I have developed a method. And for developing, I have gone through some article. You can see here. So some are that single drug, some are combination drug. But I will try to focus on the last three methods, that is 15, 16, and 17 number, where you can see all the three that articles are related to my drug only directly. But first 15 number, you can see capillary electrophoresis, and 16 is LCMS method, and 17 is HPLC and HPTLC. So when I've gone through, when I have started this project, that time methods were not there. Okay, but when I finished my that work, then I am seeing that some methods have been published. So however, my method is comparatively this better, superior, so I will try to focus on that only. So these are some information relating to physicochemical properties of dapagliflozin. That is SGLT2 inhibitor, and next is saxagliptin, which is a DPP4 inhibitor, and metformin is a biguanide. So I want to show. So main aim was that estimation develop a method and validate the method for estimation of dapagliflozin, saxagliptin, and metformin hydrochloride. And for this the study, I have used that pure drug, that dapagliflozin, saxagliptin, and metformin hydrochloride, which I have received from Dalton Pharmacon located in Vadodara. And when I have started my study, that time tablet formulation is not there in India, so I have tried to make a synthetic mixture as it was given in the manufacturers or innovators that data. And different solvent used here, it listed here: methanol, toluene, ethyl acetate, and formic acid. All are analytical grade reagents. The instrument used, you can see here, HPTLC, digital balance, and that ultrasonic cleaner for sonication. HPTLC, that obviously everybody knows, almost all the institutes and all, they are using the CAMAG instrument. So we have used CAMAG, Lehman 5 applicator, WinCat software, and Hamilton syringe. And for digital weighing purpose, we have used that precise digital balance. This is my method. So initially, from literature review, I have chosen the pre-coated silica gel, that silica gel 60 F24, coated with this particular silica gel on aluminum sheet. And the methanol I have chosen as a solvent after solubility checking. And when I have prepared the mix, that solution I have spotted on the plate, and I have taken UV spectra, and based on that overlapping spectra, I have chosen 232 nanometer as the detection wavelength. We can see here the figure why I have chosen 232 nanometer. These are various trials I have performed before that getting the optimum one, which is methanol, toluene, and ethyl acetate formic acid. The ratio is 4, 3, 3, and 0.2 volume by volume by volume. So which gave satisfactory peak and resolution, symmetrical peak and resolution. So I have optimized the mobile phase composition, saturation time, development distance, volume of mobile phase used, detection wavelength, and activation time. And after varying all the parameter, I have selected, and I came to conclusion that this particular that fixed chromatographic condition is optimum for separation of all the three drugs. So this is the mobile phase you can see here, and I have found that dapagliflozin, that RF value of dapagliflozin 0.92, saxagliptin 0.27, and metformin 0.12, and detection wavelength was 232 nanometer. And other parameters already listed here, fixed chromatographic condition. So that is the flowchart diagram, how I have prepared my solution, standard solution, mixed standard solution, and sample solution. So initially I have taken that 1 milligram of dapagliflozin, 0.5 milligram saxagliptin, and 100 milligram metformin together. I have transferred it to a 10 ml volumetric flask, and I have made the volume with methanol up to 10 ml. And then I have applied that 1, 2, 3, 4, and 5 microliter on the TLC plate with the help of Lenomet 5 applicator. And in case of sample solution, that synthetic mixture already I have prepared, and from that synthetic mixture I have that weight that equivalent to 1 mg DAPA, that is dapagliflozin, 0.5 milligram saxagliptin, and 100 milligram of metformin. And I have transferred it to a 10 ml volumetric flask, try to dissolve in methanol, and I have sonicated for 10 minutes, and after that I have filtered through Whatman filter paper. And from that particular solution, resulting solution, I have applied 2.5 microliter on TLC plate and scanned using that CAMAG 3 software, that scan. This is the one standard chromatogram I have kept. So slide should not increase more than 8 to 10, so I try to keep it less. So you can see here metformin, saxagliptin, dapagliflozin, this all the 3 drugs resolved within this particular RF value. And this is the overlay 3D chromatogram of all the 3 drugs, linearity for linearity study. These all are some information relating to method validation parameters. So we can see here that linearity of my developed method was that 100 to 500 microgram per ml, sorry, 100 to 500 nanogram per bar in case of dapagliflozin, and that 50 to 20 nanogram per band was for saxagliptin, and 10,000 to 50,000 nanogram per band for metformin, and correlation coefficient was found to be more than this 0.999. It was almost close to 1, and that regression equation is given here, by which we can find out the unknown concentration. And I have performed precision studies, and for that precision I have performed repeatability study intraday and interday, and after performing that values, you can see here all the values of that repeatability intraday was percentage RSD value was less than 2, which is as per ICH guideline. And in case of accuracy study, I have performed recovery study. So at 50%, 100% or 150% level, where I have spiked that standard, that analyte into the pre-analyzed sample solution and tried to estimate how much it was recovered. And it was found to be close to 100%. That is more than 98%, and maybe maximum it is around 101%. So, and specificity study was performed with the help of that excipients used in the tablet formulation, which I have gathered from that innovators, that list, that leaflet, and which is available online, and whatever they have given as excipient, with the help of that handbook of pharmaceutical excipients, I have tried to prepare a synthetic that mixture, which may be quite similar to the tablet formulation. And for specificity study, I have performed that peak purity checking in instrument itself, and it was found to be that values was found to be more than 999. So we can tell that that method is specific. And we can see here LOD and LOQ values of all the 3 drugs. It was less than this the initial concentration of calibration curve. This is the result of robustness study, where I have varied methanol content in the mobile phase, and not only methanol content, even I have tried to vary a volume of mobile phase, then chamber saturation time, as well as development distance. So when I have changed all the parameter, but that value in terms of RF value and peak area does not change much, or I can tell it was within the specified limit as per ICH guideline, which is less than 2% RSD. And that formulation I have that I didn't had that formulation, marketed formulation. So for that purpose, I have used synthetic mixture, and when I have analyzed that synthetic mixture with the help of my developed method, so I have found that that method is what to tell, it was giving almost nearby to 100% result, which is quite it is okay, this as per this nearby 100%. So by seeing all the values, validation parameters, the linearity range, what I want to conclude is that method is developed, which is developed and it is validated, and this method is superior to existing method, that reported method, in terms of that sensitivity, and in terms of this application. I want to clear why I want to mean that in terms of application. Actually, the method is that reported. In that particular method, they have taken that concentration of all the drugs, similar concentration. But in that condition, we may face some problem while analyzing the formulation. We can see the ratio of the that particular drug in a formulation. Dapagliflozin 10 milligram, saxagliptin 5 milligram, and metformin 1000 milligram. So if we are trying to analyze a sample, we may face some problem in terms of out of range. Means one drug, if you want to keep one drug within the range, another make it outside the range. In that case, if you want to solve the problem, you have to go for this standard addition method. But I have tried to develop a method which does not require standard addition method. We don't have to go for additional steps. So that is the strength of my this method. These are some references, papers I have gone through while performing the method.
Okay.
Thank you. Hello.
Thank you, Asim, for your presentation. Are we allowed to ask questions? How does it go?
Yes, sir.
Please.
I think you showed your result quite systematically. So it's may I know this is a result of how much time you spend on this study? Like it's a master thesis or PhD research or?
Yes, sir. Yes, sir. Actually, I have outsourced this method. Actually, I don't have that HPLC in my institute, so I have went somewhere, some other place to perform that. So it was not possible for me to use that particular instrument for a longer period of time. So, but I have tried to do it 6 to 7 times like that, whatever possible. So if full validation we want to perform as per the guideline, I think it may take at least 300, 400 or 500 applications. I mean, spot you have to apply. So it was not possible for me. So, but I have repeated linearity, precision, whatever possible, I have tried maximum number.
No, but my question is still different. My question is, are you a master student or a PhD student?
It was my PG students only project.
Okay, a PG project, basic. Okay, clear. You mentioned that you in your conclusion that your method is superior to
existing reporting method.
Yeah, illustrated like
yes, sir.
In terms of sensitivity.
Yeah.
And the major advantage of your method is you don't have to go into the standard dilution process.
Yeah.
Basically.
Yes,
could you talk about its effectivity in terms of how quick and how easy to integrate? I mean, what would be the bottleneck? Now, let's say you have developed something new and unique. What are the barriers? So is the existing methods people are more used to? Is it quicker or cheaper, or people are more aware about that, or something else? You understand my question?
Yeah, sir. Whatever available people use that particular method, definitely. This method is not reported. My method, I'm trying to communicate it. So in terms of, I can show you the paper. This published in good journal only, JPC.
No, my question is something different. My question,
yes,
I'm talking from the perspective of acceptance of your method in future. You have published it, now it's peer reviewed, and you have shown that
it is under communication, sir.
It's still under communication. Okay. Clear. Thank you, Ashim. Yeah.
Okay. Thank you very much, sir.
Yeah, Asim.
Yes, sir.
Whether you feel that your development, the result which you shows are really good results, are you really feeling that this method which you develop can replace the existing system in industry? Because I feel that still prefer choice for industry is still HPLC or UPLC.
Definitely, sir.
And how will you say that your method is superior to the existing industrial method?
So I'm not comparing my method with existing industrial.
No, actually, the results are quite good. If I see the results that are quite good, but I still feel that with HPLC method, the results actually
I really don't know, but these results are nearly equivalent to HPLC method. So the result talk that your method is superior, but have you compared with any other method?
Sir, no one can deny that HPLC in some point it is superior compared to HPTLC.
Yeah, that is theoretical understanding, but what I am able to say that your method is really easy to implement. Yes. But whether it is really, because once you compare it with your existing method, HPLC method, then only you will be able to distinguish.
Yes, sir. Yes, sir. Yes, sir. So that I'm able to that compare. That's what I was trying to understand you also. That difference is in that from the existing method is in terms of sensitivity and sample preparation. So in that case, my method is superior. And in terms of I'm comparing with HPLC, but HPLC is better. But sometimes we may get better result in HPTLC also. But reproducibility problem is there always with the HPTLC due to some weather condition, or maybe you are not using SE, or maybe in humid condition result may vary. That everybody, I think all are aware about all this.
Yeah, but if these are available, then definitely needs to prepare to implement it if it has a practical feasibility.
Yes, sir. Yes, sir. That's why nowadays people are looking at HPTLC. Initially, they are not allowing to use HPTLC, but industry people also nowadays, whatever I have that information, are trying to use HPTLC also in industry. So I think results are quite good. And nowadays, so much automation is there in HPTLC. In future, I think definitely people will look into the HPTLC also, along with HPLC.
Okay, that's good work.
Yeah, thank you very much, sir.
I don't have any next question. Pramod?
No, we can move to the next one.
Yeah.
You're muted, Mr. Manoj.
The next presenter with the code P12 is Ms. Dipali Bhandari. Is she there?
Yes, sir. I'm here.
Okay.
I'm audible, sir?
Yes, you are audible.
Okay, yeah.
Is stability testing of hydrochlorothiazide and quinapril hydrochloride in bulk and tablet formulation.
Yes, sir. Yes, sir.
Dipali, you can share your screen.
So is it visible?
Yes, it is visible.
Okay. Good afternoon, all. Today I am here to present my oral presentation on stability testing of hydrochlorothiazide and quinapril hydrochloride in the bulk and tablet formulation from the Department of Pharmaceutical Chemistry from Gokhale Education Society's Dr. MS Gosavi College of Pharmaceutical Education and Research, Nasik. My code is P12. These are the contents of my research topic.
Dipali, sorry to interrupt. Can you please keep your video on?
Yes, sir.
Thank you.
Am I visible?
Yes.
Is it okay, sir?
Yes. Proceed.
Okay. So these are some contents of research topics. That is background, that is the introduction of the topic, methodology, the methods I have applied over here, the findings, results I got, and the conclusion, and the last future scope and the references I have mentioned over here. And lastly, I have published my research work over here in the Asian Journal of Chemistry. So here, okay, ground of that is the introductory part. Hydrochlorothiazide, it is basically the thiazide diuretic, which is official in the IP, EP, and the USP. Quinapril hydrochloride, it is the S inhibitor that is used for the antihypertensive drugs. So this is the combination of hydrochlorothiazide and the quinapril hydrochloride. In the combinations of quinapril, it is of the 10 mg drug, and the hydrochlorothiazide, it is 12.5 mg of the drug in the combination in the market. So this antihypertensive drug I have chosen because this hydrochlorothiazide and the quinapril hydrochloride having the single methods are available, but the multi-component development, it is not over there. So this combination I have selected, but basically my target is here is the stability indicating assay method, that is the SIMS. So stability indicating assay method, everyone know what is the stress degradation and all. So as the in the hypertensive drugs over here, so this seems it is nothing but the, so according to the regulatory ICH guidelines and the regulatory guidelines, the stability indicating method is to distinguish each component from its the active ingredient. So the degradation molecule, it is going to be separated from its active ingredient, and we have to evaluate, or the active ingredient, it is to be, we can say it is the content, it is to be accurately measured over here. That is the stability indicating assay method. So this SIMS, that is the specific and the selective method of the SIMS over here. So I have used the chromatographic techniques over here. So stability indicating assay method we can used by the chromatographic techniques and the hypernated chromatographic and the spectroscopic methods. So I have listed over here the HPLC, GC, HPDLC, capillary electrophoresis, and SFC methods, and the hyphenated techniques like the diode array detector we can use over here, and the HPLC fluorescence detector, GCMS, LCMS. But in my presentations or in a research work, I used RPHPLC method with the UV detector for the stability indicating assay method also. So methodology or the methods I have used over here are the, this is the method which is capable of separating the or resolving any other potential interfering peak, such as the internal standards from the their components or the main peak. So there is the discriminating nature of the method. It indicates the stability indicating as well as the stability specific method. So the method development process I have done with the RPHPLC using the UV detector, and I have done their validations over here. And the main focus, that is the stress testing to achieve the mass balance and the peak purity. So mass balance is nothing but it is the process of adding together the assay value and the we can say the levels of degradation products to see how closely they are add up to the 100%. So that is the mass balance, and we know there are the various difficulties or achieving the mass balance there. So first it is the we can say there is the there is the interactions of the excipients over the main active ingredient. So it is difficult to achieve the mass balance over there. So the multiple degradation products are over there. If there is the loss of the UV chromophore, then it is very difficult to achieve the mass balance over here. Again, the incomplete detections of the some drugs because of the loss of the chromophores and the loss of the volatile degradant products over here. So it is difficult to measure the mass balance, and the peak purity we have to calculate for the stability testings. So after that, I have done the optimization studies for the microwave assisted degradation by the factorial designs. So I have done this stress testing with the conventional method by our refluxing methods for the acid-base degradations, neutral degradations, dry heat degradations, and photolytic degradations. But after conventional method degradations, I have studied and optimized the method by comparing with the microwave assisted techniques by the factorial designs. So this is the methodology over here. So the method development process by the RPHPLC method here, first of all, so my work plan of work and the whatever I have done, that is the selection of the drug. So I have selected the antihypertensive drugs at it is there is no multi-component combinations or the simultaneous estimations over there. So I have selected this drug. So I have procured this drug from the different companies. So I got the SCTZ from the Macloids and the Aurobindo Pharmaceuticals, and quinapril from the Pfizer also. Selection of the solvent and trials, I have taken the different trials for the method development. So finally, I got the solvent as the potassium dihydrogen orthophosphate buffer I have used over here with the proportion of the acetonitrile and the methanol in the proportion of the 4:4:2. And then the pH was measured of the buffer at a pH 4 with the with OPA. I have selected this pH. Then RP HPLC method, whichever I optimized for the method development, I used these parameters and I got the final method development at the flow rate of 1 ml per minute. Wavelength, it is, I overlay these two spectra of the HCTZ and hydrochloride by the UV, and I got the overlay at the 215 nanometer. Sample injector I have selected, it is because according to the literature survey and all, 20 microliter loop, and column selected is the C18 4.6 mm 150 length and the 5 micron diameter. So optimizations, this study of the microwave assisted technique and this chromatographic parameters over here. So the applications of the method to standard mixtures over here, there is no, we can say, changing in the peak area in the solubilities over there and the standard mixture. So there is the applications of this method over here, and I have validated this method and accelerated the stability studies at the 40 degrees Celsius, 75% RH humidity, and the 30 degrees Celsius and 60% RH humidity for the 3 months and 6 months. So over here, the stress testing, the degradation product, whatever I got here. So the degradation, we can say, there is the unlikely to produce this degradation product over here. So it is depend upon the nature of the individual drug substances and the drug, that of the type of the drug product involved in the mixtures. Again, there are some components which are affecting the stress testing, that is the temperature, humidity, oxidations, and photolysis over here. So the evaluation, it is because of the susceptibility of the drug substances across the wide range of the pH values over here. So I have followed this ICH-Q1B guidelines for the stress testings. So these are some findings or the chromatograms I got from the post degradation. So here I have compared this microwave synthesis with the conventional synthesis. In the here you can see the D1, that is the main component, and the D2, that is the degradant I got from this degradations product. So the quinapril acid degradations in the microwave synthesis and the conventional synthesis, I got nearly, it is 20%. By the conventional synthesis, I got the 20% degradation. So mass balance achieved is 78 and the peak purity is 105 at the temperature of 90 to 92 degrees Celsius for the 30 minutes. And as compared to that, the microwave synthesis for the quinapril, I got for a same strength for both conventional and microwave synthesis, that is 0.05 normal HCl, and the mass balance achieved is 85%. So 15% degradation I got from the microwave synthesis, and the peak purity is 97 in within 3 minutes. So HCTZ acid degradations, you can see over here the chromatograms and their degradation, mass balance, and peak purity obtained. Here microwave power 2 level, it is factorial designs I have selected for this 2^2 and 2^3 for the acid base and degradation. I selected the factorial designs of 2^3, that is the, I have changed the time, strength, and their watt, that is the power of which is used for that purpose. Next, here you can see the microwave synthesis and conventional synthesis for the best degradations that I got. It is 15% in the microwave synthesis degradations, and in this conventional synthesis I got the degradations of around 20%. So here microwave synthesis is that, again here I got the 19% of the degradations microwave synthesis, and in the conventional I got the 5% degradations. And here in the quinapril neutral degradations, again mass balance is achieved by the microwave synthesis, it is 106%, and the mass balance for conventional it is 79%. In 1 hour 30 minutes I used 3% of the H2O2 over here for the degradation purpose, and the same for the HCTZ neutral degradation. So next, it is the synthesis, microwave synthesis and conventional method I have applied for the quinapril degradations, H2O2 degradation over here, neutral, and here these are the findings of the quinapril, hydrochloride, and HCTZ for the dry heat degradations. Here D1 and D2. Somehow here the clusters are formed in the dry heat degradations for the conventional method, but in the dry heat microwave synthesis, I got this at a 140 watt, one level, that is 19% degradation over here. So these are the summarizations of the stress degradations over here for the acid, base, oxidative, neutral, and the dry heat degradations. It is a comparison of microwave synthesis with the conventional synthesis for the HCDZ and quinapril. So in between 5 to 30% of the degradation I got for this stress testings. For the microwave synthesis, it is around 16 to 22% for all these conditions parameters. And for the HCTZ synthesis for the conventional, here I got from the 5% to 18% degradation. In the quinapril microwave synthesis, I got 6% to 21% degradation. And in the quinapril conventional synthesis, I got the degradation from the 19% to 28% degradation over here. These are the some findings of the stress degradation. So whatever I have validated the parameters for the HCTZ and quinapril hydrochloride. So the retention time I got from that is the 1.8 for the HCTZ and 5.1 for the quinapril HCl, that is quinapril hydrochloride. So there is the best resolution over there, that is 3.283, and number of theoretical plates from both the drugs I got more than 2000. So the resolutions, tailing factors, linearity and range I got within a specific range. As the combination, it is of quinapril and hydrochloride of 10 mg and 12.5 mg. Linearity in range I have selected is 2.5 to 12.5 microgram per ml and 2 to 10 microgram per ml. So where that is the linearity I got is 0.995 and 0.997. Repeatability percent RSD, it is 2. And intraday percent RSD, it is less than 2 and less than 1.5. Accuracy I got percent recovery 99. Essay, it is in between 99. It is the range is 98 to 102. I got it is 99. LOD, LOQ, and the robustness. And I have changed the flow rate, wavelength, and pH for the robustness for the interday and intraday. So these are the findings of validations. So the conclusion of my topic, it is I focused on the stress degradation as it is the specific for those SIMS, that is the stability indicating assay method. Here is the conventional methods we are generally using, but I have compared over here the microwave synthesis. So it is the time taken, it is less. And from this validation parameters, I conclude that my method is simple, precise, accurate, and very cost-effective method. And the force degradation of this bulk drug, it is not we can say the commercialized over here. So I have tried to do this work on this combination. So conclusion of this, it is carried out to compare force degradation by the conventional method and the microwave assisted technique to find out the degradation and achieve the mass balance and peak purity. And these studies were carried out under a variety of conditions like hydrolysis, oxidations, heat, and photolysis. So in every degradation conditions, parameters were set to obtain between 5 to 30% degradation of hydrochlorothiazide and quinapril hydrochloride. So the future scope of this study, we can isolate and characterize of the degradation product here, and toxicity testing of degradation products, that is the impurity profiling we can do over here. So the method is stability indicating and may be applicable to the routine analysis of HCDZ and quinapril hydrochloride in the tablets. The tablets formulation I have used is the Accuratic, that is of the Pfizer. It is Accuratic. So I used this and done the assay and the stress testing. So there is no official method available for this multi-component combinations for this. And I published my this paper for the in the Asian Journal of Chemistry, which is peer reviewed. So these are my findings and the result of research work. Thank you, sir.
Thank you, Ms. I'd like to request our evaluators to ask the questions to Ms. Dipali.
Yes, sir.
Yeah, Dipali, this is nice work. Is this a PhD work?
Yes, sir.
Is this your PhD work or master's?
Sir, it was master work, still stability indicating as a method and the degradation, but I have done this comparison onwards. I'm pursuing the PhD, but this is not my PhD topic, sir.
Okay, so the mass balance which you displayed here varies from 80, 85 or something below that actually, 85, but yes, what is in the industry accepted mass balance? Do you know?
Sir, it is nearly, it should be nearly equal to 100%. It is add up to 100%. But the force degradation, we have, it is in between 5 to 30%. So it is okay, sir. Means it is up to 85%. It is okay, mass balance.
For post degradation studies, but for drug product, the method should for industry expected is 5% is allowed. Your mass balance should be 95 to 105.
Yes, sir.
So,
Yes, sir. In
okay,
industry,
so
it is.
Yeah, you said that you.
Peak purity we have to achieve here over.
Yes, yes, yes.
Yes, sir.
Yeah. One more thing, you have said that your method is stability indicating.
Yes,
okay.
Sir.
So, do you know how many impurities are reported for both APIs?
No, sir. I don't have any idea. I will check, sir. Yeah.
So you must know that how many impurities are reported, and at least each reported impurity should be you should able to calculate from your method. Then only you can say that your method is stability indicating, because there are many impurities reported for both this API and in industry also. Whenever a method is proposed, if any new article comes later post method validation also, that if our method is not able to identify that impurity, then once again agency asks us to work on your method to identify that impurity also, or otherwise we have to provide 2 methods for same product. So that need to be taken care during
Yes, that's why I yes, sir. That's why I mentioned it in a future scope that we have to do the isolation.
Yeah, so that's why I said that this is a vast area. You have done a lot of work, but there are few things need to be done.
Yes, sir. I came from the industry also, sir. Now I have joined the academic, and I did this peak purity and the mass balance in the Empower. It is easily achieved in the Empower and other HPLC systems using the DAD, that is a diode array detector or PDA detector. So we can easily do this peak purity by the threshold and peak angles over here. So it is not possible on the JASCO or any manual methods. But I will try my best. Yes.
That's good. It's good try. Good work.
Yes. Thank you. Thank you, sir.
Good work, Deepali. Quick question. You mentioned that there is no official method available yet. So,
yes, sir.
So how's the industry coping at this moment without any official method?
So it is not for commercialization.
Okay.
Yeah, I mentioned.
And you mentioned that you obtain a degradation of 5 to 30%, basically, by variety of control conditions like hydrolysis, oxidation, photolysis, and so on. Why you achieve this? Why not more than this or less than this? Why these numbers? You understand my question?
Sir? Yes, sir. I got your question. But this obtained in 5 to 30% and not more than that. Okay.
Yeah.
So it is mentioned in the ICH. It is not mentioned over here that it should be in a 5 to 20% or 5 to 30%. But the degradation, that is stability testing, it is photo stability and all we are dealing with the closure and the container systems. So the degradation should not be more than that. So that is the aspects over here.
Okay, clear. You see, I left pharmacy for a long time, so I might ask some basic question as well. So please forgive me.
Yes.
No, but thank you very much for your.
Yeah, thank you, sir. Yeah.
Thank you, Ms. Deepali, and all the best.
Thank you, sir.
Presenter is Mr. Oje Onoja from Nigeria with code P13. His topic of presentation is ceramide isolated from Tinospora cordifolia with acetylcholinesterase inhibitory activity at molecular docking. Mr. Onoja, is he there? I think he haven't joined the meeting, so we will call the next presenter with code P14, Mr. LP Kothari. Mr. Kothari.
Yes, sir. Am I audible, sir?
Yes, you are audible, and kindly share your presentation.
Yes, sir. Yes, sir.
Dear presenters, kindly make sure that your presentation will finish within 10 minutes, and next 5 minutes are there for discussion.
Is it visible, sir?
Yes, kindly make it in presentation mode.
Okay, sir. Am I audible?
Yes, you are audible.
Okay, sir. Good afternoon to one and all. Myself, Lokesh Kothari. I am giving oral presentation on the topic of evaluation of novel semi-synthetic polymers for the gastroretentive drug delivery systems under the guidance of Atish Mundala, sir. So glance looking towards the contents of my presentation, it mainly includes the introduction, objective, experimental works, result and discussion, conclusion, and references.
Mr. Lokesh, kindly keep your video on while presenting.
Yes, sir. Yes, sir. Is it visible, sir?
Yes.
Okay. In the introductions, we are knowing that for the formulation of various drug delivery systems, we required synthetic, semi-synthetic, and natural types of the polymers. So in case of, as earlier, we are focused on the natural polymers, as these are easily available, less cost, but due to the chances of microbial contamination and stability problems, that these are replaced by the synthetic polymers. But when we started using of synthetic polymers, then again, due to the cost problem, then that particularly due to the toxicity
Lokesh,
problems, these are, yes, sir.
Lokesh, sorry
Yes,
to interrupt you. I still see the first slide and that too not in a presentation mode. Are you still? Could you present?
Slides are not moving.
The slides are not moving.
Just minutes.
You can stop sharing and once again share.
Okay.
Good to go.
Yes.
Yes. Now it's visible and in presentation mode also.
So the in nowadays we are referring to the semi-synthetic polymers. And semi-synthetic polymers, they have gained an interest for the formulation of various drug delivery systems. So main objective of my present research work was to modify a gum using STMP, that is sodium trimetaphosphate, as a cross-linking agent, and also to evaluate modified gum as a suitable release retardant polymers for the formulation of gastroretentive floating drug delivery systems. Apart from this, my objective to use the optimization technique instead of normal trial and error methods, and to search the best formulation, then to study the effect of polymer to polymer ratio and other excipients on the response parameters like duration of buoyancy lag times, and percentage drug release. Then also the objective to compare the formulations, prepared formulations with the natural formulations, then to achieve the site-specific drug delivery systems, to carry out the stability studies of the optimized dosage forms. Next नहीं हो रहा.
Lucas,
We lost your voice.
yeah. Lucas, I think you need to reshare your presentation.
Yes, sir. Is it visible, sir? Hello?
Visible, but not in a presentation mode.
Yeah, you can proceed if you are not able to. Next slide. Are you able to see
Okay, sir.
in slide? Yeah.
Is it visible?
Yes, yes, yes.
Yes,
Go
yes.
ahead.
Okay, so first step that involves the synthesis and characterization of modified gum. So in this particular cases, that in a one beaker, I have taken sodium trimetaphosphate 1 gram, that is dissolved in a 50 ml of 0.1 normal NaOH. Then in another beaker, I have taken 1 gram of modified gum plus 50 ml of water. So this particular reaction mixture was stirred for 2 hours and poured into 5 petri dish and dried at 60 degrees Celsius for 24 hours. Then that particular prepared powders that has been passed through the sieves and was characterized before the preparation of tablets. In a case of characterization, I have gone through the solubility, then I have find out the Car's index, angle of repose, swelling index. So when it shows dissolutions in a water, so and as I dissolve this particular powder in a waters, it swells very well. Then I have done the X-ray diffraction study. So the X-ray XRD spectra of modified gum shows peak at 18 theta and 16 theta. This indicates that the modified gum is dispersed in a amorphous forms. Then that particularly I have gone through the drug excipient compatibility studies. That particularly this consisting of 4 graphs. First one, that is drug. Second one, unmodified drugs. Third one, modified gum. And last one, that is the drug and that physical mixture. So in case of first graph, that particularly the pure gums that are associated with OH groups and COO groups of oleic acid, that is shown in a case of that particular natural gums. Then in case of modified gums, this particularly OH group that has been disappears and the appearance of phosphate 1 and phosphate 2 groups of STMP that confirms the cross-linking reactions. Then that lastly, this particularly the FTIR spectra of the drugs as well as formulations that shows characteristics peaks of both, shows no interactions between drugs and excipients. Then on trial and error basis, I have formulated that particularly gastroretentive floating tablets using that modified gums. Then I have fixed that particular range and then I have applied the factorial design, 3/2 factorial design to these particular formulations. In these 3/2 factorial designs that I have taken 2 factors. First one independent variables and second one dependent variables. So independent variables mainly includes the concentration of modified gum, and second independent variable, concentration of effervescent agent. So levels that I have used in case of concentration of modified gum, 30, 40, 50%. Concentration of effervescent agent that I have used 10, 12.5 and 15, and their effect that may be observed in case of beyond lag time, and percentage drug release up to 12 hours. So preparation of tablets and their evaluation that is to be carried out. Here on a slide shown the formula that consisting of drugs, diltiazem hydrochloride, modified gum, sodium bicarbonates. These tablets are prepared by wet granulation methods. Then after preparation of tablets, I have gone through the evaluation of various parameters that mainly includes the tablet weight, hardness, friability, thickness, drug content uniformity, Beyonce lag time, duration of Beyonce of each of the formulations. Then this is a pictorial representation of in vitro Beyonce study that particularly the tablet that I have dissolved that tablet in a 0.1 normal HCl, and that particularly, sorry for interruption. So that as this particular tablet dissolved in a 0.1 normal HCl, so within a 55 seconds that will get beyond on that particular HCl. Then this particularly in vitro drug release studies that F1 to F9 formulations that consisting of this particular, when I have used the concentration of polymers, that is in a case of F1 to F3 30%. So the tablet that shows the release within a period of 8 hours. Then in case of F4 to F6, when I have increased the concentration of polymers, so these particular formulations shows the drug release within a 12 hours. And lastly, as the concentration increases, so up to 12 hours, they are showing the release of 80%. Then this particular data that is fitted to this particular the design expert software, the trial versions I use, that is 13. So the equation that is given by this particular software, that is in a first dependent variables, full model for Beyonce lag time, full model equation, Beyonce lag time 73.89 plus 30.17 A, the factor that is A is what? Concentration of that particular modified gum. So this minus 17B, second one that is concentration of effervescent agent. So on a Beyonce lag time, that particularly first factor shows positive effect, that is increase in a concentration of gum increases the Beyonce lag time. Second one, that is have negative effect, that is concentration of, as the concentration of effervescent agent is to be increases, that decreases the Beyonce lag time. Then second dependent variables, that is drug release up to 12 hours, Q12 is equal to 7.71 minus 17.91 A plus 1.0 B plus 1.50 AB. So here again, that is contrast, that as the concentration of gum is to be get increased, so that particular drug release is to be get decreased, and as the concentration of Beyonce lag, the effervescent agent is to be increased, so that drug release is to be get fast. Then this software provided the solutions for the optimized batch. So as we have formulated 9 formulations, so among that, whatever the solution that has been given by that software that matches with the formulation F6, and that tablet is to be prepared, and that tablet is compared with a tablet that is prepared with a natural formulation. So what I have obtained in this particular cases, the formulations with a natural polymers rapidly hydrated and forms a weak matrix systems, which is accounted for the burst release as well as rapid release. While in case of cross-linking in a modified gum resulted in a matrix rigidity and thereby slowing the hydration rate of the gum, resulted in a control release of the drugs. That release is to be takes place within a 12 hours. So conclusion, that first, the modification of natural material was successfully done by cross-linking reactions and was confirmed by FTIR spectroscopic studies. When this particular data fitted to various models, so this particular drug release follows the Horsemeyer-Pepper's kinetics with non-Fickian type of diffusions. Stability studies indicate that there were no significant changes in a drug content and dissolution parameter values. The result of comparative study revealed that the gastroretentive floating tablets prepared with modified gums were better in terms of retarding drug release as well as providing proper buoyancy lag time than that of the natural gum. It can thus concluded that the modified gum has a good potential for formulating the gastroretentive drug delivery systems. So lastly, the references.
Yes.
Lokesh?
Yes, sir.
It sounds like a complete story. Is there still some part remaining here, or do you have something to continue this research, or what next? I mean, you have concluded that the gastrointestinal floating tablets could be a better, let's say. And so what next? Is it you use
sir, in
this as a
future,
carrier to carry certain drugs, or like is the study complete in sense, or they're still open?
In this particular presentation that I have used the drug diltiazem hydrochloride and the modified gum, and related with this study that I have just done the in vitro study. Still in vivo studies to be remaining.
Okay. Okay. Okay, Sandeep.
Yeah. Hi, Lokesh.
Yes, sir.
You have said that natural gum polymers are cheaper than the synthetic one. Is that really true?
Sir, natural gums, they are easily available. That's why they are cheap as compared to the synthetic polymers.
But being in industry for last 15 years, I don't think so. Okay, keep it apart. I just intend to know that you have modified the gum and prepared a new excipient. Okay, and you have prepared a good formulation also. So, if you intend to market this excipient which you have prepared newly, okay.
Yes, sir.
So you need to file a DMF, correct?
Yes, sir.
Yeah. So what type of DMF it is required?
It is related with process parameters, sir.
No, what type of filing? DMF filing. What type of DMF filing it will be?
DMF type 5, sir.
No, it's it's not 5. 5 is for the new materials, biological materials. It's type 3.
Okay, sir. 5 types. Okay.
Yeah, there are 5 types. Type 2 is for API and new this. Sorry, and type 3 is for packing material. Type 4 is for excipients.
Yeah. Type 4, sir.
Yeah, type 4 is for excipient. So these are the minimum requirement, and for getting any new excipient which we are using for a regulated market, that need to have a DMF.
Yes, sure, sir.
Yeah.
Without of that, we can't file patent and all that.
Yeah, actually, for that also, this material which you have synthesized, actually in this study, I expect that there are certain studies on the modified gums which we have done. What are its parameters, how it got differentiated with your original gum?
Sir, original gums that when I that prepared the tablets with original gums.
Actually, I am not expecting your evaluation with respect to formulation. I am expecting an evaluation with respect to gum itself. The current gum which you have used as such with the modified gum, what parameter of these gums are get changed? On,
in this,
so actually, every excipient has some COA, and in COA there are certain tests. So, what differences your COA will have compared to the existing?
Sir, in this particular case is that cross-linking type of reaction is to be done. That when I prepared that, so that retard the drug release.
So, what parameter of that excipient it will change?
That OH group of that is replaced, and phosphate group that is to be incorporated.
That is chemical reaction.
Cross-linking.
That's cross-linking.
Increase the matrix rigidity, swelling is to be enhanced.
Yeah. Correct. Do some evaluation with respect to that polymer so that your thesis will get some more weightage, and that parameter which test compared to your original gum. And if we compare with any one synthetic material which is generally used in floating drug, because there are many formulations with deltiazem as a gastro-retentive formulation, so you can definitely get more literature and you can show advantage of your research.
Yes, sir. Sure, sir.
Okay.
Sure, sir.
Thank you.
Thank you, Lokesh, and all the best.
Thank you, sir.
Kindly remember that you are allowed to present for 10 minutes only. The next 5 minutes are for discussion on your presentation and interaction with the evaluators. Even you should make sure that you are using efficient gadgets with strong internet connection. Our next presenter is Miss Rutuja Nat. Is she there? Miss Rutuja Natkar.
Yes, sir, I am here.
Okay. Her presentation code is P15 and she is presenting on eco-friendly microwave assisted co-crystallization of dolutegravir for solubility enhancement. Rutuja, you can share your screen.
Good afternoon, respected dignitaries and all delegators. My name is Rutuja Natkar, and the topic for my presentation is eco-friendly microwave-assisted co-crystallization dolutegravir for solubility enhancement. Plan of work is phase
Excuse
first.
me, Ms.
Literature,
Natkar.
sir. Yes,
Excuse
sir.
me. Please put your slide on slideshow mode. Will you please start this slideshow?
Yes, sir. Yes, sir.
Okay. Okay. You may start.
Plan of work is phase 1 literature survey, selection of drug, selection of co-former. In phase 2 pre-formulation study of dolutegravir. Phase 3 selection of method, preparation of co-crystal, evaluation of co-crystal. Here is introduction. There are more than 80% solid tablet dosage form commercially available in the market. That are from that 40% drug having solubility problem. These drugs follows biopharmaceutical classification system class 2 having low solubility and high permeability. It shows less bioavailability. Co-crystals, according to the US FDA, co-crystals are defined as solid which are crystalline material composed of two or more components in a same crystal lattice in stoichiometric ratio to improve physical properties without altering chemical properties, where component should be solid at a room temperature. Here is a procedure for co-crystallization. Firstly, selection of a target molecule, that is an API. For co-former selection, selection of a method of preparation, then characterization study of newly discovered crystal, and finally co-crystal were prepared. Rationally behind this study is dolutegravir sodium is BCS class 2 drug that is low soluble and high permeable. It is practically insoluble in water. There is a requirement of hydrogen bond donor and acceptor as it leads to co-crystallization. Approach of polymeric amorphous solid dispersion were used, but co-crystallization approach is not. Objective behind this study is to enhance drug solubility by altering solid state properties to increase solubility of poorly water soluble drug dolutegravir to increase the solubility by using suitable co-former, evaluation of co-crystal by using various advanced techniques to develop the crystal of dolutegravir using the suitable co-former. Need behind this study is dolutegravir sodium is a BCS class 2 drug having low solubility, so there is a need to enhance its solubility. Selection of a drug and co-crystal former. Selection of a drug dolutegravir is BCS class 2 and problem associated with can be resolved using a co-crystallization technique. Drug used in treatment of HIV. Selection of a co-crystal former. Co-crystal former are selected on the basis of determination of pKa value and Hansen solubility parameter. Hansen solubility parameter is works on the principle of like dissolves in like, and pKa value should be in between 0 to 3, and Hansen solubility energy difference value it should be within say less than 7. In pre-formulation study, there is a study of melting point. Melting point of procured drug is similarly as that of pure drug. Initial saturation solubility of drug and co-former. Here, drug having very low solubility as after studying it's shown after studying the saturation solubility. UV spectra of dolutegravir at maximum absorption of shown at the wavelength of 259.8 nanometer. And regression coefficient of this is in the calibration curve, regression coefficient is 0.998. In pre-formulation study, here we done FTIR and DSC study. DSC study shows melting point of drug at 196 degrees Celsius. Methodologies were used in co-crystal formation is micro-assisted method, ultrasound-assisted method, solvent evaporation, slurry conversion method, hot melt exclusion method, grinding method. Grinding method are of two types: solid-assisted grinding method and liquid-assisted grinding method. Mainly I used for my study is micro-assisted method. Equal moles of 1:1 drug and co-former mixed with a solvent we are subjected to the microwave irradiation in 30 ml capacity glass tube in a microwave reactor. Each sample was heated at 80 degrees centigrade under a continuous stirring at 600 rpm. Form mixture were later filtered in Wadman filter paper and then taken for the analysis. And next method which I used for my co-crystal development was ultrasound assisted synthesis. Various molar ratios of drug and co-former with a solvent were subjected to the 12 ultrasound pulse for 5 seconds, separated by a gap of 1 second, using a 20 kilohertz high power ultrasound set at a 50% amplitude, delivered using a sonotrode with a tip diameter 13 mm in a jacketed vessel mounted at a 25 degrees centigrade, were checked for the solubility and other analysis. In characterization of co-crystal, melting point of co-crystal prepared by the micro-assisted technique and co-formers selected based on Hansen solubility parameter and pKa value. Next one is melting point of co-crystal prepared by the ultrasound assisted technique and co-former was selected based on the Hansen solubility parameter and pKa required values. This is the FTIR of my formed co-crystals. It shows the H bonded OH group of shows the broadening. It means there is a formation of hydrogen bond. Same as previous slide. In the saturation study of selected co-crystallization, there is the increasing solubility after co-crystal formation as shown in this result. This is the polarized light microscopy result. Evaluation of co-crystal amorphous material with DSC. In the DSC study, we found that there is a decreasing melting point of formed co-crystal. So there is an increasing the solubility of formed co-crystal. of dolutegravir. This is the XRD results. XRD pattern of co-crystal of dolutegravir pattern sodium drug, so that the third product of co-crystal was found. The above peak with highest and largest peaks indicates crystalline, and sharp peak indicate the extent of crystallinity. Summary and conclusion of this research is dolutegravir is BCS class 2nd drug having solubility issue, insoluble in aqueous PS4. It is slightly soluble in PS7.4 phosphate buffer to enhance the solubility of poorly soluble drug by forming a co-crystal. Dolutegravir sodium co-crystal prepared by the micro-assisted technique, ultrasound-assisted technique. The Hansen solubility approach was successful for predicting the dolutegravir sodium co-crystal, but the solubility mostly increased by the co-former selected by the pKa difference. Co-crystal further evaluated through the FTIR DSC XRD shown successful result for the formation of the co-crystal. Significant band broadening in the region of 3,400 and 2,400 per centimeter in FTIR spectra, and characteristics of melting point alteration in the DSC thermogram relieved the hydrogen bonding between the drugs and coformer. These are. Thank you, sir.
Thank you, Rojita, for your presentation. I would you use two methods. One is microwave, another is ultrasound. Could you please one more time specify the ultrasonic parameters?
Within 10 minutes. Yes, sir. Ultrasound probe diameter is 13 mm, and there is a power of ultrasound is 20 kilohertz, and it is kept for the 5 seconds, separated by the 1 second gap.
And these parameters, how did you find it, or did you optimize this?
Sir, I optimize it.
So you vary the kilohertz, the watt, as well as the frequency, time?
Sir, according to time.
But you maintain that 20 kilohertz. That was the only one power was coming from the probe, or?
Yes.
Okay, so you could not fluctuate the power. So you only fluctuate the frequency, basically.
Yes, sir.
Are you aware about the concept of nodes and antinodes in ultrasound? Waves, where you have to hold the. Rujita?
No, sir.
Okay, because the efficiency of ultrasound is, it's a cavitation kind of, and it can speed up certain reaction. In your case, it's a process of co-crystallization, but the distance between the horn and the reaction has to be, it's like a wave, and the place where the two waves meet is called node, and the place where the two waves are opposite with a maximum distance is called antinode. So the distance of horn has a huge influence on the reaction overall. So if I understand, you're doing your PhD, or you did your PhD, or it's a master work, or?
Sir, it's a PG work.
Okay. I don't know if that is still possible, but I would strongly recommend to look into that aspect. If not you, then other student, because that the theory of node and antinode in ultrasonic, and ultrasonic could be a very powerful tool for drug acceleration, synthesis, but even some reactions to, speed up. Yeah, thank you.
Yes, sir.
Yeah. Yeah, hi Rutuja. It's really nice work.
Thank you, sir.
And it is you have done good characterization. But I just intend to know basic thing how co-crystal helps to improve solubility.
Sir, there is a formation of hydrogen bond if co-crystal and coformer, when combined, they forms a bonding which extra bondings, and that bondings are easily break when they comes in contact with solvent.
So that leads to increase in solubility.
Yes, sir.
Okay. No more question.
Thank you, sir.
Yes, sir. Hello. Am I audible?
Yes, sir.
Good afternoon to all. My topic for today's presentation is QBD-based approach to RPHPLC method development and validation of bupivacaine hydrochloride in bulk and in-house developed nanostructure lipid carriers. The objective of study was to focus, that is, on the development of simple, reliable, cost-effective, precise QBD method for estimation of bupivacaine hydrochloride in bulk and in-house developed nanostructure lipid carriers. As nowadays, most of the work is going on the novel drug delivery system, that is nanosponges, nanocarriers, or you can say solid lipid nanocarriers. But it is found that there is no suitable method that is for quantification of these nanoformulations. So my main focus is on development of this SPLC method for nanoformulation. Initially, we have analyzed our method by defining the ATPs, that is analytical target profile, and critical analytical attributes, CAS. Then after that, primary assessment studies were checked using plaquette Berman design. Then further optimization studies were performed by applying box Banken design. Then the column is used, the C18 column is used for all the chromatographic separation methods. Now this is the Ishikawa fishbone diagram, which is used to identify the ATPs. So 5 individual independent factors were selected as ATPs, and their initial assessment was done by using plaquette Berman design, and further optimization was done by using box Banken design. From these critical method parameters were identified, and CAS were checked what is the effect of these critical method parameters on these CAS. CAS were retention time, theoretical plates, and tailing factors. This is the Pareto chart, which indicates the influence of various CMPs on retention time, theoretical plates, and tailing factors. Results: the reverse phase chromatographic separation with a mobile phase containing a mixture of acetonitrile and 0.1% orthophosphoric acid, pH 2.04, in the ratio of 69.45 is to 30.55 with flow rate 0.805 ml per minute, and lambda max was 214 nanometer, and injection volume was of 12 microliter, was developed, and this newly developed method was validated according to the ICH guidelines. These are the result of parameters of validation. The absorbance maxima was 214 nanometer, and mobile phase concentration was acetonitrile is to 0.1% OPA with a pH 2.04 in the ratio of 69.45 is to 30.55. In linearity, we got the R square value of 0.919. The regression equation is y is equal to 37241x plus 60776. The range was in the range of 25 to 80 microgram per ml. LOD was 0.900, LOQ was 2.72, column use is C18, retention time is 2.870, and the flow rate was 0.805 ml. This is the calibration plot of bupivacaine, and these are the chromatograms of bupivacaine at optimized condition with retention time of 2.870, and another one is the chromatogram of bupivacaine in in-house developed nanostructure lipid carriers with retention time of 2.871. Conclusion: the validated method is a unique, simple, accurate, precise, robust method. Then all the validated parameters of developed method were observed within the given specifications, and the developed and validated method is found suitable for routine analysis of bupivacaine in bulk as well as in in-house developed nanostructure lipid carriers. These are the references. Thank you.
Hi, sir.
Hi, sir.
Nice and quick presentation. So you have used QVD approach very well in this presentation. You have used screening as well as full factorial designs. So
Yes, sir. The screening was done by using placket Berman and optimization by using box Blanken design.
yeah. So why you used placket Brahman design for this?
Because sir, placket Berman design is the best design for analyzing the parameters, and it is for 5 factors. We have the 5 individual parameters. So for that, the best suited design was placket Berman design.
Okay. So there during the screening phase, there are various factors which we need to concentrate on. Okay. So screening design has some advantage if there are many factors and we have to select only selected factors for a full factorial design for further studies. Whether only screening design will suffice your purpose?
Because sir, these factors are the method dependent, process dependent, and these only we can choose. We can choose these factors only because, like, if the material we have reported pKa value and all other parameters, purity is reported, so we cannot choose that factors. We can choose only the factors which are process dependent or method dependent.
Okay, that's good.
Sarfaraz, great presentation, on spot, within time, clear.
Yes, sir.
So I really like it. To be honest, I don't have any questions. It's a clear story to me.
Thank you. Thank you very much, sir.
Thank you, Mr. Sarfaraj, and all the best.
Thank you, sir.
Next presentation is by Mr. Siddheshwar Zadav. Is he there? Mr. Siddheshwar Zadav. His
Yes, sir.
presentation is targeted delivery of 5-fluorouracil in breast cancer using hollow nanoparticles. Sarfaraj, kindly keep your device muted. Mr. Sarfaraj, yeah. Okay.
Am I visible, sir?
Yes, yes. Kindly keep your video on.
Yes, yes, sir. Is my slides visible, sir?
Make it in presentation mode. Slideshow.
Yes, sir. So I am Siddheshwar Zadav from Modern College of Pharmacy. The topic of my presentation is the targeted delivery of 5-fluorouracil in breast cancer using mesoporous hollow alumina nanoparticles. So there are many techniques which are available for the anti-cancer therapy, just like radiation therapy, chemotherapy, but they are cytotoxic.
Mr. Siddheshwar, sorry to interrupt. Kindly keep your video on so that evaluators can see you while presenting.
Sir, I am unable.
Please put your camera on. See to the left corner bottom of your screen.
Yes, yes, sir.
Prompts
Am I visible, sir?
start your video.
Yes,
Yes.
yes, yes.
Yeah.
So there are so many mesoporous compounds are available, just like silica, titanium, but we are trying with the mesoporous alumina with a pore size of 2 to 50 nanometer. So the mesoporous alumina have a stable structure and a large surface area, tailored mesoporous size, and have high capacity of entrapment efficiency, and it is biogenic molecules. It shows the delivery system just like host and guest like structure by forming a link between the carboxyl group of compound that we are using HA. So the advantage of mesoporous alumina includes the better physical stability, high entrapment of drug, control particle size, then efficient carrier system, and increase half-life of extended release of drug. Then small size of pores ensures better encapsulation, and it is a highly efficient carrier. Then our aim and objective is to synthesize the mesoporous alumina and to synthesize uniform and stable compound. Then we have to obtain a pure and porous alumina with a good morphology. To activate the mesoporous alumina, we are using aptus, that is 3-amino propyl triethoxysilane, and to load the alumina with the anti-cancer drug we are required. So the first step of this is the synthesis of mesoporous alumina, which is done with the help of reflux setting up between the CTAB, that is cetyl trimethyl ammonium bromide, which is used as a surfactant, novel surfactant, and with the in the solvent, in the ethanol as a solvent. And to this solution we are adding the 0.7 gram of urea and 2.7 aluminum chloride, that is a metallic salt precursor, and it is allowed to stir at a magnetic stirrer with the at 70 degrees Celsius with the help of magnetic beads. And to this solution, after half an hour, we are adding 28% 6 ml of ammonia. Then after 2 hours, we are put this solution for the aging for the 3 to 6 hours. The aging improves the gel formation in the our compound. And after that, we are removing this solvent with the help of hot plate at 100 degrees Celsius for 6 to 7 hours. We will get the powder. We will get the pellets like form of that compound, and we are calcinating it with the 700 degrees Celsius up to 6 to 7 hours. Then the synthesis of the mesoporous alumina nanoparticle carrier is synthesized. After that, we are functionalizing that compound with the help functionalizing this compound with the help of using a toluene as a solvent. After setting a reflux between toluene and solvent for 3 to 6 hours, we are filter this solution with the help of vacuum, and we are putting this compound for 30 to 40 degrees Celsius for 12 hours. We will get the functionalized compound, and we are after the functionalized compound is getting, we are loading this compound with the help of our drug, that is a 5-fluorouracil, on a magnetic stirrer with the help of methanol as a solvent. And we are after setting a reflux, after doing a stirring of 24 hours, we are filter this compound with the help of vacuum pump, and after we are removing the moisture present in it with the help of oven for half an hour for 30 degrees Celsius. We are calculating the encapsulation efficiency and drug loading of that compound with the help of using this formula. And for this we are taken 10 mg of drug in a 7.4 ml of 10 ml buffer, and allowed to stir for 60 minutes, and then we are centrifuge it for 6000 rpm for 30 minutes, and the supernatant compound is then collected, and we are taking the UV spectrometer. We are calculating this wavelength of 266 nanometer. Then we are calculating the particle size of synthesized mesoporous alumina particle, and then drug loaded compound. We have found the values of 32.7 nanometer and 56.9 nanometer, which are in the range, and increase in the particle size indicates the loading of that compound. We will confirm it by the FTR spectra. Then polydispersibility index is also checked. Then we are doing the scanning electron microscopy, that is same with the help of we found a scrambled strong cluster which shows the formation of smaller particles in our compound in the range of 2 to 50 nanometer. We are doing the EDX with the help of images obtained from the same, and we found the we detect the elements present in it, that is CO, Na, Mg, Al and O is responsible for the detection of the synthesized mesoporous alumina and other compounds which are present in our drug, which also detected. Then here is a FTR spectrum of that all compounds. First is A denotes the fluorouracil, B denotes the synthesized mesoporous alumina, and C denotes the drug-loaded mesoporous alumina particles. Then here is a DSC graph which shows the endothermic peak at 281 degrees Celsius, which is a melting point of 5-fluorouracil, and at a 320 we get a exothermic peak, which is due to the drug-loaded particle. Then we are getting the XRD spectra of that compound synthesized mesoporous, which detects the formation of gamma L2O3. Gamma phase of L2O3 is responsible for the porous and large surface area. Then after the drug loading, we will get this type of peak, which is responsible for the amorphous nature of that compound. Then upon the Bronner based study, that is Bronner image Taylor study, which shows the isotherm and absorption and desorption of nitrogen gas, which shows the formation of porous nature. The type 4 as per the IUPAC type 4 type of nitrogen graph is we have obtained, and we shows the we shows for the mesoporous compound, which in which we will get the history slip type. One history slip is obtained for the cylindrical compounds, and cylindrical nature of our compound is detected based upon the bed BJ's model, that is Barrett Jalon Haldel model. We will get the molecular diameter, that is 7.58954, and the total surface area is 176.089, and total pore volume is 0.43, which are in the range, and the compound is found to be the mesoporous family. Upon the drug release studies, we have obtained the results that the complex we have found, that is mesoporous alumina, and the drug has a greater release pattern than the plain drug. Then upon the in vitro cytotoxicity assay done on the MDMB 234-1 cell lines, that is the MD Anderson metalloblastic breast cancer 231. We have found that the 40 microgram per ml of 5-fluorouracil MA mesoporous alumina concentration is efficient for cytotoxicity. IC50 value is responsible for the inhibitory concentration responsible for the cell death. Then upon the conclusion, upon the results obtained from the IR, SAM, and XRD and DSC, we have found that the successful loading of mesoporous alumina is done, and 5-fluorouracil IMEL complex have reduced the potential to reduce the dose of drug and multi-dose dosing and the potassium targeting ability on the anti-cancer cells. Then we have prepared this compound with the help of Swalgel strategy. And these are the references. Thank you.
Thank you, Kishore.
Yes, sir.
5-fluorouracil mesoporous particles are already reported. How your work differentiate from them?
Sir, we are in a process now by targeting this compound with the help of HA, that is a hyaluronic acid, and targeting this compound on a CD44 receptor, that is cluster determinant receptor on cancer cells.
The targeting work is yet not yet done, not yet initiated.
Yes, sir. I am doing it.
Okay, so have you tested residual solvent for your mesoporous particles?
For what, sir?
Residual solvents. You have used many solvent during preparation of mesoporous particles, correct?
Yes, sir. Just ethanol.
So have you tested residual solvent?
No, No, sir. I have taken it from the other researchers that.
Yeah, actually, you are targeting this using this particle for targeted delivery. So it must be free from the residual solvent which you have used in the synthesis of these particles.
Okay, sir.
Okay, no more question from my side. Good work.
Thank you, sir.
Jadhav, just basic question. You mentioned you use mesoporous alumina. Have you explored other possible option than mesoporous alumina in your literature, or what study says? Why mesoporous alumina alone? Is there a microporous structures which are more surface area probably?
Sir, there is more surface area, but the small particles can easily accumulate and easily can be.
That is clear. That is clear.
Yes,
that
the
is
particle
clear.
size is also responsible for the bioavailability and other parameters.
But what are the, maybe differently, what are the alternatives to mesoporous alumina? If I say, okay, we do not go for mesoporous alumina, what are other possible options than mesoporous alumina?
Then other techniques that like liposome or microporous compounds, but they are different in the sizes, and I am trying to work in a range between 2 to 50 nanometer. This is only possible for the mesoporous compound.
Okay, so you did some sort of analysis where you find different type of materials, and then for your need, this fits the best. That's what you
Yes,
want
sir.
to say.
Yes, sir.
Okay, clear. Thank you.
Thank you, sir.
We could move to the next. Yeah.
Thank you, Siddheshwar.
Thank you, sir.
His eighth presentation is P18 by Miss Sneha Dove. Is she there? Miss Sneha Dove. Miss Sneha Dove. I think she is not there. We'll move to the next presentation, P19 by Miss Snehal Mule. Snehal Mule. Even she is not there. Miss Snehal Mule. Last presentation, P20, Miss Priyanka Sanse. Is she there? Miss Priyanka.
Yes, sir.
Sorry for interruption, sir. We will have to make a call for Miss Ankita Patil too. She has expressed her inability to present yesterday. She was scheduled to present yesterday, but because of some medical problem, she has requested to shift her presentation today. Okay. So we'll have to make a call for her too.
Yes, yes. Once we have finished with the presentation, we will call Miss Ankita.
Hello.
Yes.
Hello, good afternoon, sir.
Good afternoon.
Shall I start, sir?
Yes, yes, you can.
Good afternoon, respected dignitaries. I, Sneha Dove, Assistant Professor at Priyadarshini JL College of Pharmacy, and my presentation code is P18. And my topic is in silico modeling for dual inhibition of dipeptidyl peptidase 4 and xanthine oxidase in diabetes mellitus. Next, introduction. Diabetes mellitus is a life-threatening disorder, and it is characterized by blood glucose level due to insensitivity to the action of insulin. Dipeptidyl peptidase 4 is a serine epoxidase which play an important role in glucose regulation. Xanthine oxidase is an enzyme which converts the hypoxanthine to uric acid to liberate the superoxides. QSARIans is revealing the relationship between the structural properties of chemical compounds with biological activity. Molecular docking is a method which predicts the preferred orientation of one molecule to a second when bound to form a stable complex. The aim is to develop a QSAR model which based on OECD guidelines and implementation of molecular docking studies with least and most active compound. And the objective is to design and develop the safer dual inhibitors for controlling the diabetes, which highlights the structural requirements of molecular properties, and to identify the important descriptors and their explanation as well as interpretation in terms of structural features. Next, this is the material, methods and material in which in present work, DPP4 and xanthine oxidase inhibitors dataset was selected from ChemBL database for containing 215 compounds of DPP4 and 35 compounds of xanthine oxidase. Their SMILE notation copied and from ChemBL in Excel format, an IC50 value is converted to PIC50 value as it advantage to express the potency of the drug with larger PIC50 value are more potent. Then OpenBabel software used for drawing the chemical structure in which SMILE notations are copy-paste and structures are saved in SDF format, and energy minimization done by using the MMFF94 force field. For pi descriptor calculation, OChem online software is used where SDF format of optimized structure used as input. Then by using the online OChem software, pi descriptor calculated for DPP4 and xanthine oxidase, where 16,380 descriptors calculated for DPP4 and 16,380 descriptors calculated for xanthine oxidase. After the elimination of descriptors, those are nearly constant and highly correlated descriptors. It leads to the 9,980 descriptors for DPP4 and 9,064 descriptors for xanthine oxidase. Then, for development of the model, data set splits into the training and prediction set, in which 152 compounds of DPP4 and xanthine oxidase is in the training set, and 63 compound of DPP4 and 9 compounds of xanthine oxidase, it is in the prediction set. Genetic algorithm module of QSARN utilized for the selection of optimum number and set of descriptors. Training set is used to choose the relevant molecular descriptor and to obtain the model. And prediction set serves only to validate the final QSAR model. This is the model validation, and this is the threshold values of the statistical parameter, where R square stands for the regression coefficient, and it should be greater than 0.6. Q square, it is the cross validated value of R square. It should be greater than 0.5. Q square LMO, it should be greater than 0.6. CCC stands for the concordance correlation coefficient, and it should be greater than 0.8. After that, we design some molecules. In that, compound of DPP4 and xanthine oxidase sorted out on the basis of IC50 values of descriptor as active and inactive compounds. Already reported 15 compounds showed dual inhibitory activity. Taken into consideration, their PIC50 value and pi descriptors are calculated. Comparing that data with reported compounds and finding that compound number 5 selected as an ideal to design the new molecules using topless scheme biostable methods, and some molecules designed randomly. In that, on the basis of that, their descriptor values of xanthine oxidase are at optimum and little bit of modification needed for DPP4. Hence, compound number 5 modified to design the new molecules. And that molecules, this is the compound number 5. And on the basis of the different descriptors value, new 50 molecules are designed. Next, molecular docking, where Pyrex software is used for multiple ligand docking in our protein of interest. In that, we perform a ligand and receptor preparation. In that, we open our X-ray crystal structure of protein, which is taken from protein data bank, having an PDB ID 4A5S and PDB ID 3NVY for xanthine oxidase. Then we load a chain A and remove the water molecule and save in PDB format. Then we load protein and PDB format converted to PDB-QT format, then autodock selected and converted into macromolecules. Then we click on the open babel and inserts the ligand, and energy minimization is done with UFF force field and convert all to autodock ligand, that is PDB-QT format. Then we move on Vina wizards and add macromolecules and click on forward, and all the docking scores save in Excel file. Next, ADME screening. 50 design molecules are preferred for the ADME screening. Swiss ADME, it is used. It is a free web tool to evaluate the pharmacokinetic, drug likeness, and chemistry of all small molecules. 50 design molecules screen for ADME, and it gives the 27 compounds. Then these 27 compounds then sorted out according to PIC50, which have above than 6, and it leads to 16 compound. After that, this 16 compound is screening for toxicity, which gives only 3 compounds. This is the result where QSAR model is developed for DPP4 and xanthine oxidase. This is the fitting criteria of statistical parameter, which shows that regression coefficient is greater than 0.5 or 0.6 for both the model, and R square, that is regression coefficient for adjacent, is also greater than 0.6, and CCC, which stands for the concordance correlation coefficient, is also greater than 0.8 for both the models. Then external validation criteria, where R square is also greater than 0.6 and CCC is also greater than 0.8. For internal validation, also their Q square, that is cross validated of R square, is also greater than 0.6, and CCC also stand greater than 0.8 for model 2, and Q square LMO is also greater than 0.6. Then this is the graphical representation of DPP4 and xanthine oxidase of Williams, showing the Williams plot, which helps to identify the applicability domain of QSAR model. Assessment of applicability domain is important to identify whether the model is able to make prediction within the chemical space or not. If there is any chemical compound which are having the structural features distinct from the rest of the compound in the data set, it will be identified as an outlier. So for DPP4, compound number 208 and 203 is found as an outlier. Here is a screenshot which shows the ADME and toxicity screening, highlighted compound showing the inactive toxicity profile with docking score, PIC50 value, and synthetic accessibility of compound number 8, 9, and 10. This is the docking result for DPP4, where I compared the compound number 8, 9, and 10 with the standard drug for DPP4, that is sitagliptin. The PIC50 value of sitagliptin is 7.744, where compound number 8, 9, and 10 also having the greater PIC50 value than the standard, and docking score is also greater than the standard drug. Next, docking result of the xanthine oxidase, where I consider the standard drug allopurinol, where docking score and PIC50 value is greater than the standard. This is the conclusion that developed models are statistically robust with high external predictive ability. The resulting model used to predict the 50 novel molecules, and QSAR analysis and docking results reveal that compound number 8, 9, and 10 are showing better results than the standard drug by considering the PIC50 value and docking score. Hence, it may be useful drug candidate for the treatment of diabetes and related complications. It also showing molecular docking also showing the amino acid interaction as standard drugs, which can be identified best binding affinity with receptor on the basis of RMSD value. Hence, we concluded that compound number 8, 9, and 10 are safer and effective on the basis of all screening parameters to synthesize dual inhibitors for diabetes mellitus. Thank you.
Thank you, Priyanka. My question is, could you differentiate between compound 8, 9, and 10? Because they all three are good.
Yes, sir.
And which one score higher out of 8, 9, and 10? Which one stands out and why?
So the toxicity screening and ADME screening showing that inactive toxicity profile for both the all the three compounds than the standards. Docking score of all the three compounds is greater than also from the standard drugs for DPP4 and also for the xanthine oxidase.
So basically, in a layman's term, they're all equally good.
Yes, in future we can go for their synthesis, in vitro and in vivo study, and also we can do the modification of all the 15 drugs compounds which is already reported for the dual inhibitory activity of diabetes mellitus.
Now, I think 3 is enough to start with. Okay, if I understand, then all 3 are good. Which one you think would be commercially less expensive to produce or more easy to produce? Could you guess that?
No, sir.
Looking at the structure and complexity, could you make any estimate guess?
Depending upon the amino acid interaction, I will guess compound number 10, it will be easy as it shows the tyrosine and tryptophan as a amino acid interaction with the standards.
Okay, because these things are very important to make little extension to your study, because now you have brought up to the level of knowledge.
Yes,
and
sir.
I think here onward, it's like, could you synthesize in the lab? What makes it relatively easy to produce it? And then think a little bit for the industry in future, because having just functionally good might not be commercially feasible at all, because
Yes.
there is a huge competition in this market, especially
Yes.
anti-diabetic drugs. No, but great presentation.
Thank you,
Priyanka.
sir.
I don't know if Dr. Sandeep has.
Yeah, just I had was one question. So you have identified 3 structure. Have you
Yes.
correlated with the marketed drug, which is the best fit for your identified structure?
I only compare with the standard drugs, sir, for sitagliptin and allopurina. In
But
future, we can also compare with the other standard drugs.
Yeah.
Here we only consider one standard drug for the comparison.
Yeah, actually, from that you will get to know the method of synthesis. And now you are theoretically good to go. But when you start practical work, that literature research will help you to go ahead.
Yes.
Yeah. Anyways, nice presentation. Good.
Thank you, sir.
Thank you.
Thank you, Ms. Neha, and all the
Yes,
best.
sir. Thank you, sir.
Is Ms. Nehal Mule there? Ms. Nehal Mule. P19, Ms. Nehal Mule. Okay. P20, Ms. Priyanka Sanse.
Yes, sir.
Ms. Priyanka, you can share your screen. The topic of presentation is formulation and evaluation of herbal oil for prevent of hair loss and promote its hair growth. Ms. Priyanka, you can share.
Mr. Manoj, I think I got a little confused. The previous presentation was not from Priyanka, isn't it?
Yes, sir. It's not from Priyanka, it's from...
I think I confused, and my apologize to previous speaker. I think Dhanshri, I called her Priyanka twice.
Yeah.
So, but what was her code?
P18 Sneha Duff. P18.
P18. Okay.
Miss Sneha Duff.
Oh, so she was okay. So she's not even Dhanshri because her screen
Yes.
says.
Yes,
so
yes. She may be using some other device. That's why.
Okay, Sneha Duff. Okay, fine. Then it's clear. What's the confusion? Great, thank you.
Miss Priyanka, you can start.
Yeah, Priyanka, start.
Hello,
Yes.
I'm audible, sir.
Yes, yes, you are audible and
My screen is visible.
yes, your screen is visible, but make it in presentation mode.
1 second, sir.
Kindly say if your slides can move.
Sir, my slide is visible.
Yes, there it's visible, but it's not in presentation mode.
Okay, okay.
You, kindly check whether they are moving or not.
Yes, it's visible, sir.
Yes, yes. Now it's.
Can I start? Yes. Good afternoon, everyone. My name is Priyanka Promosananse. Today, my topic is formulation evaluation of herbal hair oil to prevent hair loss and promote its hair growth. Objective is the main objective of this is to develop such herbal oil formulation which can resolve the problem related to the hair fall, and main motto is to promote the hair growth and to provide nourishment and makes hair root strong. We know that somehow real beauty in hair, it is a man as well as woman, but nowadays it is a problem. It is a continuously hair loss problem is continuously occurring in every person. That's why I choose the topic and make the formulation. The aim of my present study involving preparation hair oil using the plant material caused due to the hair loss caused due to the stress, pollution, and hormonal changes and other medical conditions. Distress condition increase the number of man and woman. There is importance to develop therapy treatment of hair oil. The traditional medicine in India acclaim the number of herbal oil for hair growth promotions. We have the found that herbal ingredient use great use for consoling the various plant material like fresh flowers and leaves of hibiscus, aloe vera gel, curry leaves, amla, shikakai powder, brahmi powder, kalonji powder, and bitter leaves, castor oil, sesame oil, mustard oil, and all are the useful for the prevent the hair loss. This formulation content is a coconut oil is a base of base, and the formulation aim to provide the nourishment and smooth roots, prevent the whitening of hair, prevent the split end and promote the fast growth and the such material we have used that in my formulation. Leaves, hibiscus, amla, neem, brahmi, kalonji, and curry leaves. Method it is all the above herbals are collected in local market, and the volume of coconut oil is 800 ml, and 1 gram of each herbal powder take, like fenugreek seed, betel leaves, amla powder, neem powder, brahmi powder, kalonji seeds, rita powder, curry leaves, and 50 and in that we have added 50 ml of mustard oil and 50 ml of sesame oil were mixed and boiled. When this oil is boiled properly and then cool, and then add is the castor oil. It is a method of preparation of my herbal oil, and in that we have checked the parameter like specific gravity, pH, viscosity, acid value, saponification value, and compare with my marketed product. And then we have tested in animal study, and in which that firstly we have taken the skin irritation test in animal. The test was performed in order to irritation effect my preparation formula. So it will observe in 72 hours and check the whether it is a irritation to animal or not, and that is a non-irritation to rat skin. So further we ahead, we have taken a animal like albino vista rat, divided in 3 groups for 3 groups, 4 animal in each group as a and then give the label, control, standard, and sample. It was a in which the initiation pattern we have absorbed the responses and treated in that. Firstly, it is a control in which that the first day, first animal in left side, we have the shave the animal in 2 centimeter area, and we have to use continue apply the control, and 15 days we have checked that this is some hair growth is seen in first sample in 15 days and left side, and then 30 days completely hair is a grow in that control. My control is a coconut oil. Standard is a my marketed preparation in which that first sample, first day, third sample in left side we have shaved that, and then 15 days grow in hair in the pictures, and after the 13 days completely growing. And my third one is my sample, it in which that first day, first sample in right side we have shaved, and then apply my sample in which, and the 15 days result the growing of hair in that picture, and then 30 days it is a completely on the hairs. They concluded means our study we have to conclude that the herbal oils show the in the hair growth as compared market preparation is better or similar. In that ways my all test or test are compared to market is a satisfactory result due to me, and it can be treated and useful for baldness. Thank you. References.
Yeah. Priyanka, can you explain if this research was part of your master study, or what are you doing now?
Masters, not completed master. I'm doing second year M.Pharm.
Okay, thank you. You had 3 groups of let's say this animals, and how
Yes,
many
sir.
animals per group?
Sir, actually we have in 4 animals, but it is a limitation of slide that I am not adding this much more animal images. I'm only first day, 15 day or 13 days. we have taken all days pictures.
Because if you look at it, then first day picture, every animal is shaved, clean shaved.
Yes, yes.
30 day, irrespective of whether it's a control standard, it's full hair. So the only image I have to see the difference is 15 day. It might have been better if you would have put maybe 10 days and 20 days, or 7 days and 14 days and 22 days. So we could get more data points to see, because what you have is more qualitative estimation, because the shape also was not done in a nice circled way, or it's a bit of different from each other. So there are also some error, I would say, in that regard. So you should be a little careful. And apart from just normal photographs from the phone, have you looked at the microstructure of the skin, with not under the microscope, but there are, have you looked those sort of structures?
Yes, sir, but picture not with my picture not added, but we have get photos all the study of animal.
And in your slide, you also mention about your sample values, for example, acid values and saponification. Values. But they are in range, like in market sample, you have everything one constant. And why is that reason? Why it's in a range? Yeah, the previous slide.
Yes, sir. Actually, we have tested. Firstly, we tested my sample. According to reference, and reference is given in range, so we can adding this value in range. But we have testing in animal, so they got values in a specific manner. So we have added in that manner.
Because often for oil, viscosity is very important,
Yes.
And the market hair oil has a viscosity of, let's say, 60. You should also put units there, but the sample oil is 46 to 86. It's really large viscosity difference, isn't it?
Yes.
So these factors matters a lot when you think about commercialization.
Yes, sir. Yes, sir. This factor, we will work on it, that factor further.
Okay. And another question, you talk about your formulation and suddenly you talk about you take this base 80%, 50 ml this. How did you come to this ratio of you would use 50%, 50 ml of mustard oil and this much of, in what proportion or in what ratio you'd mix certain ingredients?
Because we have not getting an extract. We have take directly powder, and powder is concentrated, and this concentrated sufficient for the that much oil. We have tried to 2 or 3 formulation and optimize the batch, and we get this concentration. I have not put it the optimization of batches. I have directly put it my optimized formula.
Okay. And have you also tried little shelf life? Like what happens when you do you see the sedimentation and those sort of things?
Yes, sir. We have getting shelf life, but I'm only for not a 3 months. That is for only on 1 month. We have getting shelf life. Much more stability study. We will furthermore study.
Okay. Okay. You see why I'm asking a lot of questions. Your research is very important to me. I need it. So
yes.
Joke apart. Dr. Sandeep, back to you.
Yeah. Nice work, Priyanka. Just I intend to know you have performed skin irritation test.
Yes,
have you
sir.
referred FDA guidance on that?
Yes, sir. CPCA guideline we have followed because the animal handling from the.
I intend to know the procedure for skin irritation test.
Procedure. We have shaving. Firstly, we have observed the animal in normal way. What is the behavior or that all? What is the weight of that animal? All these conditions are observed, and then we have to perform the skin irritation test in that animal.
I think you might misunderstood my question, because there is a proper guidance given by US FDA on how to perform skin irritation test. Once you have a look on it, you will get to know more, because the products which are there for derma, that we usually perform skin irritation test, and there is a huge guidance and huge data need to be presented in a proper way. So you have just gone on the conclusion of the skin irritation test. So I just intend to know more detail about it. That's why I asked this question. So anyway, your work is good, but just I intend to know one thing. You have mixed so many things in one preparation. Okay. So what about the combination effect of it? Because what happened, positive effect, we can look it in a better way, but there are certain negative effect also if you mix so many things in one formulation. So have you looked at it?
Yes, sir. We have making the polyherbal, but all are a natural ingredient, or all are herbal, not a synthetic manner. And herbal product might be.
Actually, it is a general understanding. It doesn't mean that herbal product don't have any side effect. That's a general understanding. If you deep dive into it, you will get to know what exactly it is.
It could be also not just in terms of chemical antagonism with each other. Not only that, but also physically they can hamper each other's release, or imagine it would trap in a way that the release of active ingredient from one of your most delicate material would be will not release.
Actually, you correctly said before it that even in sedimentation you asked before that also one of the indication how there's the different ingredient when kept together will behave.
Yes, sir. I will study that which is
okay. Anyway, good work.
Herbal product, but herbal is one thing in our mind. It is a natural, so but it is a compatible in all ingredient in coconut oil. That's why I prepare the oil and testing it furthermore.
No, actually, if you look, just going through the conference motto, it's global regulatory challenges. So you should look into the regulatory perspective. All other agencies from except India are very strict in terms of if you have know about nitrosamine impurity. The most selling drug in India was Zantac, and it was suddenly stopped selling it.
Yes.
So just because we Indians usually prefer to ignore things. So that should not be the way, because we believe in that Ayurvedic things are always good in. But that unless we prove it, it is not the fact.
Okay. Yes, sir. Thank you, sir.
Yeah.
For suggestion.
Thank you, Priyanka, and all the best. This is last time I'm announcing the presenters who remained. If they are present, kindly reply promptly. Ms. Snehal Mule with presentation code P19. Is she there?
Thank you.
And Mr. Ponoja, presentation code P13. Ms. Ankita Patil, presentation code P9. Yes, that's all for the presentation session. I'm thankful to all the presenters and especially to the evaluators for this tedious and marathon task to do with patience. Thank you, Dr. Pramod sir and Dr. Sandeep sir. For further proceedings, I'd like to hand over the proceedings to Dr. Sanjay Patil.
Thank you, Dr. Manoj. I am very much thankful to both the evaluators, Dr. Pramod sir as well as Dr. Sandeep sir, for spending your valuable time. For Pramod sir, almost 5 hours he has spent from morning.
Since morning, he is with us.
Yes, he is known for his
Thank you very
studious
much,
nature.
sir. I am also
Thank
very
you,
thankful
Dr. Pramod.
to Dr.
Thank you, Dr.
Sandeep for spending his valuable time for evaluating the today's presentations. So on behalf of team SNJB PharmaCon, thank you very much to both of you. I request our technical team to present the certificates to both of you as a token of love, gratitude.
Kindly accept it, sir.
Thank you. Thanks a lot.
Where is the other? Okay.
Yeah, thank you very much.
Dr. Pramod and Dr. Sandeep, I thank you wholeheartedly for devoting such a time for scientific cause.
Thank you.
Your devotion and your help to our SNJB PharmaCon will go long way for the betterness of research and upcoming students. Thank you, sir.
Maybe I have 1 minute to add something.
Yes, sir.
The presentations were very interesting. For me, it was a little bit homecoming. You know, I started with pharma, but I moved to different fields. I apologize if I didn't ask really critical question everywhere. But what I really liked about is the depth. So this presentation had very good depth, all of them in a way, and they were using quite high-tech instruments. But what I missed a little bit overall is a very good problem definition. What is the problem they are intending to solve? And towards the end, they should say whether they should able to solve this problem or not, or how far they are into that journey. The second thing they should try now little bit, I'm using word little bit because they are still a researcher, to think alike little bit from industry perspective. As a matter of what you could say is they would interview at least 1 or 2 interview from industry people to add a dimension to their research. This will really help to see if some of this research could be, you know, pushed further for any possible commercialization, even possible entrepreneurship. You know, this oil could be a good story where they could create a business out of it if it would have been taken into. This is a low-hanging fruit, but the diversity of presentations is really appreciated. I really like that. Thank you.
Thank you, Doctor.
Thank you,
I
sir.
declare that the scientific session for today, day 2 of SNJB PharmaCon-22 comes to a conclusion. I thank the conductor, Dr. Manoj Mahajan, who has for 3 long hours, he was in one position and was conducting. Along with him, Dr. Sanjay Patil was also available to anchor the day for all the delegates and for the organizers. He himself was instrumental in bringing Dr. Sandeep Chauhan here, and his inputs were very important. I believe that the delegates would be taking a leaf from it, from the suggestions, and would be progressing ahead. Thank you, sir. Over to the coordinators of the program, Dr. Zadav and Dr. Mundala.
Thank you, sir. Thank you very much. We come to the conclusion of the day 2 of SNJB PharmaCon 2022. The outcome of today's sessions were really pleasing, as we could hear Dr. Vivek Borse, who deliberated related to medical devices or in vitro diagnostic kits and the regulatory aspects which are associated with it. Then in second session, we had Mr. Amit Bhopre, who elaborated related related to regulatory guidelines for herbals in India, Europe, and Africa. And the third session was being delivered by Dr. Pramod Agrawal, who spoke regarding cross-technology innovations, building ecosystem for delivering holistic health and care. And I think as a coordinator, we are really happy that we are able to serve the purpose of the theme of this 3 days international conference on global regulatory challenges to seek approvals for ANDAs, medical devices, herbals, and nutraceuticals. Delegates, we are really happy with your presence during the sessions and expect the same tomorrow as well. It's starting at 10 o'clock. There will be and second session will be delivered by Dr. Prasad Thakur Desai from Pune. Tomorrow,
I think there's some technical glitch. Dr. Mundra is not able to communicate. I think he was about to conclude the session and declare the tomorrow's session. Yeah, he is here. He is here.
I'm back. I have shifted to another system. There is some technical glitch. We are not able to overcome those. So sorry. So dear delegates, I hope we are able to serve the purpose of this 3 days international conference on drug regulatory affairs. Tomorrow, again, we will be meeting two eminent personalities online, and we'll be happy to have you all with us at 10 o'clock in the morning. Tomorrow, we'll be having the oral presentation session quite earlier at 1 o'clock, and then we'll be going for valedictory at around 4 o'clock. This is all from our side. Thank you. Thanks a lot. See you tomorrow in the morning at 10 o'clock. The link for the tomorrow's session will be shared on the WhatsApp group. Thank you. Thank you very much.
Thanks a lot.
Thank you.